Okay, I'm going to go ahead and get started. Welcome everyone to day two. I hope you had a great day with us yesterday, or if you're joining us for the first time today, I think you're in for a treat, and I hope you'll hang out with us for most of today. We have some exciting sessions planned. We will be kicking off with session 5 today. We did 4 sessions yesterday, recapped gene therapy advancements, some clinical case studies, as well as insights from molecular studies and mutation analysis in our day yesterday. Today, we'll be focusing on a couple of different topics. We have small and large therapeutics in the first part of the day. Some studies referencing clinical insights and case-based learning in CCDS, and then we'll go into our second poster session and have one on CCDS diagnosis outcomes after that in the afternoon. To wrap up today, we have a really special session. We have an interactive panel. We've titled it Road to Rare Innovating and Delivering Therapies. And joining us on this panel today will be Andrew Steinsapper, Clayton Beard, and Dave Jacoby. The session will be moderated by Heidi and myself. We do expect fully this to be interactive, so please do pop in questions that you'd like to hear everyone answer. In terms of house rules for today, very similar to yesterday, please do post questions in the Q&A boxes on your Zoom panel. If possible, please do specify which speaker the question is addressed to, because we have multiple speakers in the session. We do have some more polls planned today, and I hope to have you guys participate in those. And don't forget to tag us on your preferred social media platform. And with that, I know we're a little bit early, but if we do have our speaker available today, then we can go ahead and get started. It's my pleasure to welcome CARMON. CARMON and her husband live in New York. They have 3 children, and their daughter Elizabeth was diagnosed with GAMT in December of 2023. And CARMON will be sharing their story navigating through CCDS with us today. CARMON, if you're ready, I'm going to stop sharing, and it'd be great if you are able to bring your camera on. And yes, there you are. Welcome, and thanks for spending your morning with us.
Yeah, and hopefully I'm get this right with the video or whatever, the slideshow, I guess. So
yeah, we'll help you.
Okay, so my son said just to swipe it over and it would show. So hopefully I don't like knock myself off, but if I do, I'll get right back on. So, okay, there we go. Well, good morning. My name is CARMON, and
CARMON,
yep.
one moment.
Yep.
I think you have to click the share button. Do you see at the bottom of your Zoom panel, there's a share button?
I don't see that. Let's see
if we can help with that.
How's that?
Yeah, that's perfect.
Okay. Awesome. Okay. Okay, so we'll get started. Like you said, I am CARMON PHILLIPS. My husband Warren and I do live in upstate New York. We have 3 kids. Elizabeth is 31, Daniel's 29, and Stephen is 26. And I'm honored to share our story with you. Let's see. How do I go to the next picture? Do you know? Oh, there. Elizabeth was born in May of 1994. She had porcelain skin, bright red hair. We were infatuated with her. She was our first child and a first grandchild on both sides. So when she wasn't meeting milestones, our doctors just kept telling us, let her cry, let her lay on the floor, etc. At both her 4 and 6 month checkups, our doctor really didn't think anything was wrong, even though I felt like something was off. And then at her 9-month checkup, they knew something wasn't right. And this started our years-long journey of seeing many specialists and doctors doing lots of different types of testing, trying to figure out what was wrong. She did not grow in height, weight, or head circumference for over a year. And at 2 years old, she only weighed about 15 pounds. In June of 95, at the age of 13 months old, we started seeing an endocrinologist who did genetic testing. And although it was inconclusive, it did point to a metabolic disorder. And at that point, she was given the diagnosis of lactic acidosis secondary to mitochondrial myopathy. And we were told then that we were going to get to see how little the medical world knew. She had global developmental delay, was nonverbal. She didn't crawl until she was 2, and she finally started walking around the age of 3. And at 3 is when she started having grand mal seizures. And those were a pretty regular occurrence for many years. We tried many different seizure medications, sometimes being on multiple ones at the same time. And then, for no apparent reason, her seizures seemed to stop when she was 15. Over the years, we've seen many different specialists for mitochondrial disorder. We've been to Rainbow Children's Hospital and Cleveland Clinic, both in Ohio. We have been to Kennedy Krieger in Baltimore, seeing the top doctors in the field. She had a muscle biopsy and lots of blood tests, but never really getting any answers. Just a lot of, though we've come so far, we still don't really know a lot. And other than the seizures, she was pretty healthy for most of her life. She started early intervention at around 2, received speech, OT, PT, ABA, play therapy. She started school at 3. Her receptive language has always seemed to be really high, but her expressive language just was not there. As she grew, so did her frustrations at not being able to communicate, which led to a lot of behavioral issues. Lots of tantruming, throwing herself down, biting herself, stripping off her clothes, throwing whatever she could get her hands on. So at 18, we transitioned her to a residential group home. At 21, she graduated from high school, and she's now involved in a community HAB program and has amazing mentors that are with her every day. So in 2019 is when our journey really took a turn. In December of that year, she ingested Lysol, which caused her to aspirate and put us in the hospital with pneumonia. And around that time, we had started noticing some abnormal mouth and tongue movements, which led doctors to believe that it had something to do with her medication levels. She had been seizure-free for about 10 years at this point, and so we started lowering her seizure medication as they thought it might be causing tardive dyskinesia. And the next few years are kind of a whirlwind. The 2020 lockdown of COVID, we were unable to see her for about 3 months, which caused her to go into a really deep depression. In 2021, she had COVID and then the stomach bug. She aspirated, and that landed us in the hospital once again with double pneumonia. And in 2022, she just seemed to be going downhill physically with no real explanation as to why. Her behaviors became erratic. She was having major mood swings. Her mouth movements became worse, and nothing seemed to be helping. She started having difficulty swallowing, and in March of 2023, we had a swallow study and found out that she was silently aspirating. So we had to start thickening her liquids and pureeing her foods. In June of 2023, the neurologist we were seeing was moving, and because he had always operated under a mitochondrial disorder, he suggested starting her on arginine and CoQ10. He also talked to us about getting genetic testing redone to see if they could pinpoint the mitochondrial issue that she had, so maybe we could treat her better. They had a genetic doctor in-house, and so we agreed. They were doing a mitochondrial genome. September 14th of 2023, she started having seizures. By the end of September, she was stuck in a seizure cycle that was causing her to seize and pass out about every 20 to 30 seconds for just a couple of seconds. She was hospitalized multiple times, one being 11 days in the neuro ICU unit. There she was on 24-hour EEG video monitoring. She was put on 3 different seizure medications. She had a G-tube for nutrition, a PICC line. It was a pretty scary time for us. And the doctors at the hospital could not figure out why she was seizing from every part of her brain. And they started questioning us where we got the mitochondrial diagnosis. I had all of her medical paperwork from when she was little, so I brought it in for them to look over. And one doctor said he could see where they got that diagnosis, but that she wasn't presenting as a mitochondrial patient. In the beginning of October, we got her genetic tests back, and it was negative for a mitochondrial disorder. And we were just flabbergasted, because then what did she have? By November, she had lost 40 pounds. At this point, she was down to about 80 pounds. She was completely bedridden. She had to be carried everywhere and fed. Even sitting up was difficult, as the seizures would make her just slump over. And at this point, she had been in the seizure cycle for over 2 months. The geneticist decided to do a broader test, and in December of 2023, we got the results, and she had GAMT. We realized that the arginine that we had started her on was poisoning her. It was building up GAA in her brain and becoming toxic, and that was why she was seizing so bad. So we stopped the arginine immediately and started the process to get into a metabolic specialist and doing all kinds of research on what this new diagnosis meant. We are very blessed to have a pediatric metabolic specialist here in our town, and she agreed to take Elizabeth on as an emergency case, even though Elizabeth was an adult. And we had our first visit with them in the middle of January 2024. So before starting any supplements, we had to do blood work to get a baseline of her levels, which took a few weeks. She started creatine, ornithine, and sodium benzoate in mid-February of 2024. And at that time, she had absolutely no creatine in her blood level, and her GAA was at 19. The dose had to be small so that her organs didn't fail, and increased every 6 to 8 weeks following blood work to check her levels. She also started a strict low-protein diet. She started getting better. Seizures stopped almost immediately. She started gaining weight back, and her smile returned, and she was able to start walking again. We are now about 19 months into this new season of her life, and it's as if we've been watching a miracle happen in front of us. She's now this happy, bubbly, full of life girl. Her laughter is contagious. She's learning new skills and has a great sense of humor. Although it took over a year for us to get her, she's finally into therapeutic range for her creatine, and she's been there for about 3 months now, 3 to 4 months. And we've gotten her GAA down under 5, and that has also stayed under 5 for about 3 or 4 months. We were able to wean her off 2 of the seizure medications, and also she had been on clonidine since she was a teenager, and we have taken her off of that as well. We did stop taking the sodium benzoate in December, as it was causing like rashes and like almost like burned look on her tongue. So we stopped taking that. So we feel like we're at the beginning of a new journey with a much brighter future. We really aren't sure how much growth she can have, since she was misdiagnosed for many years, but she is doing amazing. Every day she seems to do something new. She seems very happy, very settled emotionally, and seems to be much more aware of her surroundings and just loves being with people. Our hope for her future is that she would be able to communicate verbally. Actually, this past week we went to visit her, and when she saw us, she yelled out what sounded like my mom and dad. And so it wasn't clear, but everyone in the house realized what she had said, and it was definitely a surreal moment, one that we never thought that we'd hear. We are still searching for answers on her abnormal tongue movement and dysphagia, but those seem to be getting better slowly. Her swallowing is still a work in progress, and we hope that her swallowing will eventually return to normal. Other dreams we have for her is that she would be potty trained, so that she would be able to dress herself or do personal hygiene on her own. So the hardest part of this new diagnosis was learning that had we had this testing at birth, her life would have been very different. This disorder was discovered the year that she was born, but it wasn't available for testing until many years later, and at that point, we would not have had any reason to do more testing. But had our neurologist not suggested redoing the genetic testing, we don't think she would have survived the medical crash. So we are thankful that he did and that we did get answers, and her future looks much brighter now. Our hope is in that sharing her story, that it will help other families that are struggling to never give up, but also to the doctors and the scientists that are trying to find a cure, just to keep working towards that. So thank you very much for listening to our story.
Thank you, Carmen.
Yeah.
I love hearing Elizabeth's story, and it's such an inspiration for how important creatine is, and
Mhm.
for that potential for adults, for everyone. And the newborn screening is such a gift, but all of us who have these later diagnoses and these older children with CTD, I just
Mhm.
think Elizabeth is a real beacon of hope, and I appreciate your story.
Thank you.
Yeah. Thanks for sharing your story, Carmen. Would you like to take some questions from folks? I don't know if there are any, but you're here, and I'd love for an opportunity to interact if that's an option. I know I had a
Sure.
question,
Sure.
if that's all right.
Yep.
I was just wondering, you mentioned that you measured GA levels for Elizabeth before you started the creatine, and then you measured it every couple of weeks. Do you have a sense of, like, did those numbers drop pretty quickly? How long? I don't know if you have a sense of how long it took to maybe taper down the GAA levels itself post-treatment.
Yeah, the GAA levels were coming down slowly every time we would have a blood draw done, but it wasn't until probably a year into that the level dropped to under 5, and we were able to keep it there. I think a lot has to do with the diet. She is so restricted still. She no meat, no beans, soy, tofu, whatever, but she's also gluten-free and dairy-free, and so I think that plays a lot into it for her right now. I don't know what it would have been if we hadn't changed the diet so drastically as well, but it definitely, the creatine levels were slowly bumping up as the GAA was slowly coming down, almost in a matched sequence, if that makes sense.
Yeah, yeah. No, that does make sense. Is it it is still, I've understood from speaking to GAMT parents in the community exactly what you said, that the managing with just diet restrictions alone is really tricky.
Mm-hmm.
And also, as the children grow, the amount of supplementation that they need to take is pretty large.
Right.
And I know we heard from Heidi about the amount of creatine. She's been measuring for a while how much they have to
Right.
give in the whole year, which is a significant amount. I mean, yeah, hopefully we will be getting to treatments in the near future. I know there's gene therapy under works, but we're hoping to initiate other kinds of therapies as well.
Right.
Yeah, for researchers in the audience, this is I mean, I think this really illustrates the need for more therapies, ones that are applicable not just to young children at diagnosis, but because there could be adults out there that are, having a later diagnosis in life, and we need medications that will work for them as well.
Right.
Thank you so much for sharing your morning with us,
We
Carmen.
do
Thank
actually
you.
we have a question that came in.
Okay.
Okay.
Carmen, any advice on how to help support staff, such as the group home and community rehab, to understand our children?
So I am very involved in her group home. I have been since we placed her there. I see her every other day pretty much. And it was just really showing them, like, unfortunately for them, they walked through this scary part of it with us. And so they were very eager to learn how to help her and to, help her to heal and get better. And then they've been seeing the slow progress of the new things that she's doing, and it excites them. So I think just the support is we're a team and we work together, and I support them and do my best to help them answer questions or making up menus and, cooking with them, going shopping with them. And we've been very blessed with the staff that she has, that they are like our family. So I don't know if that really answered the question, but
No, that's helpful. There is another one that came in. Um, any encouragement for parents as they look to making a decision of whether or not to go the residential care route? It is so hard.
it's very difficult. That's probably one of the most difficult decisions we ever made. She was turning 18 when we placed her, and I kind of just pretended she was going to college. Um, but I did want her to have a sense of independence, and we were pretty involved with the community at that time as well. And the group home agency had actually reached out to us multiple times because the room was opening for placement, and they were wanting families that would be involved to be in this group home. So the first two times, I think she was 13 and 16 when they offered it, and we rejected it. And then when she was almost 18, I think I was more emotionally ready. But even though it sounds like it's a great thing, it's still really hard. There's a lot of people in and out of the house, and it takes a lot of work to get to know them and just to trust them that they're caring for your children the way that you would want. And you have to stay involved. They never know when I'm popping in. So, and she lives pretty close to us. We can actually see her house from our backyard. So I'm over there a lot, and I think they appreciate that. I think at times she looks at me like, "Great, you're here again." But yeah, so it really is all about networking and staying involved and supporting each other.
Yeah. My pleasure to introduce session 5 for today. Our first speaker in this session will be Alex. While Alex brings up his slides, I'll give you all a brief introduction to him. Alex is a graduate student at Stanford University. He is working in the Montine lab at Stanford School of Medicine. He's used molecular modeling to design novel creatine prodrugs. We learned about some of his work at last year's ACD conference, and he has done further studies and is currently an ACD fellow. Really excited to welcome you to day 2, and the stage is all yours, Alex.
All right, thank you very much for the nice intro. Hope you guys can see my screen. Please let me know if not. But good morning. Thank you guys for your time today. I've been working here for about the last year and a half on this project, and I'm really looking forward to updating you guys on the work we've done in developing a small molecule for creatine transporter deficiency in the Montine lab at Stanford. So today I'm going to hit on 5 points. First, I'll talk about creatine mimetics, and then I'll move on to creatine prodrugs, which are the molecules we're currently researching. Then I'll go into the assay development that we worked on, and then into our experimental results and the preclinical study preparations that we have ongoing now. So first with the creatine mimetics. Maybe just a little bit of background. There's a little bit of chemistry throughout these slides, so I'll try to bridge some gaps here. But creatine mimetics, or mimetics in general, are molecules that are structurally different but functionally active in comparison to the molecule they're sort of designed after. And so, for example, here with the creatine mimetics, the development of these molecules has a goal to enable the creatine molecule, a novel structure that allows it to bypass the blood-brain barrier and enter into neurons independently of the creatine transporter, while also preserving its activity in being phosphorylated by creatine kinase. So one of the best known candidates for this was cyclocreatine. You can see here their structures on the left, and the difference being that cyclocreatine kind of wraps up these two nitrogens here in what's considered the guanidinium group of creatine to give it a little bit more, I always refer to it as greasiness or lipophilicity to bypass membranes more easily. But unfortunately, that molecule in preclinical development had some challenges regarding being able to penetrate the blood-brain barrier, and there were some signs of toxicity. So at the beginning of the project, this is the avenue that we first explored. We designed about 53 creatine mimetics over the course of 7 or 8 months and tested their ability to be phosphorylated by creatine kinase. And unfortunately, we didn't have any success. And so what this taught us was that creatine kinase has a very strict specificity for creatine. And as a result, it precludes our ability to develop mimetics. And so I'll just show you a few of the compounds that we designed here. We actually kind of learned as we were going about this work that these molecules were very weak inhibitors of creatine kinase. So in the molecular modeling work that we did in the lab, that kind of gave us a clue that we were perhaps binding into the creatine kinase pocket, but these molecules were not accepting a phosphate from ATP. And so I just have the creatine molecule in the box up in the right-hand corner here to give you a reference to see that there are some structural similarities in these mimetic designs. Most of them preserve this guanidinium group, and some preserve the carboxylic acid group. But we tried to incorporate a bit of steric hindrance in some of these molecules, a lot of additional carbons for not electrophilicity, but for lipophilicity. And then there's a handful of molecules like the one here that replace the carboxylic acid with what's considered a bioisosteres in this molecule as well. But that didn't work. So we moved on to creatine prodrugs, which is what we've spent a lot of our focus on over the past year and a half. And so for a little bit of background on these molecules, differently than the mimetics, these prodrugs are structurally different and functionally inactive. So I always like to think of prodrugs as a sort of drug structure where there's a scaffold that's attached to your active drug that confers it an ability, some sort of ability. In this case, these prodrug attachments or scaffolds confer the ability of membrane penetration, which is not the case with creatine, why it needs the transporter. And so attaching these scaffolds, we're able to get creatine into the cell, but we need to be able to target some mechanism to remove that scaffold and liberate free creatine. And so in our case, we were interested in looking at a particular amidase enzyme, fatty acid amide hydrolase. And in part, that's actually due to the work that Ultragenyx Pharmaceutical did in identifying FAH as a capable enzyme in liberating creatine from a creatine amide bond. So I just show these amide bonds here in the red boxes where there's this oxygen, carbon, and nitrogen. That's what's considered an amide bond. And the ester over here in the blue box has this oxygen, carbon, oxygen, and there's an attachment, a carbon attachment here at this oxygen. So I refer to these as creatine amides or creatine amide esters for the rest of the presentation. So just to clarify what I'm talking about. And so in our prodrug design, we split our work in our designs into these kind of 3 different categories. They're the creatine amides, the amide esters, and the macrocycles. Again, this is taking a lot of influence from the work from Ultragenyx. But the creatine amides we found, so I should say there's some pros and cons to these molecules. And the creatine amides we found were by far the most stable prodrug design. But the downside is that if you can see here, there's still a free carboxylate anion. So this oxygen carries a negative charge, which is not good for designing compounds that are greasy or membrane penetrable. And so one way to resolve this then is to introduce this ester group here and making this side of the molecule more lipophilic. But as we've come to learn, actually, this is largely what contributes to the instability of these creatine prodrugs. So creatine amide esters and creatine esters, introducing this ester group replaces, for the chemists in the room, that replaces what would otherwise be this resonance that occurs in the carboxyl group, making the carbonyl much more electrophilic. And so these creatine amide esters have much more rapid cyclization into creatinine, which is a byproduct that we no longer can really rescue and liberate free creatine from. And so one way to try to resolve this issue is by making these macrocycles here, where you're essentially taking these carbon tails and wrapping them together and holding this creatine molecule in sort of a hands behind their back position, in a constrained position, so that the intramolecular cyclization is more difficult for the creatine molecule. So there's a bit more stability that comes from this design by means of introducing steric hindrance. So when designing our prodrugs, we, like I mentioned, we wanted to target FAH. And so we learned kind of through this process how to do molecular modeling and get a decent sense and sort of generate ideas about what kind of prodrug designs might be able to liberate free creatine using this enzyme. So this is just a bit of a visual, because I'm a big fan of visuals, of the fatty acid amide hydrolase protein here in gray. And as we zoom in, you'll be able to see, if the video keeps going. Let's see here. You should be able to see, as this video zooms in, the fatty acid amide hydrolase inhibitor. Oh, and hopefully it doesn't buffer me too much. We'll try one more time. Well, sorry about that. The computer seems to not want to show it, but I'd love to show it to you guys some other time if you're interested. Basically, the gist is these fatty acid amide hydrolase proteins, you're able to look at where the molecules bind. And so we use, you can see here in green, this fatty acid amide hydrolase inhibitor, which Pfizer developed and we actually have utilized in our testing. And in these co-crystal structures, you can see where those substrates bind. And so we're able to identify the active site of the protein as a result of this co-crystal structure, and use that active site to dock in our small molecule prodrugs and get a sense of, does this creatine molecule, does this creatine prodrug have similar binding orientation as some of these native substrates and inhibitors. And it's really important that this amide bond that I've mentioned gets very close to this catalytic serine residue in the active site pocket. And so that was sort of a way for us to judge whether or not these prodrugs would hydrolyze, is based on the proximity of that amide bond to this reactive amino acid in the protein. So then for assay development, there were a couple of things that we needed to work on to be able to identify successful delivery of creatine from these prodrugs. And so a lot of work went into the assay development here. So early studies taught us that some of the commercially available creatine detection kits, like the one supplied from Abcam, for example, were not capable of distinguishing between creatine, phosphocreatine, and creatine amide prodrugs. They actually reacted with all three of those, which for us was not necessarily a good thing, because we wanted to detect not only if an amide prodrug was able to get into a cell, but that it was able to release creatine. And so not being able to distinguish between the three was interference, and we were not able to get a clean read on our prodrugs. And so we actually identified ninhydrin which was a small molecule used in the publication from Dr. Schulze's group as a small molecule that is capable of reacting with creatine and not creatine amides. And here I just give a little bit of a schematic on why that is. You see the small molecule ninhydrin up in the top left corner, and in the presence of a base, a strong base like potassium hydroxide, this ring opens, and it'll react with a guanidinium group such that these two nitrogens will form a bond with these two carbonyls, making this ninhydrin creatine fluorescent complex that we can detect. And the reason that we can't distinguish or that we can't measure creatine amide prodrugs is because this nitrogen is already bound, and so it's incapable of forming this complex with ninhydrin. And so in that way, we're able to distinguish the two, which is what we were looking for in an assay. And so this is just to show some of the capabilities of this ninhydrin assay. You can see that among a handful of other guanidinium-containing groups here, we've got phosphocreatine, creatinine, GAA, arginine, and a handful of our creatine amide prodrugs. You can see that ninhydrin really prefers the creatine molecule to produce a strong signal, and that this signal is stable over time. So it makes the assay a fairly easy one to run. And it generates a great linear curve that we can interpret creatine concentrations down to about 500 nanomolar. So now that we have this ninhydrin assay, we wanted to see what's a good cell model for us to screen these creatine prodrugs in. And so we tested a handful, and we found that these HAP1 cells were our best candidates. So these HAP cells are a human haploid cell line, and we actually had a large stock of these in liquid nitrogen from some of the previous studies done in the Montine lab. And just to validate this model, we ran a fairly simple experiment, essentially exposing creatine to either a wild-type cell line or a knockout cell line, and seeing whether or not we could detect an increase in intracellular creatine as a result of this treatment. And we see there's a difference in the genotype that the wild type cells will result when treated with creatine. There will be an increase in intracellular creatine levels, but in the knockouts, there's no increase in those creatine levels. And so this validates the model for us and really sets us up to be able to look through all of our creatine prodrugs. So the results then. So actually first, before we went into these HAP1 cells, we tried just an even more simple experiment where we added these prodrugs into test tubes containing this FAAH protein and incubated them to see if they would behave as substrates and liberate creatine. So we only did these with the creatine amide prodrugs because the molecular modeling suggested that the amide esters and the macrocycles would certainly not be substrates for this protein, and that esterase activity would have to occur in both of those prodrugs first before we were able to test its ability to cleave in the presence of FAH. And so we designed about 30 creatine amide compounds and identified 6 that were capable of being hydrolyzed by FAH in our biochemical assay. You can see that the fluorescent intensity of these prodrugs, which don't react with ninhydrin over time, increases in the presence of FAH, suggesting that there's an increase in creatine that is quantified at 4 hours over in this right-hand graph. And so then we move on to the cell screen. So we designed about I think about 78 at this point creatine prodrugs and had them synthesized. It's this mix of creatine amides, amide esters, and macrocycles. We added the prodrugs to our HAP knockout cells and incubated for 4 hours, then collecting the cells and inspecting the amount of creatine that's in the cell. And from this screen, we saw that there were 5 prodrugs that were capable of delivering creatine to the knockout cells, as you see in the orange off here to the right of the graph. Interestingly, all 5 of these are creatine amides, which were confirmed as substrates for FAH in the previous slide. And so this confirms previous findings, previous work by Ultragenyx, that the creatine amides specifically, and not the amide ester hybrids or the macrocycles, are effective prodrugs in vitro. And so I'll just mention that 8227 is a compound from Ultragenyx. So we're happy to see that we're able to validate their prodrugs, and that these 4 compounds off to the right are novel designs that we plan to iterate on, and I'll mention this here. So then the next experiments, these are actually ongoing. We wanted to get an understanding of the role that FAH plays in converting these creatine prodrugs. And so follow-up experiments have started with this one here that I'm showing, where we co-incubated with this FAH inhibitor to see that if when you reduce the activity of the FAH enzyme, if creatine delivery decreases in parallel. And we actually saw that this did not happen in 4 out of the 5 compounds. And so we're currently trying to rack our brains as to what might be happening here, but we have some ideas and we're going to test them. One of the ideas is that these HAP1 cells, and a bit of a limitation of these cells, is that they have low expression of FAH as compared to the human cerebral cortex. You can see I've pulled the Human Protein Atlas data here just to show the large difference. And so this may suggest actually that what we're seeing in these assays is the activity of other amidases. And so what we want to do then in some follow-up studies is transiently overexpress FAH in these cells to see if when FAH activity increases, if the release of these creatine prodrugs also increases, if the rank ordering of these prodrugs changes around at all, that sort of thing. And in addition to that, we'd like to synthesize derivatives of these 5 hits that we have for a structure-activity relationship investigation. So for example, this compound here, 9310, has a single chiral center. And so when we tested this compound, it was in its racemic mixture. And so we want to separate out the enantiomers. We have a pretty good reason to believe that one of the enantiomers may carry a majority of the efficacy in delivering creatine over the other enantiomer. And some of the structural components of 9310 give us a sort of idea of how to modify these other 3 as well. So now that we've finished with this in vitro study, we're starting to prepare for in vivo work. And so I'm just giving you a bit of an outline. This is actually ongoing right now. So this in vivo study will be separated into 3 phases, I would say. The first is discovering what this maximum tolerated dose is. Then we'll do a pharmacokinetic study, and if successful, a long-term dosing study. So briefly, the maximum tolerated dose study. Each day, the mice will receive an escalatory dose of the compound. And we'll see as we monitor their well-being and the body weight where this maximum tolerated dose, or MTD, resides. And then we'll do a 5-day dose at that dosage to see if it's something that is manageable for the mice. And if that's the case, then we'll test for just a brief 2-hour dosing study. I should mention this is IP dosing that we plan to start with. We'll do a PK study collecting the tissue to quantify creatine in muscle, brain, and blood to see where our compound distributes. If that's successful, then we'll move to long-term dosing and look at some of the behavioral effects of our compound. So we're particularly interested in looking at muscular and cognitive abilities of these mice as dosing goes along. So we'll be looking at the activity chamber, the inverted grid test, and the novel object recognition test. And then in the last week of dosing, we'll switch over to a deuterated prodrug such that we can detect our creatine levels as compared to the creatine that endogenously is present in these mice to get a better sense of the compound's capabilities, quantifying creatine in the same tissues as in the PK study. So in summary, I'll just go back through these 5 points that I have mentioned here. So first, we worked on creatine mimetics, and we learned from that study that the substrate specificity really blocked our abilities to develop a mimetic. And some of us will go back and forth, and there was a bit of a limitation in this approach here that the crystal structures that were available at the time of our work were not ideal, I would say. Maybe not ideal for developing a mimetic. If that's something you guys are interested in talking about, you can email me and I will probably talk your ear off about it. But we tested over 50 and none of them were effective. And so we moved to the creatine prodrugs because we saw groups like Ceres and Ultragenics were having a bit of success there. And they really were able to guide some of the work that we did here and inspire some of the compounds that we designed. And so with the assay development, we were happy to find that ninhydrin was a small molecule that could effectively distinguish between creatine prodrugs and the active creatine molecule in a way that a lot of the commercially available kits could not. We screened over 75 compounds in our HAP cells, finding 5 were effective in delivering creatine to the knockout cells, and that all of 5 were amide compounds. We found that FAH contributes but doesn't fully explain hydrolysis, and there's likely other amidases involved. And so we plan to overexpress FAH and synthesize derivatives for an SAR investigation. And lastly, the in vivo studies are now beginning, so we're really excited. So I'd just like to take a moment to thank my lab. I'm really a product of all of their knowledge, especially Dr. Montine and Al. They've really taken me under their wing. Really, everybody in the lab has, and I've been fortunate beyond words. And I'm also very grateful for ACD, the inspiration that you guys give our lab just by being you, the families, and specifically the organization as well for supporting me as a fellow. So thank you very much. That's all for me.
Thanks, Alex. That was a great talk. Lots of interesting stuff there. Yeah, really nice assay development as well. Stay tuned for the Q&A panel. I'm sure there'll be a number of questions for you. I'll bring up our next speaker, introduce him anyway. John SCHLEBACH is up next. John is an associate professor at Purdue University. His work focuses on the biochemical and biophysical processes governing biosynthesis, folding, misfolding of membrane proteins. He has been focused on the development of precision therapeutics for a number of genetic disorders, including cystic fibrosis, retinitis pigmentosa, as well as cerebral creatine deficiency syndrome. John's lab has been focused on finding pharmacochaperones for SLC6A8, and his talk today will provide an update about that. John, if you're ready, you can bring up your slides.
All right. Can you all see this?
Yeah.
All right. Thank you for the introduction, Sangeetha, and the opportunity to share our work. As some of you may know, I've been working with the foundation for a while, and it's been very rewarding and exciting to see everything that's going on. So one thing I would, Sangeetha touched on this, but just as an FYI, right? Like we do work on many diseases that seem different on the surface, right? Cystic fibrosis is not CTD, but they have similar molecular underpinnings. And so one key goal of our lab is to try to understand what works and what doesn't work for these types of molecular issues, and then can we translate things across fields. So, we work on existing drugs that are on the market for other diseases. We work on cases where there's people making drugs for new diseases. And then this is the one case where we are trying to make the drugs for CTD based on what we know on other things. So with that in mind, I will start with a general overview. Most of you are familiar with the genetic aspects of creatine transporter deficiency, but I'll go through this again just to give you an idea of how I see this problem, right? So, CCDS, creatine deficiency syndromes can arise through multiple genetic mechanisms. One possibility is that you have mutations in AGAT or GAMT that disrupt the production of creatine from arginine and glycine. And as we've heard through this conference, right, there are options to improve the metabolism of individuals that have these classes of mutations and restore or partially restore balance in the metabolome that is responsible for the syndrome. But there's a third class of mutations in the SLC6A creatine transporter gene, CT1, where basically you can try to change metabolic composition or input, but you don't have the transporter to take the creatine into the cell or across the blood-brain barrier, right? And that's a very different problem, right? Because basically, like you can change the diet all you want, but the nutrients don't get where they need to go, right? So what do you do in this situation, right? The great thing about working with ACD is there's many possible strategies you could take. One, you could use a gene therapy to try to replace the creatine enzyme. I'm sure you're hearing about these things. The CTD, let me see one second, let me get a laser pointer. You could try to replace the transporter itself with gene therapy. Alex gave a really nice talk about how you could try to circumvent the need for the transporter and make drugs that go through the membrane and release creatine. There's also people working on nanoparticles to do that. In our case, we want to try to fix the creatine transporter protein with a small molecule class called pharmacochaperones. So if you're thinking about creatine transporter, what do we know about these transporters from a 10,000 foot perspective? First thing I'll tell you is the creatine transporter SLC6A8 is actually one of 19 transporters that have very similar function and mechanism. And we know a lot about these transporters. So this is sort of a map of all the transporters you have in your body. Some of them are specific for things like dopamine or serotonin. Those are actually super major drug targets for things like SSRIs and stimulants, things like that. Creatine transporters over here. It looks like it's distantly related, but actually it's about 45% identical to the neuroepinephrine, dopamine, and serotonin transporters. And since these are major drug targets, we know a lot about how they work and how the drugs influence them. And so from many decades of work on the structure of these things, we have an idea for how these class of transporters work, right? So basically what these do is they take sodium from outside the cell along with a cargo molecule, in this case creatine. Both of those things bind to the transporter. Transporter closes, it flips upside down more or less, and then it releases into the cell the creatine and the sodium. So it's basically using a sodium gradient as a source of energy to push creatine into your cells at really high concentrations. Now, you can bind small molecules to these transporters and lock up this cycle, right? That's what most therapeutics that target these transporters do. They're inhibitors. SSRIs, for instance, are reuptake inhibitors for serotonin. So we obviously are looking for something that restores activity, right? Not something that inhibits the enzyme, but we actually there's evidence that you can have both classes of these things for these enzymes. So we've studied the mutations to figure out specifically what do they do to this cycle or this enzyme to figure out how we can fix the problem, right? One common theme is the mutations compromise the function, but they can do that in multiple ways. And it turns out that most of the mutations we've studied, instead of affecting the function of the creatine transporter at the surface of the cell where it does its job, they actually affect the ability of the cell to produce the transporter early inside the cell before it can ever make it to where it needs to go. So you can see this is some microscopy that we did where we show if you have the wild type sequence of the transporter, you can see the protein is at the surface of the cell where it takes up creatine. But some of these mutations, for instance, G466R, actually the transporter is stuck inside the cell, and the outside of the cell doesn't have that sort of ring of transporter. So the protein can't do its job. Now, in cases like this, in the last decade, there's been a renaissance in the production of drugs called pharmacochaperones, right? And it's a very simple idea. Most of the mutations in creatine transporter and other diseases like this, what they do is where you would have a functional protein or enzyme or transporter, right? The mutations basically prevent that protein from adopting the correct shape in order to have its function, right? And so what pharmacochaperones do is you bind the molecule to the folded protein, and that basically forces the protein to fold up into its correct shape. Now, these are current drugs. This is actually from a review we wrote where we talk about creatine transporter as a target for these types of drugs. This is the drugs that are either in the clinic right now for cystic fibrosis, sickle cell anemia, transthyretin amyloidosis, and Fabry disease, right? These are very different diseases in terms of how they present, but the underlying similarity is they have mutations that causes misfolding, and now we have these small molecules that can restore folding. And so we want to find small molecules that sort of do this for the creatine transporter. Now, luckily, when you think about where do we start with this, if you dig into the other literature on the SLC6 transporter, specifically serotonin, dopamine, and norepinephrine, there's been a curious quirk of pharmacology that others have noticed, specifically involving this drug bupropion, which is Wellbutrin. Wellbutrin is an inhibitor of the serotonin transporter. That's supposed to be how it works. But it is so-called atypical inhibitor. So you can see if you add in bupropion, right, the dopamine uptake goes from 100% down to 0. So this, you add this molecule in, it plugs up the transporter and inhibits its function. However, they also found for dopamine and serotonin both, that at low doses where you don't inhibit function, you actually see this small molecule activates the transporter. Here's a case for dopamine transporter where you have these mutations that cause a very rare type of sleep sickness and a dopamine deficiency syndrome. And you throw in bupropion, and basically you see both more protein makes it to the surface of the cell. That's what you're seeing here for each one of these mutants. You treat with the drug, it goes up. And the silver lining here is even for wild type and for these mutants, you restore function. So this is a counterintuitive aspect of how these drugs work. You throw in an inhibitor, you should inhibit it. But in this case, you throw in a small amount of inhibitor, you get the protein back. Why is that the case? It's because of how the protein is produced in the cell and what happens as a result of this, right? So what we know about the dopamine transporter is most of these things inhibit the enzyme by sticking to the outward-facing conformation that binds to the cargo molecule, in this case dopamine. Bupropion and these other atypicals, what they do is they bind the inward-facing, right? And this actually explains a lot about how they work, because at the surface of the cell where these things function, there's a ton of sodium that basically locks it into the outward-facing conformation, right? But where these things are made in the ER, you don't have that sodium gradient, so the protein is free to adopt whatever conformation it wants. So when you throw in these inhibitors, you'd have to throw in a lot in order to plug up the functional transporter at the surface of the cell, but you'd have to throw in far less to actually stabilize the protein in the ER because you're not competing with sodium. And so we set out to try to make molecules that will select for this structure over this structure in order to get this chaperoning effect in the cell. And so Chuck Kuntz, the staff scientist in my group, set out a few years ago to basically do this using computational drug discovery. And so what he did is he took computational modeling. He modeled the transporter in the outward-facing conformation, which is what you see here. This is a bound creatine molecule. And then he modeled it in the inward-facing conformation using what we know about the other structures, right? This is a strategy that actually for serotonin and dopamine has been working for 20 years to find inhibitors. And more recently, with advances in structural modeling, has proven useful for a wide variety of drug discovery efforts. And so the idea is we have a structural model of the outward, a structural model of the inward. We're going to put a bunch of small molecules into these pockets and find molecules that bind better to the inward-facing conformation to mimic the effects of bupropion on the serotonin transporter. And so we did this. We started with a library of 95 million compounds. We found a few thousand, five or 7,000 compounds that had something that looked like creatine so that it would sit in the enzyme, in the transporter the way that the substrate does. We want to mimic the effect, but we want to find versions of the creatine molecule that will stick selectively to this conformation. And so we found 53 compounds that we could purchase that basically, again, this is basically showing you these compounds prefer to bind to this conformation. That's why they're up here and not down here. And so we took those 53 compounds, we measured whether they affect the amount of creatine transporter, this is wild type transporter, that is produced in HEK293 T cells, which is just a model human cell line. And so in this graph, basically each one of these squares is a different compound we purchased. If the square is white, you see little change. Anything that's red, you see increase in transporter levels. Blue basically means we have less. So if we're looking at the total creatine transporter, you see a lot of these molecules increase the total creatine transporter as we'd expect. So this actually is a really decent hit rate for our molecules we found using computational screening. More importantly, if we look at just what's at the surface of the cell, right, which is where the protein needs to be in order to pump creatine, we found a couple of really strong hits, especially this one right here we call C4, where we got a 60% increase in the wild type transporter at the surface of the cell. So if we look at the structural models, it really sort of shows us how this should work, which is to say you have some elements of the creatine molecule that would fit in the active site of the transporter, but you also have this sort of long lipophilic tail that snakes down into the intracellular permeation pathway and forms extra contacts that will make the drug stick selectively to this compound. We started out with some fluorescent creatine quantification to look at uptake to see, it's great that we're getting more protein, but if it really tightly inhibits the transporter, it's not going to be so useful. And so we looked to see how this affected uptake. We were pleased to find that we did see a bump in transport activity with this fluorescence assay, although the fluorescence assay itself is not super reliable. So rather than doing a bunch more measurements, we've moved on to new functional assays, which I'll tell you about. Because the next step here, this molecule I'm showing you is our top hit. It doesn't really look like creatine, but it does have the right size and hydrophobicity, has a good log P, follows Lipinski's rules. This is a drug-like molecule, but it probably needs optimization in order to increase its potency and selectivity. So the next step is to do structure-activity relationships. But before we do that, right, we have some work to do in order to make sure that we can scale this assay and look at lots of compounds. So the first thing we want to do is find lots of compounds. These are the hits that we found from our first version of the screen that looked promising. We only bought 50 of these, but this is the top, I don't know, a couple hundred compounds. And these are basically projected to show you how similar they are. This is something called a UMAP, which is basically like it's a way of clustering compounds that look the same based on Tanimoto's similarity scores. So you can see we had a fair amount of structural diversity, but after this, we found a few hits where we increased protein levels. We go back to the 95 million compounds, and now we look for things that have structural similarity to our top compounds in order to sort of search a little broader in these areas where we're finding molecules that have the desired activity. So we have a lot of good candidates that we could look at based on our computational docking. Now we need to basically buy the second-gen compounds and look more closely at what they do. To do this, the first step was we wanted to get a really robust way to look at metabolite production or uptake from that changes that occur as a result of these compounds. And so Haritha Manoharan in our lab has been developing a mass spec-based proteomic workflow, which is working really well. It's very simple. You take the cells, you do your uptake assay to measure creatine transporter activity. You extract the metabolites with a very simple protocol involving a methanol extraction. You put them through this amino acid separation column, which seems to work really well for creatine. And then basically, as the metabolites from the cells come off this column, you spray it into this mass spectrometer machine. You look at the masses of the metabolites, and then you break them into pieces and you look at their fragments so that you can identify what's creatine, what's phosphocreatine, what's guanidinoacetate, et cetera. And so this is some of Haritha's first data where she just looked at the precursor size and our ability to separate it coming off the column over time. So you can see here's a few molecules of interest, including creatine, a deuterated creatine that has a mass shift, creatinine, and our inhibitor that we would use in our experiments. And so we can resolve these in terms of when they come off the column, and then if we fragment them, here we're basically showing like the fragmentation products of creatine. And so here's like the main fragments of creatine. There's a peak here at 132. And if we add deuterium heavy hydrogen, so D3 creatine, that will allow us to track uptake and or quantify creatine in cells, you can see this shifts from 132 to 135 because we've added 3 hydrogens. So currently we're trying to expand this assay so that we can not only look at creatine, but hopefully phosphocreatine, guanidinoacetate, et cetera, so that every time we treat with this, we can look at the broader set of metabolites and how they're changing as a result of these compounds. Now, moving forward, right, as we develop this compound, it's not so simple as to say, can we find a molecule that works really well for wild-type creatine transporter? I'm sure that is true. When we look at, for instance, our work on rhodopsin and cystic fibrosis, so retinitis pigmentosa and cystic fibrosis, there's a cautionary tale that to be told here, which is to say these are versions of Pharmacochaperones that we're trying to develop for rhodopsin through a collaboration with Case Western. And here are the clinical variants that cause this. There's 123 prominent variants in the clinic. We have a way that we've developed where we can quickly screen to see how a molecule affects all of the different variants. And the reality is, if you look at the positions of these mutations on the molecule, basically in this case, you throw the molecule in, if it turns red, you've improved that mutant. You can see for the natural cofactor in this case, we see a lot of improvement all over. But for these boutique molecules that we're designing, they seem to fix some mutants and not others. Now, this could be a problem, right? But I would argue the reason why this occurs, and again, we touch on this in this review, is because when people look for these drugs, they tend to look at one variant and try to find compounds that are good at one variant. And they do find those compounds. And then when they look at the other variants, there's problems. So as we develop these, what we're going to do is use our technology in order to find modifications that not only improve potency for one variant, but look at the whole panel as we go to see, can we find molecules that are not only more potent, but fix a lot of variants so that they can benefit the most people. I will give you hope, though. This looks like we're going to find specific molecules, and we probably will. Our hits might be specific, but there's hope here. So in this review, we talk about how these things work. And so I won't get into the nuances of the chemistry here and how this works, but suffice it to say, this basically tells you how much of the creatine transporter would be folded, right? Now, we have every reason to suspect for the wild-type protein, it should be towards the higher end, right? We should get sort of 80 to 100% folding for the wild type protein. A lot of times the mutations push this down the curve to 0, right? And so if we're starting out with something that's already folded, like wild type, right, you can throw in these molecules and they will stabilize the protein and push it up. But there's not a lot of room for improvement because the protein's already mostly folded, right? If you have mutations that destabilize the protein, as you might find in CTD, you start here, it pushes you down the hill, right? You add a small molecule, and what we're looking for is to see the small molecules push it back up the hill, right? And if you have the right amount of change from a particular mutation, the small molecule can do its job. This is why these work for some variants, right? But for other variants, right, that are, for instance, quantitatively have bigger effects, right? It goes down the hill, you add the molecule, it is not enough to push it back up the hill. And this is why a lot of the mutants don't respond. And so this is something that has become abundantly clear in the context of cystic fibrosis. And this is some of our work on cystic fibrosis where we've looked across the variant spectrum at the drugs. And so if you're in the cystic fibrosis community, the #1 treatment right now is something called Trikafta, which is a combination of 3 molecules. And what they found in what we've shown in the case of Trikafta is you have 2 different molecules in that cocktail that both enhance the levels of the protein, in this case CFTR, what you're seeing here. If you do one drug, right, and you look at the variants that respond, the variants that respond here are in blue. And you can see you throw in one drug and you fix some of these variants, but you don't fix all these other ones, right? You throw in another drug and it fixes different variants for reasons that we are actively investigating. But if you throw in both of them, now you're seeing, a lot of most of all of these variants respond. So, based on what we've learned from cystic fibrosis, this is a generalizable principle. What seems to be the winning strategy here is not just putting all your eggs in one basket and getting one winning compound, right? But getting a combination of compounds that hit different sites. And this is why, and the idea here is, again, if you have these really bad mutants, now you add one compound, it's not enough. You add 2 compounds, and now you can start to roll back up the hill, right? Now, moving forward, I think we're really optimistic about this. So this is a work from Janana Therapeutics. They did a very different type of screening than we did. They didn't really use structure. They just did a high throughput screen, and they found these active molecules. Importantly, in their screen, they checked to see whether these molecules inhibit the enzyme, and they only picked the ones that they didn't, right? Which to me says they're actually hitting a different part of the protein than our molecules do. Now, they showed, again, like you would see with any of these molecules, they work better for some variants than they do for others, right? There's some variants down here where they don't see the effect. But for the ones they do see the effect, you actually do see an improvement in the activity of the transporter when you throw in these compounds, right? Now, is my understanding that they had other issues with the behavior of these compounds that were not ideal. But actually, this is a really decent starting point where we could start to make modifications to improve this compound. And then hopefully, what we'll find is if we mix that with our compound, we might have a combination that can fix a lot of different variants, not just the ones that you see here. But it will take a considerable amount of medicinal chemistry. Fortunately, Purdue has a pretty robust medchem community, and maybe within the network we can find other people that would be interested in trying to refine and combine these compounds and look at how they affect all of the variants in terms of their expression, activity, and their improvement of the metabolic profile. And so that's where we're headed with this. I'd really like to thank Sangeetha, Heidi, and the rest of the ACD for their support over the years. This is Chuck Kuntz, who sort of pioneered this project with me. He's done all the computational work along with Haritha Manoharan, who is looking for the second-gen compounds. Jackie Gallagher helped us look at how these affect the expression of the transporter protein. And as I said, Haritha is really pioneering the functional metabolic assays as we speak. And so that's sort of where the project stands and our outlook on how to go about doing this. And so again, thank you for your attention, and I'm happy to address questions in the Q&A.
Thanks so much, John. And thank you for sticking with our community as well. I remember when we first reached out to you several years ago, you had the first paper on SLC, I think one of the papers on SLC68, but it was sort of an offshoot. And I don't know if we knew at that time that you'd be spending so much time on the project, but there have been some really interesting learnings, I would say, in the years past. So thanks for sticking with us.
Great.
Next up, I'll invite our last speaker for this session, SARA BIAGOTTI. SARA is affiliated with the University of Urbino. She's been working on RBCs as a drug delivery system for more than a decade. And more recently, she's moved to RBC-derived extracellular vesicles to deliver RNA molecules. She is the principal investigator of a next-generation EU-funded project named RNA-based therapy by RBCEVs, or RBC-derived extracellular vesicles, for the treatment of GAT deficiency. And so we'll be hearing from Sarah on this topic today.
Thank you, Sangeeta, for your nice presentation. First of all, I'd like to thank the organizer for this opportunity to share our work with you. And here you can find an overview on what we are going to talk today. Firstly, I will briefly introduce you RBCs, red blood cells, and red blood cell derived cellular vesicles as drug delivery systems. Then I will spend 2 words about the methods we use for cargo, for drug loading into red blood cells, and for the production to produce RBCEVs or RBC-derived cellular vesicles from them. Finally, I will show you some in vitro results that have been already published and some other ones that are unpublished about the preliminary results we had in a murine model of GAM deficiencies. Last but not least, we will have, we will have some final consideration, and I will show you what we are going to do in the next future. Let's start with the red blood cells, or RBCs, as I already told. They are naturally born to carry molecules in the body, such as oxygen, carbon dioxide, and so on. However, they can be further engineered to increase the transportability. These are some of the molecules I have personally loaded during my life and comprise both small molecules, and biologics. Small molecules such as immunosuppressive, and anti-inflammatory agents, or biologics such as recombinant proteins, short and long RNAs. Please note that we loaded both the recombinant protein and the messenger RNA coding for the GAMT enzyme. The way to transform an RBC into a carrier is quite simple and is based on the hypotonic dialysis and isotonic resealing principles. The main steps are the following. So we purify RBCs from patient or donor's blood. Then we put our purified RBCs into an hypotonic buffer, during which we have the membrane swelling and the transient opening of pores. The drug is usually put during this step. Then we have the resealing under isotonic condition and the washing to remove the unentrapped drug. And finally, we have our RBCs loaded with the desired drug or cargo. This strategy has been already used in clinics thanks to the setting up of this equipment that is basically a blood washing machine that have been modified to perform the drug loading under sterile and a pyrogenic condition. All this technology has now been acquired by Quint Therapeutics, that is a biotech company based in the USA. RBCs can be used in several ways. For instance, they can be used for the slow delivery of a drug in the circulation. This is the case of dexamethasone. Here you have the pharmacokinetic after an oral administration, while on the right we have the new pharmacokinetic obtained with RBCs that are able to slowly and constantly release the drug for weeks in circulation. This was a very lucky case because it already reached the clinics. And here you can find some references to the last year article that was published on the prestigious Lancet Neurology and to an ongoing phase 3 clinical trial. In addition, we can use RBCs as circulating bioreactors to set up enzyme replacement therapy. This is the case of GAMT enzyme. So we produced a recombinant enzyme, we loaded it into our RBC. In this, the enzyme can work directly inside the cell. And for example, we have the toxic metabolites such as guanidinoacetate that can enter the cell, being metabolized by the recombinant enzyme, and then toxic metabolites such as creatine can be released in the circulation. Actually, in our lab, we produced a mutated form of the GAMT enzyme with a few mutagenic sites to have a more active enzyme. However, in this strategy, we needed to load a second enzyme that is a recombinant form of methionine and adenosyl transferase because the S-adenosyl methionine substrate was limiting in RBCs. Also, these results were published and you can find the references here. A few years ago, we wanted to move from a cell-based to a cell-free strategy. But what does it mean, cell-free strategy? A cell-free strategy can be set up with everything that is produced or released by cells, such as extracellular vesicles. Extracellular vesicles, very briefly, are very small particles surrounded by a lipid bilayer that are released by any kind of cells and that can be taken up by a recipient cell. After the uptake, they can release the cargo in the receiving cell. So in their physiological role, they are involved in cell-to-cell communication, but we can exploit this feature to set up a very convenient drug delivery system because they can bring their cargo directly in the cell. We have different kind of EVs. For example, we have exosomes that are smaller and are produced, from the inside of the cell by the multivesicular body, or we have microvesicles that are larger and are produced by the membrane, the plasma membrane. Regarding the size, we are in the range of nanometers with the EVs, while with cell we are in the range of microns. Among EVs, red blood cells derived extracellular vesicle are one of the most promising tools for several reasons. First of all, they are safer than other EVs released from other cells because RBCs lack DNA. They are fully non-immunogenic because RBCs lack the major histocompatibility complex. In addition, they are very easy to obtain on a large scale and at low cost, starting from the patient's or donor's blood. So we can set up both autologous and heterologous administrations. And finally, they are highly versatile because they can be used fresh or stored and can be obtained in several ways. Regarding the way of production, we developed and patented a new method and is based on 3 steps. We had the idea to combine our expertise in drug loading. So the first step is the cell preloading according to the hypotonic dialysis, as I mentioned before. The second step is the new physical vesiculation method, the so-called soft extrusion. And why inspired by nature? Because it resembles the natural squeezing occurring to RBC in the spleen. So we are the production of membrane microvesicles already loaded, as you can see. The third and final step is the deep characterization of our obtained product according the minimal information study needed when working with EVs, because as I told you, we are in a world that is very, very smaller than cells. At the beginning of our story, we set up the procedure at the lab scale using an hand extruder. Very quickly, because of time, we started with a very small amount of blood. We purified RBCs by centrifugation. We completely removed white blood cells, and then we performed the cargo loading on purified RBCs. In this example, we put a microRNA. Then we performed the soft extrusion with the hand extruder, and this way we could process 1 milliliter of sample. And then we have the steps of purification: a first centrifugation to remove intact cell, a second centrifugation to remove cell debris, and finally the ultrapurification step, ultracentrifugation step, sorry. And at the end, we obtain our product. About the characterization, we used nanoparticle tracking analysis to get information about the particle size distribution, about the particle concentration, and about the zeta potential. By transmission electron microscopy, we demonstrated that our particles were rounded EVs and lipid-bound EVs, so with the membrane bilayer, while by flow cytometry, we demonstrated that they maintain the same surface antigens such as mother cells, RBCs, that are glycophorin A antigen and CD47 receptor. And also the phosphatidylserine exposure, they do not expose phosphatidylserine in the outer membrane. All these data have been already published on scientific reports. Sorry. As the first in vitro application, we wanted to produce RBCEVs loaded with a small RNA, namely a microRNA 210, to treat an endothelial dysfunction, for example, in type 2 diabetes patients. First of all, we set up our in vitro cell model. We used human umbilical vein endothelial cells. We assessed the uptake that was maximum after 24 hours. So we efficiently produce RBCEVs loaded with a high amount of microRNA inside. We gave them to cells, and the cargo release was effective. And once in cells, UVEC can exert its biological effect as demonstrated by the target protein downregulation and by the reduction of mitochondrial metabolism. Also, the results can be found in this publication. In the second part of my talk, I want to share with you some unpublished data about the scaling up of our procedure and about the first preclinical application in a GMT deficient model, in a preclinical model of GM deficiencies. Concerning the scaling up of our procedure, we translated the method from the hand extruder to the use of an automated one. Actually, we have a family of extruders that are currently used in clinics to do liposomes. We started with the smallest one, but we can further increase the volume. Once again, we started from a little bit higher amount of blood. We purified RBCs and we performed the drug loading, as I mentioned before. In this case, we put inside a long RNA molecule, a messenger RNA, and we performed the vesiculation with the automated extruder. In this way, we could process 10 milliliter of sample instead of 1 milliliter. And in addition, we could further control the production conditions such as temperature, pressure, and so on. And the purification steps are the same. Once again, we had the final product in our hand and we wanted to characterize it. Regarding the physical characterization, by dynamic light scattering and once again by nanoparticle tracking analysis, we demonstrated that our RBC population was very homogeneous, that the procedure was highly reproducible. Sorry, this is small, this 130 nanometers. I was saying that the procedure is highly reproducible and gave a good yield if considering that we started from about 5 milliliter of blood. And once again, by transmission electron microscopy, we demonstrated the perfect integrity of our membrane bilayer. Regarding the biological characterization, we analyzed our RBCEVs by flow cytometry. First of all, we gated our population, the physical parameters. Then we used the lipophilic cationic dye to select only the particles with the intact membrane. They were higher than the 80%. And on the positive population, we analyzed once again the presence of the RBC antigens such as glycophorin A and CD47. The positivity was even higher than before, the 19%. And the CD47, the presence of the CD47 on the membrane is very important because it can act as don't eat me signal. Thus avoiding the early recognition by the spleen. Also, the phosphatidylserine in the outer membrane layer was lesser than before, less than the 4%, and also this feature can avoid the recognition by the spleen. So, with this information, we move to the first preclinical application. We set up an RNA-based therapy to treat the GAMT deficiency. The strategy is based on the following steps. The first one is the production of RBCEVs, this time loaded with a long RNA, a messenger RNA coding for the GAMT enzyme. We injected these particles intravenously in a murine model of GAMT deficiency that was already available and characterized in our lab. We wanted to target the liver. Why? Because the liver is the most important organ for GAMT, for the GAMT metabolism. And we evaluated the biochemical parameters before and after the treatment. And we are also working on the behavioral features. About the GAMT RBCEV production, we started with the GAMT mRNA synthesis. As you know, RNA therapies have emerged after the pandemic, and we use more or less the same strategies as in vaccine. So we, in this case, we use the coding sequence of the GAMT enzyme. Actually, we use the mutated one I showed you at the very beginning of my talk. And we put all the tricks to increase RNA stability, such as pseudouridine, and also to improve its translation in human and mouse hosts. So we use this long RNA to load our RBCs and to produce our RBCEVs. At the end of the procedure, we evaluated the amount of RNA inside them, and for the first time, we were able to efficiently to load this big molecule with a very great efficiency. But most importantly, it was also retained in our RBCEVs, thus producing particles with up to 50 copies inside. We move to mice. First of all, we wanted to set up some studies about the pharmacokinetics and the biodistribution. So we administered our particles intravenously in our murine model, and the plasma half-life was longer than usual, with an estimated half-life of 15 minutes, while usually is 1 to 2 minutes, and almost the 10% was still available after 2 hours. Were they able to reach the liver? The answer is yes. And you can see here that the liver accumulation was very, very higher than the spleen one, according to the excellent biological feature I showed you before. And the particles were able to be taken up by hepatocytes as demonstrated by immunofluorescence. Okay, but the key question is, are they effective? So we evaluated the biochemical parameters. As anyone of you should know, GAMT deficiency is one of the most devastating among creatine deficiencies, not so much for the lack of creatine, but rather due to the very high level of guanidinoacetate that is produced in the liver. after the first step of creatine synthesis, it can be released in circulation, reaching the brain and causing toxicity. On the right, you can see our preclinical results. So we evaluated the blood GAA levels in wild type and mutated in GAMT mice, and in GAMT mice before and after the treatment. And you can see the blood GAA levels in a wild type mouse that are normal, while here you can see the very, very high blood GAA levels in a mutated mouse, and this is what happened after the treatment. 4 hours, 3 days, and 5 days. So a single administration of our RBCEVs was able to reduce blood GAA levels up to 5 days. So this is proof that our strategy could work. In fact, we are doing some deep behavioral profiling on untreated and treated mice. These are some of the experiments we are performing in collaboration with the University of Rome, such as ultrasound vocalization, homing, open field, and so on. But these results are in progress. These experiments are in progress. So, to conclude, some of the already obtained deliverables are the following. We set up a method to produce RBCEVs that is easily scalable and translatable to clinics. We used this new method to produce RBCEVs loaded with a messenger RNA coding for the GAMT enzyme. And the production was very efficient, and we have the procedure and also the product available in our lab. Then we performed bioavailability studies in a murine model of GAMT deficiencies and demonstrating very excellent features, a good pharmacokinetic and bioavailability profile. They were able to target the liver, and the mRNA cargo was effectively released in hepatocytes. In hepatocytes, the mRNA is translated into the respective protein and enzyme, which can metabolize the guanidinoacetate. And as I showed you, a single administration was already able to reduce the GAA levels up to 5 days. And as I showed in the previous slide, some behavioral studies are ongoing. What's next? So in the next months, we are setting up some dose and time funding studies to find the best effective dose between RNA and RBCEVs and to find the best interval time. We want also to set up, to put on some repeated administration studies to fine-tune the protocol to treat our mice. And then we will use all this information to set up efficacy studies in which we want to treat our mice for a 3-month period, during which we will do our biochemical evaluations, such as guanidinoacetate and creatine. But in parallel, we also want to perform a deeper metabolic profiling and behavioral studies during treatment. Okay, to conclude, these are the findings that made everything possible. And these are the people I want to thank, starting from my collaborators and friends from the University of Urbino, our RBCEV team, like a stream of RBCs, and also our collaborators from the University of Rome. This is a wonderful picture of Urbino, the Urbino skyline, that is a World Heritage Site. And finally, thank you all for your attention.
Thank you so much, Sarah. That was a great talk. It looks like we have questions steadily coming into the Q&A box. So I'll invite our moderators for this session, Katie and Matt. You guys are online. So Yeah, so it'll be great if you can start maybe with a brief introduction, and then we'll do round robin questions. So dividing enough time between all the speakers for the session. Okay, those are my house rules. Over to you.
Okay, I'm Matt Skelton. I'm a researcher at Cincinnati Children's, developed the first mouse model of CTD, and have been working on it ever since.
Okay, and I'm Katie Pulsifer. I'm staff at ACD, but I actually have a scientific background. I have a PhD in chemistry and then worked in biopharma for a few years, most recently at Roche before joining ACD. And my nephew was actually diagnosed with GAMT via newborn screening. So it's really rewarding to kind of be able to combine professional background with also a cause that's very personal to me. I was really looking forward to this session this morning, and I thought the talks were fantastic. Matt, do you want to go ahead with the first question and then we'll trade off?
Sure. Actually, I'm going to pick the third one, and I'm going to answer it myself if that's okay with the panelists, if you guys can see it, because it kind of fits exactly what I do. So the question is, all the in vivo studies are in the same Black 6 background, actually the same shared mouse. There are two actually, but is anyone exploring the 129 background to understand if some observations may be associated with the endogenous background mutations in inbred lines? That's a great question, and it really is very knowledgeable about mouse models. The problem is, the first answer is everything's been done in the Black 6 background. The reason for that is because the back crossing to the 129 is time consuming and labor intensive, and we just don't have the funds and the time to do that. It's a great question. It's a question I've actually written grants on and gotten rejected for, but it's something that really hasn't been able to be done just because we only have so many hands to do the work. So that takes care of that question, I hope. We'll go, the first question is for Alex. Will you perform the transient overexpression in the knockout background?
Yes, that is the plan. I'm not sure if we'll use the wild type cells to test the overexpression. Just keep it narrow and use the knockout cells for that experiment.
So there was a question in here, let's see, that I think was directed at Alex, but I feel like would maybe be applicable to you and Jonathan. It was asking about thinking forward to dosing in humans. Are your prodrug candidates, or I guess pharmacochaperone candidates, stable enough for oral administration, or is this something that would likely need to be an IV infusion?
So I can start. I mean, this is a great question, but this is why we need to do medicinal chemistry and structure activity relationships on both kinds of compounds, right? So doing screening in cell lines and finding active compounds, it can be done for a lot of different targets, right? And so, I think both Alex has promising things in vitro and we have promising things in vitro, but getting across the finish line will require more modifications to the compound, and then taking those modified compounds and looking to see how stable they ultimately are. So there's multiple reasons why you want to optimize a compound. One is to make them super potent and helpful for lots of variants, but just as, if not more important, is to make sure that they live in the body and get into the brain, right? And so that's part of it. Like, there might be compounds that can kind of do that sort of well, and in those cases, a lot of times you need more than just oral dosing. Maybe you get lucky and you find a compound that's active and is bioavailable where you can take it as a pill. But those are questions that come down with and or after a lot of medicinal chemistry.
Yeah, I think John explained that really well. I guess the only thing I would add is that in our work, we're planning to start with IP dosing, and if we can see something from that, we'd move to try oral. But I think we would rather switch to an approach like nasal delivery to investigate before we went to IV, because we acknowledge the fact that this would be a drug that would need to be taken regularly by patients, and so an IV infusion is just not feasible. So we'd much rather switch to nasal before going IV. So.
Yeah, I mean, it does make to that point, right? I know that Sangeetha and company have invested in a lot of different delivery technologies over the years too, and that's where the nanoparticles, the red blood cells, other things like that could be all useful tools to make these therapies like, you actually practically useful, right? So yeah, these things can at some point hopefully become synergistic.
Okay, we'll go on to a question for Sarah. What is the widespread feasibility of your technique? Would patients have to travel to specific hospitals for this therapy, or is it something that can be done at most hospitals?
About the feasibility?
Feasibility, yeah, widespread. So is it something that would need maybe an academically focused institution, or is it something that local hospitals could take care of?
As I showed in my talk, the great strength of our strategy is the use of RBCs that is already in clinics. So the first part of the procedure is already used in clinics. So we are a step forward for our strategies. And also the second part of the methodology can be easily translated to the clinics because, as I told you, the automated extruders are currently already used in clinics to do liposomes. So the best wonderfully in a not so far future, we wanted to combine, so to do the loading with the red cell loader, that is the equipment I showed you, and the automated extruder, all done under sterile and a pyrogenic condition, or in cell factory, or I don't know, maybe in a biotech company to produce our RBCEVs and to be used fresh or stored. And maybe the more feasible thing is to use the same blood from patients as we already do in clinics. So in this way, I think the best option is to work in the hospital. So I know I am running a lot with my fantasy, but I am with my finger crossed.
Great. For Jonathan, how does inhibiting or locking the transporter in the inward-facing conformation influence the recycling of the transporter back to the outward-facing position? Once it's transported to the membrane, does that inhibitor go away and then the transporter is active again, or how does that work logistically? Oh, I think you're on mute, by the way.
Thank you. Sorry. So one, there's a few classes of inhibitors that one can make. Where you'd really have to worry about that is if this were something called a covalent inhibitor, or a suicide inhibitor, which basically will stick permanently through a covalent bond to the protein. In that case, it's really locked. But what these things do is they look like creatine. So the important thing is the way that creatine works, right? As what makes it a substrate, which is something that's pumped by the enzyme, rather than an inhibitor, right, is that creatine can bind equally well to the two conformations. So it binds in the out, then it flips to the in, and releases. It binds and releases, right? The inhibitors that prefer one state, right, anything that sticks to one state over the others has a risk of locking up that cycle so the enzyme can't go back and forth. But none of them will permanently, unless they make that covalent linkage, none of them will permanently lock the transporter one direction, right? Because they're constantly, these inhibitors come on and off and on and off and on and off. So what I would think is normally the enzyme would be cycling back and forth. The inhibitor might bias that, so it spends more time like this, but it's not going to really stop it from rocking back and forth. Now, in terms of how that, one question we have is, will these ultimately act as not just inhibitors, but also partial substrates? And that's certainly we can look for with our mass spec assay. And that actually might be great if the creatine transporter pumps our inhibitor into the cell even a little bit, that might help the cells concentrate it and make it a more effective drug. And in fact, the fact that we are seeing more protein expression does suggest that the protein or the inhibitors are getting in the cell, potentially by way of the transporter itself. So this is something we'll be looking more closely at, but the reality is the computational screen gives you things that are active, but it doesn't tell you the nitty-gritty details of what happens in cells. That's something we really have to do the experiments to determine.
Okay, for Alex, and I guess, Jonathan, you can answer this one too. Are there plans to test the prodrugs or the chaperones in patient-derived cell lines? Can other cell types be used to confirm differences? For Alex, can other cell types be used to confirm differences in FAH expression or activity across various tissues?
Yeah, that's a pretty good idea, actually. I think we have at least one more cell line still available in liquid nitrogen to test the knockout cells in their abilities to, or I guess in the prodrug's ability of delivering creatine to those cell lines. Maybe that's something we look at. I think we're currently more interested in just moving into the mouse model. We've spent about a year and a half in cells, and so I think there's a lot to learn from the cells, but we want to know the translatability of our cell assay to the in vivo assay, maybe sooner rather than later. Um, so so it's not a bad idea. It's actually quite a good idea, and maybe it's something we can look into. But I think for now, we plan to just start testing in vivo.
Yeah, I agree. I think it would be nice to have a more relevant cell line. I mean, Alex and I have used different cell lines, not for any like, I mean, for us, we use hex cells because they don't have a lot of residual expression of creatine transporter and they're just amenable. Like they won't, they're pretty robust, right? Having something more relevant would be helpful, but that's not something that we obviously have access to. If there were like an immortalized, patient-derived cell line, we would certainly use it. So that might be something we can talk to within the network. It's certainly a good idea. It's just something that we don't have the means to do right now.
Got another question for Sarah. You showed data demonstrating high liver uptake of the red blood cell vesicles. What are your thoughts for being able to use this to treat the neurological symptoms of GAMT? How easily does that pass the blood-brain barrier?
We hope that after repeated administrations, the blood GA levels will be reduced and stabilized, and so also the brain GA levels will be reduced, and the toxicity should be reduced. This is what we hope. But as I showed you, we need, this is the reason why we need to perform a more study, because these were very preliminary results. So we performed only one administration, and this is not a gene therapy. So we need to perform repeated administration, how much RBCEVs, how much RNA, how often. So we need to increase our knowledge about the strategy.
Yeah, that totally makes sense. There's another question here that kind of follows along with that. How long are your particles stable? And I'm sure maybe you don't have a good idea of how long they're stable in vivo yet, but like in a test tube, how long are they stable for?
Sorry, for me?
Yeah.
Oh, okay, sorry. As I told, usually we use them fresh. So we produce RBCVs and we use them. This is the best way. However, they can be stored, and we have very small data about the long-term storage, but we saw that they can maintain at 4 degrees for up to 2 months, more or less. We still don't have data, sorry, about the long-term storage for example, at lower temperature, as for the vaccine, as I told you before. We have to perform this analysis. I don't know.
Okay, so
if I
I'll go back
answered to the question. Yes, sorry. Please
Got it.
go
Okay,
on.
sorry. For Alex, how are you planning to confirm that your prodrugs pass the blood-brain barrier? And I think that this could again could go to Jonathan as well, since both of you are working on strategies to address that.
Yeah, so for our in vivo work, when we do our PK study, we plan to collect brain and muscle and blood, and then run mass spec to see where the deuterated creatine levels are and in what concentration. So when we measure the deuterated levels of creatine in the brain, we'll get a sense of how easily these compounds, or how little, we'll find out, I guess, with time, how these compounds bypass the blood-brain barrier. And I think maybe it's important to mention these creatine amides. We have some foresight that there's going to be some challenges with their blood-brain barrier permeability. I think just from a chemist standpoint, which I'm not one, but maybe referencing Al here, that having a free carboxyl group in your prodrug design is not great for membrane permeability. And so we want to learn whether that or maybe its macrocycle comparator is more effective. And if, for example, these compounds get into muscle at a high rate, then maybe that could be used as a proxy to use those compounds as a nasal formulation, thinking that you can get similar just tissue access from the nasal administration, bypassing the blood-brain barrier that way, and using muscle as a proxy of just the ability to get into certain organs and not bypass certain barriers. But it's definitely the million-dollar question going into our in vivo study to see, is it brain penetrant? And if it's not, what are the workarounds?
Oh, yeah. So I would say, again, I think that this is part of the hard part of the med chem. I will say when we do our screening, like, as I mentioned, we start from this pool of 95 million molecules, and then we sort of put a bunch of filters in it. We filter for things that we think will, again, have the right stuff inside the molecule. Also things that follow certain empirical rules. I sort of alluded to Lipinski's rule of 5. Basically, those are sort of rules in terms of not looking at molecules that are too big to cross membranes or not too polar, things like that. So we don't know until with things like pharmacokinetics, right? And metabolomic stuff. But again, like you can't, we have limited our search. We've been intentionally excluding things that we know can't be drugs. Whether the things that we do end up having the right properties or not will, again, is to be seen. But it's certainly an essential question that must be looked at with anything that you would even remotely take seriously moving forward.
All right, for Sarah, there's a few questions here kind of along similar lines, asking like how often a patient might need to be treated.
To answer this question, we need to perform the repeated administration studies, as I told you before.
Yeah.
So is in line what we were talking about a few minutes ago.
How long were the mice treated repeatedly? Was it just the 5 days, or do you have any like longer time course
The
experiments?
results I showed now were a single administration, and we measured the levels of the GAA levels different times over time. Sorry, so at day 3, at day 5, and so on. For example, in the repeated administration study, we want to try every 3 days, every 5 days, every 7 days. That can be more or less a good range, but we have to try.
Okay.
We have to perform
And
experiments.
one more clarification question. After the 5 days, did you measure again after that point and see like an increase in the GAA then, or was it just 5 days
7
and then
days.
that was it? Okay.
Sorry. Yes, I got the question. We measured till the 7th day after the treatment, and the number of mice weren't so much. So the situation was different in the different animals. So maybe there is some variability to take into account. So in this case, the levels, if we made them in, they were a little bit higher. Not so much. They didn't reach the starting levels, okay? But it was a little bit confusing. So I didn't put the data in the graph. At the moment, we reached the 7 days after the treatment. However, honestly, I don't think that 7 days, that we can do a treatment every week. Maybe we need a shorter time, a shorter interval between 2 different administrations.
Okay.
But we can be in the range of 5 days, as I showed you.
And that would extrapolate to people as well? You anticipate like less than once a week needing dosing?
Are you meaning if we can translate what we saw in mice to humans, more or less?
Yeah.
very, very difficult to answer this question, but I can say yes, but maybe someone in the audience is more expert than me to answer this question. I really don't know. Maybe we can do the parallelism, but I don't know if it can be the same.
Yeah, maybe I can chime in here. Dose finding studies are typically done in stages. You'll first do some in mice, but then you will eventually do them in monkey or other kinds of models where you will titrate back from a maximal dose to understand what is the safe starting dose in humans. Those typically come in the later stages of drug development, more closer to when you're developing your IND package. That's kind of when you will ramp up and do those types of studies.
Yes, of course, step by step. At the moment, we are not allowed to do experiment on monkey, sorry, but in the future, we don't know. Why not?
Right.
Okay. Just a couple more for Jonathan, real quick, it looks like. Did the new paper with the Creatine Transporter cryo-EM structure update your modeling, change anything, or affirm what you knew?
I think it's more of the latter. So we looked at that structure. The resolution isn't awesome to the point where, like it's not a crystal structure. But to be honest with you, my fears about this, we've gotten this a lot when we try to get federal funding to beef up this project. Everybody says, oh, you need a structure, you need a crystal structure, whatever. But, the advances in modeling, there have been a lot of studies. First of all, like I said, for serotonin and dopamine, people have been using models going back 20 years. The second thing I would say is, again, like empirically, we're getting hits that are active. And the third thing I would say is like this is modeling in general with AlphaFold has reached a level where other groups are showing that, like there was a really good study in Cell showing they did a virtual screen using a model and using a crystal structure, and they got different hits, but they got the same proportion of active hits. So like I've always been a believer in the models, mostly because there's like 30 some odd structures of very similar proteins. And sometimes like, crystal structures and cryostructures aren't always the active conformations you want to target. That particular structure more just affirmed that like we looked at them, they look very similar. So I think our modeling is pretty good enough for this. So yeah.
Yeah, those grant reviewers love low-hanging fruit, don't they?
They do.
So last question that I see on here is, will all the experimental, for assessing different variants, will all that be in experimental work, or will you model the different variants?
Sorry, sorry, say that one more time.
So for assessing different variants, will that all be in experimental work, or will you use modeling for the different
Yeah, so I think
variants?
experiments it has to be, because honestly, like on our work and other stuff, you can make models of mutants all the time. They don't actually tell you what the mutants necessarily do, because mutants don't just manifest in the structure of the protein, right? They affect biosynthesis, trafficking, all these other things, right? So this is why like we haven't like really seriously tried to do screening on modeled variants. I'm just not convinced that it's going to tell you the relevant stuff. But yeah, certainly, but the experiments are king. Data are king when it comes to it. So that's where we're going with it.
Gotta have the empirical evidence.
Yeah.
I will introduce our next speaker, Olivier BRAISSANT, as he brings up his slides. Professor BRAISSANT joins us from the University Hospital of Lausanne in Switzerland. His research focuses on a number of inherited metabolic disorders. In particular, he is an expert in the field of creatine metabolism and has published several papers as well as book chapters on them. Today, he will be speaking to us about a Swiss cohort study around diagnostic delay in CCDS. Over to you, Dr. BRAISSANT.
Thank you, Sangeeta, for your kind introduction. Do you hear me?
Yes.
Do you see my screen?
We are seeing your slides in the PowerPoint window. So I think if you click slideshow, hopefully it'll be the right version.
Like this?
Perfect.
Okay. I don't see it on my screen, my slides, but it's okay. I see on the other one. So good morning, everyone, or good evening here in Europa. Dear families, dear colleagues, dear friends, thank you for giving me the opportunity to share with you this study we made in collaboration between the team of Johannes Eberle in the University Children's Hospital of Zurich and myself in Lausanne. Creatine deficiency disorders are caused by mutations in the genes coding for AGAT and GAMT, the two enzymes allowing creatine synthesis, and the creatine transporter, CRT or SLC6A8, allowing our cells to import creatine. In peripheral tissues, creatine synthesis appears dissociated, the main AGAT activity occurring in kidney, the intermediate guanidinoacetate being then transported to the liver where GAMT allows creatine synthesis. The main role of creatine serves as energy storage under the form of phosphocreatine, which then allows the restoration of ATP from ADP through the creatine kinases system. The most severe symptoms of creatine deficiency disorders are due to creatine deficiency in central nervous system, where blood-brain barrier shows a low permeability for creatine coming from periphery, and which, as in periphery, shows a kind of dissociated creatine synthesis pathway between brain cells. The part of energy support through ATP regeneration, creatine appears to play other roles in CNS, being an important osmolyte in the brain, and also playing roles as neuromodulator or even maybe a true neurotransmitter. Creatine deficiency disorders show a complex of variable symptoms such as speech-language disorders, behavioral abnormalities, seizures and epilepsies, developmental delays, movement disorders, and cognitive dysfunction. The most common of the three diseases being speech-language disorders. To help diagnose creatine deficiency disorders, simple tests in blood and urine are performed. The measure of creatine, guanidinoacetate, and creatinine in urine being the better. For AGAT deficiency, extremely low or to undetectable levels of guanidinoacetate are characteristic. For GAMT deficiency, it is the contrary, with elevated levels of guanidinoacetate. And finally, for creatine transporter deficiency, the elevated ratio of creatine over creatinine is characteristic. Among creatine deficiency disorders, creatine transporter defect has no satisfactory treatment so far, but AGAT and GAMT deficiencies can be treated. They are treated by creatine supplementation with, for GAMT deficiency, the addition of ornithine supplementation and possibly arginine or protein-reduced diet to limit the toxic accumulation of guanidinoacetate. As very often for treatable inherited metabolic diseases, the earlier the treatment, the better, which can lead to normal development if started very early, therefore showing the importance of the shortest delay before diagnosis of the disease. Creatine deficiency disorders appear underdiagnosed. Here are two examples of core studies discovering GAMT and creatine transporter deficiencies among children with mild to severe mental retardation or unexplained neurological symptoms. We were therefore interested to know what was going on in Switzerland, and in particular to evaluate the delay between first symptom presentations and diagnosis, and to analyze who was prescribing creatine and guanidinoacetate measures to help for diagnosis of creatine deficiency disorders. I will start here with the example of one of our case reports. A male patient with normal motor development was seen for a regular checkup by his pediatrician at 20 months of age. It was observed during this visit an absence of syllable and word pronunciation, despite normal autoacoustic emissions documented at birth. And the description by the father was that his boy was hearing normally. The boy was referred to the ear, nose, throat specialist of the Kinderspital in Zurich at 22 months of age, who concluded of normal hearing. 1 month later, the pediatrician prescribed the first urine analysis. At the age of 23 months, thus, this first analysis in urine showed a strong increase of the creatine over creatinine ratio, therefore suggesting a creatine transporter defect. Laboratory tests were repeated twice up to 5 months after the first test, confirming the strong increase of creatine over creatinine ratio in urine. Proton MRS in the brain showed the typical strong decrease of creatine peaks. And finally, genetic analysis led to the discovery of a mutation in the SLC6A8 gene, causing a premature stop codon within the creatine transporter coding sequence. All this leading to the confirmation of a creatine transporter defect. Within our quite small cohort, we had patients with creatine transporter defect and GAMT deficiency. Their leading symptoms at first presentation were seizures and epilepsies, global developmental delay, sometimes movement disorders, but in most cases, severe delayed development of expressive language. The delay from first symptoms presentation to diagnosis varied from 3 to 27 months. Within this small cohort of patients seen in Zurich, it was thus observed that depending on the leading symptom, which was age-dependent, they were first referred to different departments like ear, nose, throat specialist, speech therapy, and neurology. From this first referral to the diagnosis of creatine deficiency disorder, a delay from 3 to 27 months was observed with a mean of 11.2 months. In all creatine deficient patients, a severe delayed development of expressive language was observed, which may be the early symptom to recognize to trigger diagnostic testing. We were therefore interested to better appreciate who in Switzerland is asking to test for creatine deficiency disorder. In Switzerland, creatine and guanidinoacetate are measured in the 2 metabolic centers of the Kinderspital in Zurich and the University Hospital of Lausanne. During the last 10 years, we have seen a continuous augmentation of their prescription, urine-only analysis accounting now for about 2/3 of the demands. During these years, the identity of the prescriptors did not evolve much. The vast majority, about 93%, being tertiary care hospital units. Within these tertiary care hospitals, about half of the demands came from the neurology departments, followed by metabolic and developmental units. Only very, very rare demands came from ear, nose, throat, or speech language specialists. And as an example, for the year 2022, from the about 1,400 pediatricians installed in Switzerland in private practice, only 16 did prescribe creatine and guanidinoacetate measures. In summary, creatine deficiency disorders can be simply diagnosed through the measure of creatine and guanidinoacetate in urine. One common symptom for the 3 diseases is the expressive language delay. They appear underdiagnosed, leading to large diagnostic delays with a mean of almost 1 year in our study. However, for the ones that can be treated so far, namely AGAT and GAMT deficiency, the early treatment initiations appears essential for a good outcome. In our study, covering the years 2015 to 2023 in Switzerland, the vast majority of creatine and GIA measure prescription was sent in by tertiary care hospitals, essentially from neurology departments. Very few were prescribed by pediatricians in private practice, ear, nose, throat specialists, or speech therapists. This leads thus to the conclusion that a strong increase of awareness and information on this disease appeared necessary in the first contact groups. This work was performed between the team of Johannes Eberle from Zurich and myself in Lausanne, and it was essentially done by Dr. Christina Kaufmann, a postdoc of Johannes' group. I thank you for your attention.
Thank you so much, Dr. BRAISSANT. I'll invite our last set of speakers for this session. We have two speakers for this session. In fact, our next speakers are unicorns to our community because they will be talking about AGAT deficiency or case studies from an AGAT deficiency paper that they published recently. So it's my pleasure to welcome Dr. Terrance Dolan and Dr. Sophie SCHARNER to our conference here today. Dr. Dolan is a child, adolescent, and adult psychiatrist at McLean Hospital. He also serves as the instructor of psychiatry at the Harvard Medical School. And he's joined in his talk today by Dr. SCHARNER. Dr. SCHARNER is a child psychiatrist in training at Massachusetts General Hospital. And their talk is titled Case Report of Catatonia and Psychosis in a Person with AGAT Deficiency. I see you have your slides up. Could I just request you to swap displays because we're seeing your presenter's view?
Very good. Can you hear? Can you see it just fine now?
Yes, looks great.
Wonderful. Thank you so much for having us today. And Sangeeta, thank you for that spectacular introduction. I think our presentation here actually pairs quite nicely with the one that just preceded it. So again, thank you very much for having us. Dr. SCHARNER and I are not metabolic psychiatrists. This is a case that the two of us encountered in the context of our training at Massachusetts General Hospital. At the time, we encountered this young man in July of 2023 after he was transferred to our hospital in the context of receiving medical care for his presentation. My supervisor, Dr. Hazen, couldn't be here today, and Dr. SCHARNER will, I think, take over in presenting about halfway through the slides. So here we go. We published this article in just past June, and we did encounter a young man with an AGAT deficiency. So as we've talked and as you've seen in this conference already, creatine is essential for brain energy metabolism. The AGAT deficiency is the rarest, and it affects creatine synthesis, which the pathway of which we saw so delightfully in our previous presentation just now. And I think curiously, recent studies have found creatine metabolism possibly being linked to certain psychiatric conditions. So again, as we talked, all brain creatine depletion, can all these deficiencies lead to creatine depletion, which you can see in magnetic resonance spectroscopy, and AGAT, of course, is the rarest. So our patient, again, AGAT itself being a deficiency, it's autosomal recessive, leads to endogenous creatine synthesis. So 50% of creatine can be produced by the body, but the other 50% is acquired in the diet. So again, having an inborn error of production of creatine is challenging. It can lead to specific developmental delays. So our patient was diagnosed with the AGAT deficiency at age 3. About a year had passed, and so he began to develop, I think, very classic symptoms for AGAT deficiency, primarily expressive speech delay. He demonstrated, I think, some symptoms of a lack of social reciprocity, some rigid thinking. But broadly overall, he was caught about a year in and was supplemented with creatine. And despite these delays, was functioning fairly well with support in the community. So he was in his normal state of health until perhaps about a month before his presentation to us. So this would be June 2023 when he initially presented to us. So at this time, for reasons that are not clear to our team, he began to stop taking his creatine supplementation. Whether this is a factor of normal adolescent, I don't want to take this anymore, I'm not interested in having it because my parents want me to do it, or perhaps for an alternative reason, is not clear to us. However, he began to develop psychiatric symptoms, particularly paranoia, disorganized thinking, and then perceptual disturbances, primarily auditory hallucinations. I think these symptoms ultimately resulted particularly in him eloping from the home, which was quite disturbing given that he's never engaged in any kind of behavior like that before. So at which point, I think when he was found and he was brought to, I think, a local emergency department, and he was admitted to an adolescent psychiatric facility for treatment of his psychosis. So backing up here, what is psychosis? So psychosis itself is defined by, I think, two ways of evaluating behavior. The first is the addition of things, such as the addition of delusional thinking and perceptual disturbances in the form of hallucinations. So what are delusions? Delusions themselves are fixed beliefs that are not amenable to change in light of conflicting evidence. Our patient specifically began to develop a paranoid thinking. He began to worry that his uncle was out to get him. He began to grow very concerned that his diet might be poisoned. And no amount of reassurance or comforting was changing his mind on this. Separate from the rigidity of thought that comes with an autistic perspective, there was no amount of evidence that can dissuade him of these thinkings. But other forms of delusions that are common in psychosis are grandiose, referential itself being believing that certain things we see in the world mean specifically for us. So one of the most common is seeing messages in license plates or seeing messages in the news, thinking those messages are just for the patient. Other delusions are somatic, so believing that there's something physically wrong with the body when there's no clear evidence of that happening, and no amount of biologic or medical testing can prove to a person otherwise. Hallucinations are perceptual experiences, so just like hearing things and seeing things are the most common, but they are not limited to visual and auditory. They can be tactile, gustatory as well, though those are much less common. And again, perceptual experiences that occur without any evidence of external stimuli, and they must occur when a person's wide awake. Someone falling asleep or waking up, I think people commonly have odd perceptual experiences during that time, and those wouldn't qualify as hallucinations. The most common in psychosis is auditory, which were the hallucinations that our patient was experiencing. So those are symptoms that emerge. What factors about normal behavior that changes, or rather that's eliminated, is disorganized thinking patterns, which we can infer from disorganized speech and then disorganized behavior. So what that would be, disorganized thinking. So when someone communicates, when we have a conversation with someone who's experiencing psychosis, oftentimes the answers to the questions that you might be asking them in an interview are either obliquely or completely unrelated to whatever the question is. And then in most severe cases, someone's speech is so disorganized, they may no longer have traditional language syntax and context, so the words really can disintegrate into meaninglessness. And we tend to infer that as an ability of understanding how someone is thinking, and oftentimes in those instances of disorganization, people's behavior can be altered. Again, I think a common, misconception about psychosis is that it is only occurs or happens in specific mental illnesses, and they don't. Psychosis can occur across a variety of physical ailments and mental conditions. So I think psychosis can be induced by medications, or it can be induced in the course of like a cancer, or all sorts of different, medical causes can drive this. So I think just the presence of psychosis does not indicate one particular diagnosis. It can cause across many areas, and one of the common things we as psychiatrists do is when we encounter someone with psychosis is rule out what medical conditions could be contributing to it. And of course, psychosis is not limited to a schizophrenic illness spectrum. It can occur commonly within a bipolar spectrum illness or a mood illness as well. So In addition, and what led to the transfer of our patient from the psychiatric facility to us was his development of catatonia. So catatonia, a more, I think, elusive, presentation is, I think primarily results in transfer and not being able to treat it at a psychiatric facility when it begins to threaten to biological health. So our patient began to develop rigidity. He stopped speaking at all, began making, I think, grimacing, okay, and then developed symptoms like apraxia, imitation of behavior. The way we can measure catatonia, because catatonia itself is so challenging to understand and so challenging to diagnose, we use a rating scale. And our particular patient scored about a 23 out of 69. Curiously, when he passed through our emergency department and his cerebral spinal fluid analysis was performed, his creatine was low, not that time. So catatonia itself is a clinical phenomenon. It can also occur in a range of psychiatric and medical conditions. And we truly, as a field, don't understand the pathophysiology of it. It's primarily characterized by abnormal motor behaviors. However, when testing someone's physiological body, there's no clear abnormal measures indicating something's going different with the body. So it's a psychomotor diagnosis here. And again, that can look like grimacing, mirroring, stereotypies, ambitendency. So ambitendency is a perfect example is a person who is looking to step through a doorway, but they step through the doorway, they step backwards, and then they step through the doorway again, and they step backwards. So it's this organized motor symptoms that we see that are, again, challenging. And again, the catatonia can come from autoimmune, it can be medication-induced, folate deficiencies can do this. We see catatonia coming in features of psychotic illnesses with mood illnesses and neurodevelopmental. In order to diagnose a catatonia, a person must have at least 3 of 12 variety of psychomotor symptoms. Some of these symptoms are evidenced by, I think, a freezing withdrawal of normal motor behavior. Other instances, they are seen as an addition, so an agitation or an irritability of some kind. So moving forward, unfortunately, again, this is quite scary for the family, of course, to have their child begin to develop these symptoms. Unfortunately, once recognized, catatonia can be treated through a benzodiazepine. So in this particular patient, his catatonic presentation, he was mute, he was rigid, he was had some senses of automatic obedience. When we evaluated him, he had some mirroring. So when we scratched our heads, he then moved his arm to scratch his head. And unfortunately, after I think several weeks of IV and PO lorazepam treatment, he began to improve. In the time, because he wasn't having free movement just yet, we ended up using a nasogastric tube to supplement and get creatine restarted again. Once the catatonia lysed and he began to have more movement and more social interaction with us, we began to appreciate that the psychosis continued to be present underneath. At this point, we ended up having him transferred back to his psychiatric facility to continue. Overall, after about a month or so of treatment, the catatonia resolved entirely. And then at the 6-month follow-up, he had returned to his baseline and again was continued to be treated overall, to be followed up from a psychiatric perspective. Now I think I'll go ahead and hand off our presentation over to Dr. SCHARNER.
Yeah, thank you very much. Yeah, so this, as my colleague Dr. Dolan just shared, this was a patient who had AGAT deficiency and then developed 2 kind of psychiatric syndromes, one being psychosis and one being this catatonia. And it could obviously be complete coincidence. Kind of sadly, around 1% of the population will develop something like a psychotic syndrome, something like schizophrenia, for example. And people who develop this will typically develop it in their adolescence or young adulthood. So kind of he was an adolescent. But we kind of were curious about it because first, what obviously made us curious was that it kind of correlated time-wise with this patient stopping his creatine supplement. And so that kind of made us think, oh, was he somehow, by some way, a little bit precipitating this by stopping his creatine supplement? And then the other thing that made us curious was that there is now an emerging literature. It's still very much in its early days, but people who research psychosis, and kind of the hallmark disease of psychosis is schizophrenia, they are finding that there are some often kind of subtle creatine abnormalities in some, often like not the whole brain, but like specific parts of the brain. So there is, for example, kind of some studies showing that there are reduced creatine levels in some parts of the brain in some people with schizophrenia, and sometimes even in people with kind of first episode schizophrenia, meaning that it's the very first time that they have any symptoms, and even in some high-risk individuals. And this seems to be kind of interesting because these kind of data is not shown in patients who have other psychiatric illnesses like mania or depression. So it seems to be kind of specific to schizophrenia or psychosis. At the same time, this is, as I said, kind of, it's very kind of emerging. I think this will be like the next 10, 20 years where there will be much more data coming out and much more maybe kind of pathophysiology understood or things like that. Next slide, please, Terrance. Thank you. So kind of, we were kind of, as we all, I think, are aware, the brain is obviously the organ that's kind of the most most sensitive to any creatine disruption, which shows you as well that patients with creatine deficiencies, typically the one organ that really has problems is the brain, kind of in a way, with the developmental delays. And so kind of we know brain needs, just like all cells needs creatine, but it as well needs it specifically for neurotransmission. And I think kind of the proposed mechanism would be that this disruption leads to this energy crisis, and that the neurons are just really much more sensitive to this, and kind of psychiatric symptoms can emerge. There's as well this interesting thing that creatine acts as a GABA co-transmitter, and this might as well explain to some extent this increased risk of epilepsy in some people with creatine deficiency, because GABA is obviously very important when it comes to not having seizures. And this might as well play a role in catatonia. At the same time, as my colleague just said, catatonia as a syndrome is still basically a not really understood syndrome. Like psychiatrists and neurologists, nobody really understands what actually catatonia is in a way. Okay, next slide. So in this patient, he had as well this catatonia, and the people who have severe psychosis sometimes do have catatonia. So it's not something complete, it is something completely separate, and at the same time it happens more often in people with kind of a real severe psychotic break, that they have as well catatonic symptoms. Something that we found interesting is that there is as well as kind of some described in the literature a syndrome that's called periodic catatonia, and it is kind of a syndrome where people have really more catatonia than they have actually psychotic symptoms, and they can really have over a long time, sometimes even over months, catatonic symptoms. We do as well sometimes see catatonia in people with autism spectrum disorders who do not have actually clear psychotic symptoms but still develop catatonic symptoms. So in a way, I think there's going to be a lot to learn over the next years, but there might be some shared vulnerability. I think at this point nobody can really tell, but it was kind of interesting for us. Next slide, please, Terrance. So the limitations of this case, obviously, this is a single case. It cannot prove any causality at all. There's a very high chance that this was just coincidence, that this patient who has both an AGAT deficiency then was so unlucky to as well then as an adolescent develop a psychotic disorder with catatonic symptoms. But there is kind of, as we just talked about, there is some biological plausibility that that he somehow might have been at increased risk for developing these symptoms. Next slide, please. So future directions, we thought that I think kind of people who research psychotic disorders and schizophrenia will continue to research creatine abnormalities. And I'm curious whether at some moment people will start to screen for creatine abnormalities in new onset psychosis. The other thing that obviously is happening, that the few patients that have known AGAT deficiency are prospectively monitored over the years and kind of people look whether they develop any other illnesses, including psychiatric illnesses, obviously. And the third thing would be, but this is obviously very future, that maybe at some moment something related to creatine metabolism might be used actually as a treatment in psychotic disorders or catatonia. In our patient, he did get better once he took again his creatine supplement, but this was not the full solution. So he was not just, all his psychotic symptoms or catatonic symptoms didn't improve with the creatine supplement. He actually did need the full kind of psychiatric treatment that patients with psychotic disorders or catatonia in general get. So it was not just the solution to take again his creatine supplement. Next slide, please. Yeah, so this is already the end of the talk. We want to thank the parents and the child for trusting us with their care and consenting as well to this case report. And we want to thank all the other teams that we collaborated with at our hospital that all took care of this patient so well. Like, it was pediatrics, neurology, and genetics. We three have no financial conflicts of interest, and we thank you all for your attention. And if you have any questions or comments, we would be very interested to hear them. Thank you.
Thank you so much, Dr. SCHARNER and Dr. Dolan. Okay, so we will be moving on to our Q&A session. Our Q&A session is moderated by Dr. Longo and Mikkel Law. So as before, if you could do a brief introduction, Nicola as well as Mikkel, and then take questions round robin for the speakers in the session, it would be great. All right, over to you, Mikkel and Dr. Longo.
Thank you for having me here. So my name is Nicola Longo. I'm a clinical and biochemical geneticist at the University of California, Los Angeles. And I follow many patients with both CCD and with GAMT deficiency.
Thank you, Dr. Longo. My name is Mikkel Law. I'm a volunteer and board member for ACD. I'm also the parent of a GAMT patient. Dr. Longo has also followed my son, and so I'm thankful to be here.
It's nice to have all of you here, and I was impressed by the quality of the talk, and there are several questions. Dr. Curie, which of the measures that you have shown you think might be usable in clinical trials? For example, the touchpad would be accessible to children, but the problem is that some of these measures are relatively novel. Would they be accepted by regulators for application of new drugs?
Thank you for this great question. So basically, I think that some of the outcome measures that we tested, the purpose of the study was to test really a lot of different outcome measures in order to be able to find the best ones for assessing each domain, because we do not know what's going to happen when I don't know when a treatment will be available. For example, Ceres Brain Therapeutics, they are developing and very enthusiastic about their treatment. So what will happen when we start to give creatine to patients with CTD? Because we do not have a specific hypothesis regarding, for example, yes, I'm sure this is going to be an improvement in social response or in language, or we do not actually know. I think it's safe to try to keep at least some assessment on different domains, as it was also recommended by the core setup that ACD designed too, saying that it's important to assess different domains. So I think that our study can help better understand, what type of measures were helpful, what were not. For example, when a test was only performed by, I don't know, let's say 20% of CTD patients, for me, it's not helpful because it's not going to be feasible by a lot of patients. So I think that now we have, we are almost, the study is almost finished, and we are really starting to see clearly what type of outcome measure we can recommend. So I think that we did try during the trial to have French but also international tests in order for us to share also the knowledge that we could learn through this trial. So I think that for each domain, we have clearly now defined that this test is helpful, this one not so much. I mean, like domain by domain, it's kind of getting clear for us now. The other question is very important. Can we or can we not use newly designed measures? It's a very good question. I think that it depends on the contact of each group can have with the regulations. I think that it's impossible to take that as a primary outcome measure, but as a secondary ones, I think it's feasible if we can have, the publication accepted before any discussion with regulation authorities.
Thank you, Ms. Law. Your turn.
I also have a question for you, Aurore. In your observational studies, you mentioned that you were able to avoid MRIs to measure various demonstrated delays. You avoided the MRI typically by observation.
No, there is something wrong in this. I said that maybe I misspoke, and in this case, I'm sorry, I was not clear. It's just that we managed to avoid to have general anesthesia to perform the MRI. So to perform MRI in a patient with CTD, as all patients will agree with me, usually you have to get general anesthesia to do the MRI. But because of this conditioning that we use, the contribution and the collaboration of the family and all this nice, little introduction step by step, it took us some time, but we managed to be able to do MRI without any general anesthesia in 71% of the CTD patient, which is unheard of, I think.
Did you compare the MRI results to what you observed in some of the testing that you did?
Yes, exactly. This is exactly the point. We have all the behavior test result, neuropsychological data, genetic test, everything for each child. And of course, this is how I could show you, for example, it's a very nice correlation between the IQ level and the amount of creatine measured in spectroscopy. We have similar results using, for example, the reasoning test on tablets and the amount of atrophy that we can observe on the brain. So yes, we do. It's a combination of it, which is very interesting. The neuroimaging part in combination with all the different neuropsychological tests that we did.
Thank you.
Olivier, the next question is about the diagnosis in Switzerland. So is urine testing the most prevalent form of testing done? So, and just to give you a perspective, in the United States, now most patients are diagnosed by genome sequencing. How is genome sequencing compared to urinary testing in the Switzerland?
So blood and urine testing are the first row of tests and helps, of course, very rapidly to suggest the diagnosis of a creatine deficiency disorder. But then, of course, genetic is prescribed to certify the diagnosis. But the first row in both our centers in Zurich and in Lausanne is made by simple blood and urine analysis. And indeed, the most useful appear to be the urine one, because with this you can distinguish quite easily between the three disorders.
I totally agree. And, obviously you need that test anyhow, even if you find a genetic variant, because in many cases you cannot classify the genetic variant without additional data. The only thing is that at least in the US, there are now more centers doing genetics, then center doing urinary creatine and creatinine. So that it is the biggest issue, even though the cost of the test is different.
Another question for you, Dr. BRAISSANT. I think you mentioned 188 cases of delayed cognitive or mental retardation. And of those, a subset of them were diagnosed with GAMP deficiency. Are those numbers what you shared, right?
I think you speak of the two publications that I mentioned as suggestive that creatine deficiency disorders are probably underdiagnosed. In our case, in Switzerland, we are a very, very small country, so we don't have a lot of patients. And that explains this very small cohort that I presented. But nevertheless, with the results of the delay of diagnosis that we could observe.
Were there any other rare disorders that demonstrate similarly of those 188 cases that you were able to diagnose, or were you exclusively focused on the GAMP diagnosis?
So for our study, we were only focused on creatine deficiency disorders. But I'm pretty sure, or I'm sure, that this kind of delay in diagnosis concerns, let's say, the vast majority of inherited metabolic disease. Very often the patients follow a very complicated pathway between different specialists before being addressed to, let's say, a metabolic center who will perform one of these very simple tests, like creatine or guanidinoacetate measures, or for other inherited metabolic disorders, let's say organic acids in the urine or amino acids or whatever, acylcarnitines, which can very speed up the diagnosis.
Thank you.
Dr. SCHARNER and Dr. DOLAN. So in the patient with AGAT deficiency, the patient was diagnosed pretty early in life, but the question that people have here, what type of symptom was the patient experiencing before stopping creatine? We know that it was mildly delayed, but can you be more specific on what was the patient experiencing? And also maybe go back to the diagnosis, how was it diagnosed? And my personal question is, was somebody continue to follow this patient in the clinic to measure creatine level in plasma to make sure that the patient was compliant with that? Sorry, with supplement, not with diet.
Yeah, so he was diagnosed at age 3, I think, as a part of a workup to have an understanding of why he was presenting with a mild cognitive delay. And Sophie, fill in if I'm missing something, but my understanding was that he presented with expressive speech delay, with a cognitive delay as well. Beyond that, I think there may have been either a fine motor delay in his presentation as well, but beyond that, no other significant delays to our understanding. Given that they presented, it was found and I think he was probably diagnosed with the delays prior to age 3, and they started a workup, and then at age 3 is when they recognized it as an AGAT deficiency and started oral supplementation right then and there. So I think he had been on oral creatine for most of his life prior to presenting to us. In terms of follow-up, I think us as consultants to the case did not, I think, beyond checking on a chart review, our electronic medical record did a nice job, I think, going on and following with him. He was continued on a clonazepam as an ongoing benzodiazepine treatment and continued on olanzapine moving forward, at least at 6 months in time. It's not clear to me whether that continued moving forward. And to our understanding, at least, creatine supplementation began again. So Sophie, would you have anything to add to that?
Yeah, so I think just kind of the question of whether he did, when after being diagnosed at age 3, then always take his creatine. So he did follow up in kind of the clinic for the years until he had these psychotic symptoms, and did have from time to time like an brain MRS that showed a rather normal, like still low threshold, but kind of rather normal creatine levels. So I think we can kind of assume that for the majority of his life, from age 3 until he had this time in adolescence when he stopped the creatine, that for the majority of the time he took the creatine supplements. I think we can kind of assume that. Yeah.
And part of what makes this case study so interesting is it's quite possible he got paranoid about whatever his food was and stopped taking the creatine. So we can't prove causality that's here.
So many patients with this condition are followed in the metabolic clinic. Was this patient followed in the metabolic clinic?
Yes.
So just became non-compliant. So the spectrum of AGAT deficiency is expanding, and now there are even adults who are described with milder variant who present mostly with muscle symptoms. They also have some cognitive delay, but not terrible, and they seem to be doing relatively well apart the muscle symptom and sometimes acute muscle symptom as well.
I have a question for Aurore. How do you see opportunities for your work to complement the existing core outcome set created with the families of patients and researchers around the world to guide comparisons across treatments?
I'm willing to collaborate with everybody. I mean, it's totally fine. We could discuss things. I think that for us, the main thing is to publish quickly our results, because I think this way it can be accessible to everybody. But of course, I'm very open to any collaboration with ICD or the group of TERRANCE or RIS.
That's great to hear. Thank you. The more collaboration, the better. Thank you.
Dr. SCHARNER and Dr. Dolan, there are other questions for you. And the question is that, the question in the comment that, the one comment is that there was another patient with GAMP deficiency at age 17 diagnosed this year at Stanford who developed psychosis and Capgras syndrome. So obviously Capgras means that it has been seen by a neurologist, because neurologists are the ones who use this type of abbreviation. And a reduplicative paramnesia after a viral illness. And obviously they confirmed the diagnosis, they started creatine and ornithine with marked improvement. Any thoughts about what you have seen in your patient with AGAT and this finding in GAM deficiency?
I mean, no specific thought. We find it very interesting that there is kind of another case of psychosis. And I think it's as well interesting in this case, if I understand it correctly, that this patient was actually not before even known to have GAM kind of deficiency, and was really then only tested. And that's I think very interesting, because I'm obviously wondering whether maybe more patients, for example, that are out there with who are maybe just diagnosed with autism spectrum disorder and mild developmental delay or things like that, would, if they would get like a full genetic workup, would actually have a creatine deficiency, things like that. So and as well, whether maybe at some moments patients who develop psychotic symptoms should be more kind of checked on their creatine levels. So I think it's a very interesting case.
I would certainly agree. I think it does add more of a nidus to kind of measure creatine for our patients who present with the first break psychosis. A Capgras syndrome and a reduplicative paramnesia, delusions. This patient, our patient at least, had the delusion about his uncle. And I don't think it was specifically that his uncle, the person he thought, I don't think it was a Capgras-like delusion. But I do find the similarity of family member and close contact being the delusional content to be interesting. So I think, yeah, I think it adds more fuel for us to be measuring this for patients with first break psychosis.
I have another question for Dr. Curie. How extensive, in other words, how long and how many sessions is the training with the mock scanner needed for patients to successfully complete the MRI? And what aspects are the most helpful to help the patients acclimate to that physical environment?
I think it was really dependent on each patient, because some patients, they came the first time, it was okay. They managed to accept all the different steps I mentioned. They accept to put one of the parents inside. It was okay. Everything was scripted. Like, for example, one of my collaborator, we did put the person inside the scanner. And when the person went outside, it was very important for the person to smile and to look like everything was going well, because I think that sometimes it's much more reassuring to see someone, smiling and feeling at ease. It's more reassuring for the patient than a lot of words. So everything was scripted this way. So for some patients, it was very easy. 20 minutes, 30 minutes was enough. And then we did organize, we find it helpful if they could come back a second time only for the MRI, because otherwise there was a lot of things going on at the same time. So we prefer to make them come a second time, and then they did do a training again in the mock scanner, and then following by the actual MRI. So it was really, if everything was smooth, it could only last half an hour plus 15 or 20 minutes before the real MRI. But for some other children, it was really necessary to go multiple times. And every time they could improve a little bit. Sometimes it took them 3-4 times to be ready and comfortable enough to be able to do it, with the commitment of the family, of course.
Thank you.
Dr. BRAISSANT, one question is, where do ENT fit in the diagnosis of a creatine deficiency syndrome? Obviously, is that a typical thing to happen, or it is not very typical? Then I have a second question, which is more related to Europe in general. Is there any movement to start newborn screening, at least for GAMT deficiency in Europe?
So I will start with the second part of your question. With what I presented, for sure, let's say now for GAMT deficiency, it would be very useful to have it in a newborn screening program. In Switzerland, it's not on the radar so far as I know, but I'm not in the commissions looking at this. I think the Netherlands were programming to introduce it. I don't know where they are now. But for sure, at least for GAMT deficiency, which is much more frequent than AGAT, it would be, and also much more easy to detect through the strong increase of guanidinoacetate. It would be very useful to introduce it in newborn screening program. And sorry, I forgot the first part of your question.
The first part is that you were focusing on the ear, nose, and throat specialist in the diagnosis of creatine deficiency. Is that very common? Is that, or that everybody goes to the ENT doctor in Switzerland?
No, but what was one of the strong conclusions of this work was that the most common symptom in the first presentation of the patients was the very strong delay of expressive language development. Therefore, one should really increase the information and the awareness of these creatine deficiency disorders in the specialties that are dealing with language, with speech, And that is one of the conclusions of this work.
So
And
basically,
that would probably facilitate and accelerate some diagnosis.
for a hearing test, and the hearing test is normal, they need to think to something else. Is that correct?
That was one of the points. The case report I showed, the hearing test was normal, and the patient did not went further into metabolic test or so really the expressive language may be something strong to think of.
Thank you.
Question.
Sanjit, I believe that we start getting over time. I do not know. Are we still on time?
Dr. Longo, no, you're right. But we are over time, but we do have maybe time for like 1 or 2 questions more. Let's do that because I see there's a couple more.
Thank you. Question for Terrance and Sophie. How is the patient doing now? And would a measure of his psychosis symptoms be something to explore for clinical trials in all patients with AGAT?
So, in short, given that we were consultants, we didn't follow longitudinally this patient. So I don't know how he's doing today. We did, I think, peek in the chart again at 6 months and then briefly to 12 months to see he was doing back to his baseline, although continuing on psychiatric medications at that time. So as of a year ago, we can say that he was continued with following with outpatient psychiatry. He was on an antipsychotic and he was on a benzodiazepine at that time with creatine supplementation. Regarding to the second part of the question, I think, it's unclear whether the psychosis was a part of an AGAT deficiency, given how rare it is. This could be something that occurs in 1 in every 100 people who have an AGAT deficiency. It could be something that only occurs after a prolonged period of creatinine not taking it. And again, as mentioned before, this could have been a situation where he's just happened to be a very unlucky person and developed a psychosis anyway, completely independently of his creatine deficiency, became paranoid about his food and just stopped his creatinine. So I think it's certainly worth, I think, knowing that this is something that did happen to someone with an AGAT deficiency, and for us to be just on the lookout, see if it happens again. So Sophie, what would you add to that?
And I think, yeah, so on this question of like a psychosis scale or measuring psychosis, I think if you have a child with AGAT deficiency and they develop psychosis, you will know because they will somehow do something completely different than they did it for the time before. So psychosis, we typically don't have like a measurement scale or something like that, but it's something you will notice that kind of your child will suddenly do something that's completely out of the ordinary and that you can really not follow at all anymore, and it will be completely different from the maybe like developmental delay or autism symptoms that they had for their childhood before. So it will, and typically it will start only in young adulthood. So I think it's something, if it happens, you will know it right away. Like it's not like you will like something will feel totally off about it. That's kind of almost the hallmark of psychosis, is that it's something that nobody else can follow anymore, like a worry or a behavior that's really bizarre, kind of, in a way.
Dr. Curie, a question for Dr. Jack Di Vaglia. Can the test that you have done be done to an online platform rather than a tablet for family who do not have a tablet?
So usually what we do is that we provide a tablet for the family because it's a specific tablet with a specific tests on it. But otherwise, I mean, it's feasible to have access to, I mean, to put the test online. We did that when we tested it a long time ago, but what we like with the tablet is the fact that for the child, it's very implicit, because if he click on the right picture, then the picture turns on itself, which is very rewarding for them. And if it does that online, I mean, except if you have a touch screen at home, it might be a little bit more difficult, but I'm sure that we can manage to find a way.
Thank you.
So I think that brings us maybe to the close of this Q&A. For this next set of sessions, I'd like to welcome my co-chair, Kausalya Murthy, to take over as host of the conference. Kausalya, feel free to introduce yourself as well as our next speaker.
Hi, I'm Kausalya Murthy, an ACD staff member and part of the organizing committee for the conference. I previously trained as a molecular biologist, and I've previously worked in a couple of biotech companies that worked on rare diseases, specifically in drug discovery. Before I came to ACD, I joined ACD a year back, and I've been enjoying my experience working with the different members of this community, and I can say it's a beautiful community. So to begin this session, we will start with the patient spotlight segment, and it is my pleasure to introduce Andrea. She is the mother of Shubin. Did I get it right, or is the name slightly different? Siobhan, sorry. She's a 19-year-old woman diagnosed with CDD, and she feels extremely privileged to share her journey with this condition. She's also a teacher by profession, and she's also a strong advocate for children with rare diseases, and she strongly believes in a great quality of life for them. And we are very excited to learn about her journey with a 19-year-old child, and we would love to hear her experience so that we can learn from it.
Thank you. So, has that worked? Sharing my screen there?
Yes, we can see your screen.
Great, thank you. I'm in Australia, obviously, from my accent, and it's 4 a.m. here. I haven't had a lot of sleep in the last couple of days, so I'm hoping I can be coherent. But I'm really grateful for the privilege of being able to share something of my daughter's life and our family's journey with CTD. So, just introducing Siobhan. Most people know her as Bonnie. We have pretty strong social role valorization approach to life, and she's focused on thinking about herself in a lot of ways and what the roles are that she has in life. And we've listed some of them there. She was hoping to share that with you herself, but she's sound asleep at the moment.
What do I
Just a tiny little, not sure if this is football.
But most people call me Bonnie. I'm 19 years old. I'm a daughter. I'm a sister. I have a loving family, and I love my dog. I love being at home. I love seeing new schools and going to visit new sites and countries. I'm a workaholic and a friend. I'm a tennis player, a volunteer, coach, and a proud athlete leader. I'm kind, understanding, and brave.
And we already been introduced, so, but I'm fortunate to be mum to these 3 girls. The older 2 girls are my stepchildren, and we have a pretty complex household because they all have significant health issues. The oldest, Emily, has agenesis of corpus callosum and other brain malformations, so we just spend a lot of time dealing with seizures. We've dealt with brain surgery, things like that. Her sister Gabby has recently had heart surgery to address SVT, ended up with pulmonary embolism and has POTS. So Bonnie is in a household where there's quite a lot happening. And as a mum in that situation, I'm pretty tired. a quick run through on some of Bonnie's life. So things started out pretty normally, and we soon found ourselves in that space where she was slow to gain weight, milestones weren't being met. She had some breath holding incidents. We were referred to pediatricians. It all got pretty, well, complex in a way, but there was a lot of wait and see kind of stuff happening. We were referred to early intervention programs. We had a great team of people helping us, and lots of therapy that was super helpful. Before we knew it, she was approaching school age, and I made the call to send her to our local mainstream primary school. Doing that meant we had to step into more formal diagnosis stage, which I found really, really difficult, particularly as an educator, knowing that kids with intellectual impairment are the most isolated and vulnerable sector of our community. However, she made a great start to primary school. She was confident, told me she didn't need me, went to drop at the door. And I did a lot of work advocating for her and negotiating for her to be in a rich learning environment with her peers. She fortunately had a really great peer group, and she was safe and happy. It was really hard measuring her growth because of her communication, and we had some ups and downs sort of navigating how we would handle that. Throughout her primary years, intervention continued, changed, looked a bit different, sometimes at school, sometimes out of school, but that was able to happen really consistently, and she had really good general health. She barely missed any school at all. We reached the teenage years. In that time, we were referred to several paediatricians in her very early years, and then once she reached primary school, we were referred to a developmental paediatrician, and we settled with her. She settled with us. It was a great mutual working relationship for the rest of Bonnie's childhood years. However, we were in a wait-and-see kind of space. High school, Bonnie was really happy to attend high school. Again, we went mainstream. She had pretty good support. It was a bit more challenging in terms of peers, but COVID lockdowns happened, and they were probably pretty timely for us. She managed to miss a couple of years of pretty high hormone levels in peers and was stuck at home, forced to do a lot of learning with a teacher mum and a uni student older sister. I realised that primary school hadn't really helped her meet her potential, but I could also understand that it was really difficult for them because of her communication. As we went through, we faced challenges such as the kind of general typical trying to steer a kid with disability into vocational pathways, hospitality, things like that. Bonnie wouldn't have any of that and chose her own subjects and really challenged the education system. We're not used to a kid with an intellectual disability asserting that she wanted to continue studying German, biology, and business management. Intervention continued throughout those years, and we had pretty good control of that funding for her and some really great people. The continence physiotherapy space was really important as we were still dealing with continence issues well into teenage years, and we'd been set on a path really that didn't get to core issues for many years. Strength and conditioning with physiotherapist, podiatrist, elite athlete team was introduced to her life, and that made a huge difference. I think particularly she scores really low on physical strength, balance, coordination, and having that team work so closely with her helped her develop her thinking about how she used her body, so it really helped develop her thinking skills as well as her physical skills. She developed a passion for tennis, and we have channeled much of her therapy through that love. There are heaps of positives about those first 17 years. Bonnie was working really hard. As a family, we were working really hard, and all of the people in her support team were working really hard. But we still had this feeling that we weren't on track with the real issues, and we were still facing that wait and see kind of scenario from the health system. With the end of paediatric space sort of coming up quickly, I got a bit more assertive about that, and our paediatrician helped us with referral to our local children's hospital. We finally connected with them in 2022, and after a few months, managed to see them face to face for speech assessment and a consult with their team. We waited for a speech report, and we were told we'd possibly be able to access genetic testing, but I had to keep asking about that. Eventually, that was made available to us early in 2023, and we spent some months waiting for results. By September 2023, we got some results, sort of. Well, it was great to have results, but it was pretty confusing. Genetic reports, so really used to it. It's pretty big learning curve. We were in the VUS kind of space. We're unclear about pathogenicity, as we hadn't had access testing for her dad. So it was decided to follow up another family member. So her sister Gabby, being paternal half sister, presumably the same X chromosome from her dad as Bonnie, and Gabby was happy to do that. We thought that would take 4 to 6 weeks. It took another 6 months. In the meantime, we were not referred to a metabolic specialist, and the waiting was really stressful. Eventually, we got those results that there was an upgrade on her pathogenicity, and we got a report. Excuse me. I was pretty floored when we did get that report. The primary recommendation was stated that Bonnie was at risk of having a male child with severe intellectual disability. And as a mum who had an incredible kid, who was a incredible daughter who was working really, really hard, despite her moderate intellectual impairment and other challenges that she was facing, it was really confronting to have possible future fictitious male person seem to kind of step in the centre of her report. We were also told that there was some evidence that treatment with supplements might be helpful, but that MRS was not recommended because it might not be definitive. By this stage, he was now 18. Knowledge is power. So I spent time it's a gradual process with kids learning about anything really, but it was really helpful to have information. It really supported her in understanding who she is, and she's quickly got to a stage where she's able to explain to other people that she has a metabolic disorder. She experiences challenges in getting energy to function, that brain and muscle take a lot more energy to work, and that's really why she struggles and experiences disability. And it really changed her perspective about herself to understand that she had a health condition, and that it was just tangible understanding. Life kept moving on, and we had a sense of urgency about actually doing something. We were still waiting then to see a metabolic specialist. She was moved out of the paediatric space in terms of hospitals, and we faced a 2-year waiting list for access to a metabolic specialist, which was really, really difficult. End of school was 6 months away, And I was really concerned because I was very conscious that as a mum, I'd see her in some situations, I'd see her through the eyes of a mum, and we were losing a window of school observation that might help, that might be helpful, that would be helpful. Finally, we hit some progress. We had, it was a bit of a chance thing, but Bonnie's orthodontist was really interested, and she found this case study. I went back to the geneticist with that, and pretty quickly things were escalated for us, and we were able to meet a terrific metabolic specialist here in Melbourne, who's also a paediatrician, who's been happy to continue to see us. Despite that, we quickly set about collecting baseline data. I'd already been thinking about that myself, wanting things to be as structured as possible for Bonnie, so that our way forward was as factual and measured as possible. And being a teacher, I'm also pretty keen on contributing to research space where I can, and our experiences being beneficial to other people if that's possible. We planned for her to start a trial on supplements as soon as the MRI scan had been done. She launched into supplements. Being Bonnie's age, that it was really important to discuss that with her. She had to be on board. It had to be her choice. I knew full well that some patients experienced no change at all, and that other patients, gosh, that went fast. Other patients did, but she's very willingly taking supplements. We're holding hope cautiously. It's early days, but she quickly got feedback about her articulation from other people. Her vocab's developed. And I think one of the really important things for me there is that I feel as though I'm getting to know my child for the first time in her life, because she's able to communicate so much more. We're planning to revisit baseline data at the 2-year mark and hope to have some useful info to share with other people there. She's in post-life stage now. She's super keen to be part of the solution, so she's proactively contributing in any way that she can.
When I was a kid, I loved dressing up as a superhero. Now I'm a creatine champion. I want good treatment for my health. I want other kids to get help and get help faster than I did. I want very good treatment to help us. The best plan is for a cure.
Thank you. We're really grateful for ACD, and the community is super, super grateful for all of you in the expert space, and really appreciate the way that you connect with us and can come to the table to really work towards great treatments and cure for all of our creatine kids. Thank you.
Thank you, Andrea. That was very beautiful, and so nice to meet Bonnie through your recordings. And if you would like to stay in the panel, I'm sure we'll have some questions. But also, considering the time of day for you, please feel free to drop off, and we'll connect anyone that wants to ask more questions about Bonnie as well. Thank you.
Thank you, Heidi. Thank you, Andrea. And we love Bonnie's story. We now have to move on to the next session, which is the poster session. We will kick off the session with our two-time ACD fellow, Alex. He's a grad student in UCLA, and he will speak about his work on the organoids in the context of GAMT and SL6C8 deficiencies. Thank you.
Good afternoon. My name is Alex. I'm a fourth-year PhD candidate at the Lipschitz Lab at UCLA. And what I study is neurodevelopmental alterations from SLC6A8 mutations. Now, I know we've gone through this several times, but the SLC6A8 mutation is an ineffective creatine transporter, which leads to a lack of intracellular creatine, and it also leads to an accumulation of guanidinoacetate as well. Common symptoms include intellectual disabilities, seizures, muscle weakness, behavioral disorders, autism-like behaviors, prolonged movement disorders, and then also a prolonged QT interval with the heart rate, gastrointestinal problems, general constipation, and failure to thrive. As this is an X-linked mutation, it is more prevalent in males, and there are variable symptoms among female carriers. And what we're studying is human cell culture. The reason that we're studying within the human cell culture is that with the SLC6A8 mutation, we found that the mouse models do not fully accurately represent the mutation expression patterns within humans, especially within the brain. And what we did is we analyzed bulk RNA sequencing data from BRAINSeq, and we saw that with neurons specifically, so it's going to be on the right-hand side of this figure, there was around a 13.5x increase in SLC6A8 expression in mouse neurons compared to human neurons. This trend continues throughout many of the neuronal cell types, with between 10 and 40x higher expression within mouse tissue compared to human cells. Additionally, the minimum expression of SLC6A8 for function is currently unknown, and the failure rate for translating drugs from an animal testing to human approved clinical trials remains high, often around 90 to 92%. With my project, I have 3 different objectives, the first of which is to fully characterize the SLC6A8 mutation in human stem cell, neuronal, and organoid model systems. The next is to evaluate the efficacy of current clinical therapeutics, which are creatine, L-ornithine, and sodium benzoate supplementation on both stem cell function and neurodevelopmental as a whole. And then the last objective is to assess the efficacy of AAV gene therapy on humanized cell lines. So moving on to the results section, what we found so far is that with the cerebral organoids that we have generated, there is no significant difference between SLC6A8 patients, shown in here from red to purple, compared to both our isogenic CRISPR-corrected control, shown in gray, and the wild-type control end that we have, shown in black. And on the right-hand side, we have images, 2D images of the organoids. We took 3 and we averaged them to try to get the area of the spheres. Then what we did is at day 50 and day 100, we froze the organoids and we cut them to allow us to measure for different protein level interactions between cell types. What this is on this slide is a day 50 organoid, and on the left-hand side is the patient cell line, and on the right-hand side is the corrected cell line. In green are stem cells, and in red are neuronal progenitor cells. So a little bit more mature than the stem cells, and as they mature, they turn into mature neuronal cells, mature astrocytes, and mature oligodendrocytes. What this is showing here is that with the patient corrected on the right-hand side, there's more potential for this mature cell types at an earlier stage compared to the patient on the left-hand side, which is mostly stem-like cells. Continuing on to day 150, we found that these alterations with neurodevelopment continue even to a very mature stage, with the patient 4 corrected on the upper left-hand side and the patient 4 on the lower right-hand side. What we're measuring here is we're measuring red is astrocytes, green is oligodendrocytes, and yellow is synaptophysin, which are mature neurons. We didn't find much difference with the expression of the synaptophysin with the mature neurons between the cell lines. But what we did find is that there was a significant decrease in the number of high energy oligodendrocytes in the patient line compared to the corrected line. Additionally, the astrocytes are relatively similar between both cell lines. However, this needs to be re-quantified, as when we're measuring organoids, there's an edge effect where the edges are brighter than the inside of the sphere, and we need to better quantify the inside, excluding the edge of the sphere. Next, we ran a protocol for day 21 eye neurons. So we took stem cells and we slowly turned them into neurons. And what we found is that at day 21, the patient neurons, as indicated on the top of this figure here, have a hyperexcitability and a lack of synaptic pruning. And synaptic pruning is where the neurons.
I do believe we have Alex live, so maybe we'll switch to his live presentation.
So we were currently at this slide here, which shows that there is an increased or a decreased amount of synaptic pruning within the patient's cell lines. And this synaptic pruning is a very high energetic process that is impacted in some seizure disorders. And on the bottom hand of the slide are patient 4 corrected neurons, which show very simple bipolar neurons, and they're very similar between. And these are cortical in nature. Moving on to our next aim of characterizing the mutation on stem cell lines, we're using this IP11N iPSC line, which is a completely characterized neurogenin 2 inducible, meaning we could turn it into cortical neurons. And in the absence of doxycycline, it remains in a pluripotent state, so stem cells. So we can study the neurogenesis system in one cell line. Currently, we are in the process of knocking out SLC6A8 and doing a clonal selection. We just completed that last week. And anything that shows is related to imaging. Cells were grown on 96 well plates, automatically imaged every 8 to 10 hours using an automated imaging, automated microscope, and automated plating system to get rid of human error within these systems. So with this, we first started by charting the wild type growth of these cell lines. And as this was our first time doing this, we plated 2,000 cells per well. We imaged 2 days later. So this day 1 is going to be 2 days after plating. And then we imaged over the course of a long weekend to get the growth curve. And what we found is that in the doubling time of this cell line, it's roughly between 17 and 24 hours. Then when we performed a growth curve of a SLC6A8 knockout, so this is a non-clonal selection. So we performed the knockout, it's around 95% efficiency. And on the left-hand side of the screen, we can see the cells plated. Now, this is the same timeline as the previous one. We found that in terms of plating the number of cells to viable cells at day 1 of imaging, there were significantly less. And then looking at this chart on the right-hand side, most of the wells were below our threshold for quantification. So with this previous growth here, we had around 60% of the cells above the threshold. And for this one here, we had around 20% of the wells above the threshold. And then in terms of the growth is relatively the same if they were plated on the well. But again, these are a non-clonal population, so it could be the wild type growing. We're working on generating a clonal population. So in conclusion, we found that there were no growth alterations between the organoid models, indicating similar overall cellular growth. Next, we demonstrated that the SLC6A8 mutations demonstrated developmental delays in mature cell types in the organoid models. Next, we showed that the patient neuronal models show a lack of synaptic pruning with increased neuronal connections between individual neurons, which possibly contributes to the clinical phenotype of seizures. Next, we showed that the SLC6A8 knockout stem cell model shows inconsistent cell growth, with most wells having no or negative growth. However, the knockout line was not a pure clonal population, and we are completing this within the next couple of months. Lastly, the SLC6A8 knockout line displays inconsistent growth with difficulty passaging in culture, and this is just from my experience working with it. In terms of the future directions, for AIM-1, which is fully characterizing the mutation, we're currently generating these clonal lines and validating. Again, we have 400 wells that were completed last week. Once they're grown and once they're sequenced to make sure that they're knockouts, we should have it done by October. Next, we're going to use that stem cell line to run an automated growth and analysis just to be completed one more time to confirm the growth models, which will be completed in October. Next, we will be running neuronal differentiation of these lines and fully characterizing them, which will begin once they're validated, estimated around December, because it takes around a month for them to turn into neurons. In terms of evaluating the effect of current clinical therapies, this will be completed once the clonal cells are produced and fully validated, estimated by January of next year. Supplementation, again, that might take another couple months after that, so estimated around March of next year. In terms of the AAV gene therapy, that again is going to take a little bit after the supplementation, estimating around March or April of next year. And there is one more thing that we're looking at, which when we're doing gene therapy, one of the most important things is determining the minimal correction of efficiency. So we're going to be modulating the level of SLC6A8 between 0 and 100% within these knockout lines to determine what's the baseline level that's necessary for normal neuronal development within these cell lines. I just wanted to give a big shout out to, of course, the Lipschitz lab, Rachel Reyes, who I have the privilege of working with. And then without the molecular shared screening resource, which they have so many very fancy robotic machines, this data would not be possible. And then the stem cell engineering core, because in addition to generating knockouts, we're also generating induced patient lines. And hopefully, with the same parental line, we can generate SLC6A8 mutations and GAMP mutations and potentially AGAT mutations to show relationships between all three of the disorders within one parental cell line. Thank you.
Thank you, Alex. We next have Chinyi Chen. Her work is focused on focused ultrasound-enabled creatine delivery to the brain in CDD. Chinyi, if you're around, can you come on? Yeah. I think we are having issues with loading the slides. Would you be able to load up your slides and speak from them?
I'm going to do it for her.
Okay.
So just give me a minute to bring up her slides. Thank you. All right, if that looks okay, Shinyi, then you can go right ahead.
Shinyi, you were on mute.
Can you hear?
Yes.
Thank you to have me and help me again here. Very surprised day and lesson a lot from last 2 days. So our lab is basically is a translational lab. We try to do some disease directly on basic science, then directly transfer to clinical trial. So when we hear about the ACD disease from Dr. Zesberg, we decide to apply this research progress. So our proposal is to use the focused ultrasound mediate blood-brain barrier to open the brain blood vessel, then can allow the creatine can transmit into the brain system and can be potentially become a treatment for the creatine transporter deficiency. So here we are a multiple expert joint team to help me to finish this project. So we appreciate everyone here. So have a basic introduction here. Yes. How I can, okay, thank you. So how we can apply the application from basic research to clinical is because this the device actually is already a FDA approved device, and such as the ultrasound image is a daily use in the image for medical diagnosis about utero or cancer or your body organ development. And ultrafocusons is a engineered device that allow the ultrasound can become more focused on specific area. So in that way, when you apply different parameter to the brain or tissue, it can become a different therapy. So currently, this FDA-approved treatment already have multiple clinical trial ongoing, including Alzheimer's disease, Parkinson's disease, and such as our lab is doing the brainstem tumor disease. So we have multiple clinical trial before, and GBM, brain tumor, epilepsy, and ALS all have been trying to apply this focused ultrasound media BBB opening to do some drug delivery into the brain. Next one. So how focused ultrasound can make the BBB opening is because when we are trying to scanning the tissue integrity, the contrast agent for the ultrasound actually is a microbubble. When you deliver the sound to the tissue, it will make the bubble have a dynamic size change, and that kind of change can allow your blood-brain barrier potential temporarily open, and then it can open about maximum for 3 days. So in this way, if you have a drug or a chemical that you are interested about want to deliver into brain, or you already know it can be function in other tissue, but somehow it cannot go through the brain, then this is a potential approach for the cure in the future for treatment. So in next one, here is our previous study, because we study on the brainstem tumor. It's also very difficult to treat, and also it's a kind of a rare disease for pediatric brainstem tumor. It's control your heartbeat, your breathing, your everything. Your vital is controlled by here. So when we have the animal model disease, we have the tumor and we treat with the focused ultrasound to deliver the drug into the brainstem. We do see some of the drug already go into the brainstem area, and also the brain tumor size change. And for chemical drug, we try here, the size could be from 600 Dalton to 300 Dalton, including to the a bigger molecular weight of the antibody to 150K Dalton size. So when we consider about the creatine, it's a amino acid only about 130 Dalton size. It could be very high potentially can through our BBB opening to penetrate into the brain. So since we can apply the animal model disease from brainstem tumor and can recruit multiple clinical trial for the brainstem and use the non-MRI guiding the focused ultrasound treatment to treat the patient. You can see the picture here. So that's why we start all of this new project. So in the next one will be how we design our model here. Since the creatine is a daily uptake for the patient, and any kind of the current patient already take medicine, we can just assume that could be our target of the drug in the future. And so our principle here is to know whether our BBB opening can enhance the creatine going to the brain. So we have multiple different plane on the BBB opening in the brain system, including the cortex, hippocampus, or here's some difference, and including in the future might have the whole brain opening in the health. And we will follow with the MRI to confirm the BBB opening and MRIs to make sure the in vivo creatine uptake is increased, and including the LC mass to measure how was the creatine change the whole brain metabolism. So we have the MRI machine, MRS scanner, and CT, so we all can check how animal is healthy or not. So in that, we have just showing our very exciting data in the next slide. We are new lab, so we just have a very young baby show up from the animal. So this animal, we scan the best MRS scanning. So it shows in the MRS in the wild type. We use the left side and right side as a control and also wild type as control. It's similar to the patient's data. The creatine shows normal in a wild type animal, but somehow in a creatine knockout animal, it's all very low metabolism at the creatine peak. So after we know the baseline of this creatine knockout animal, we start our health brain treatment in the next one. So we start to giving the animal with daily water with creatine. So they will take whatever they want in the drinking water with the creatine, including in the wild type animal and the knockout animal. So we observe when we open the health brain at the right side, wild type animal at left side and right side, their peak metabolism profile in the MRI scanning still look very similar. And in the very promising data we have here is the left side, the creatine peak is still very low. But in the, in the creatine knockout left side, it's still very low. But when we open the BBB opening, everything, the creatine peak at the 3 ppm, they have a significant increase. So means that our creatine do increase after we do the focused ultrasound mediated BBB opening. So in the future, we will accumulate more of this to measure all the baseline of the animal with baseline of MRS and with a different repeat of the focused ultrasound and to see how was their MRS scanning will look like. And in the future, we might apply to behavior tests because we already see multiple signs they already present they have a behavior very significant change on the animal. So next one will be, since we know the creatine can go into the brain, but there is other mechanism we are not absolutely know how it could be can be proved. So we need to know whether when the creatine go into the brain, does it really go into the neuron or oligodendrocyte, glia cell, microglia cell, or astrocyte? So here is because of the BBB, because the creatine transporter mutation is a whole body mutation. So all the cell will have the issue on the uptake of creatine. So even though we already opened the BBB, but we don't know how it can transmit into the cell because we are not destroy the cell. We are temporarily open the gap between the cell adhesion. So next step will be we have to know how this can allow creatine go into the cell. So for patient, we use the patients derived the fibroblasts. So we select from Colonial Institute, which also is from the ACD associate organization. So they have multiple ACD creating CTD mutant or GAT mutant fibroblasts. And I try to choose the most early onset cell line. And because they also have a clinical profile and database there. So I go through all the data and choose the most potentially most severe cell line to use it, and including the 2 healthy control. So we will use in vitro focused ultrasound to test this to based on the different mutation from patient. But because our device is originally for in vivo study, not for in vitro, so we spend some time to engineer this whole device into an in vitro setting. So here is how we can the mice area will be replaced as a cell dish, and we need to focus ultrasound induce one by one and work on different fibroblasts. And after that, we will harvest the cell and check how was the dye intact into the cell and how was the creatine in the intracellular level is increased or not, and including in the future, we might use the LC mass to profile how was this different patient have different metabolism change. So here we are still ongoing and troubleshooting some of the technical part. So in the future, hope we can share much more with everyone. So here is our current result and take-home message is we knew our hypothesis that focused ultrasound mediated BBB opening for patient could be potentially enhance the creatine delivery into the brain. But we also still need to know whether we can increase the intracellular uptake in the brain system, in the glia cell or kind of a neuron cell. So we are very excited by the MI study, and we will try to do more in the future to know. And we're already on the way to apply the R21 and also other clinical trial in the future. So keep your eye on with everything and contact Heidi and my PI in the future. Yes. So next one, thanks to everyone. The lab leader is Jan Frey Wu, and he is at the Children's National Hospital with the Salzburger. And we have the two labs doing the same thing. And James helped us to do the BBB opening, and Jackson helped us to engineer the focused ultrasound machine, and we have the MRS expertise. But can I also want to ask Kim later on about her opinion? And Sarah Epstein also helped us a lot on the animal handling. So our lab is supported by the lab startup funding, and the project is also by support by the fellowship award from the ACD Foundation. So we do appreciate you give us a chance to do this very rare disease, and we can contribute our science skill to advance the therapy potential. We also thank all the cooperation people in the Plattsburgh in our core facility, Children's National Hospital, and also Duke, Sarah Young discuss with us, and Purdue over the Jonathan help us to do the LC mass assay. We all appreciate everyone to supporting our research, and thanks everyone.
Thank you, Chunyi. That was a good talk. We will move on to our next speaker, who's Tesla Peretti from Queen's University. She's going to speak about gene replacement therapy in AGAT deficiencies.
Okay, so thank you for the introduction. My name is Tesla, and I'm one of the 2025 ACD fellows. My project is working on a novel gene therapy approach to AGAT deficiency, and I'm really excited to share my progress with you all. I'm going to forego some background about AGAT for this conference, but I will put some background about gene therapy. So let's focus on how it is delivered. I'm using gene replacement therapy, which is great for monogenic recessive disorders like AGAT deficiency. So it works by delivering healthy genes to cells, and then viral vectors are of interest here, just because they have the ability to manipulate the host cell's machinery to produce the desired proteins from the delivered gene. We use adeno-associated vectors, known as AAVs, as they have several positives, such as long-term expression, little cytotoxicity. They can transduce many cell types, and there's different serotypes. So a specific one known as AAV9 has the ability to cross the blood-brain barrier, which is the type that we are using in this study. So over here is just how there are wild-type AAVs, and we replace viral genes such as the replication encapsulation proteins with our promoter and transgene of interest. So these are known as recombinant AAVs, which carry our desired transgene. And then this is just a little bit about how the AAV enters the cell and has the encoding to then produce our desired protein. So for this study, we have AGAT deficient mice, which will be referred to as the affected mice. They enter our study at 5 weeks of age. This is when they have their baseline serum collection and they begin immunosuppression. At 6 weeks of age, they receive their injection. So we have our affected mice that either receive the vector injection or a vehicle, and then we have non-affected mice as our other control. So these mice are receiving an IV intravenous injection in their tail vein. And then throughout their lifespan or the span of the study, we have different collections at different times. So we have serum collections and a muscular strength test at 3 different time points before their endpoint at 13 weeks, where we collect all the gross organs and central nervous system for some biochemical analysis, and some of which I will be sharing with you today. So first, we're looking to see if our treatment can restore AGAT protein production. So we see that our affected mice have no AGAT protein produced in the kidney, whereas our non-affected mice do have that AGAT protein. And then in the liver, we see that our affected and non-affected mice both have the GAMT protein being produced. So that's protein here is not affected. And then we also see that our affected treated mice do have that AGAT protein production. The reason why it's in the liver is because this is a big sink for AAVs. And since this is delivered systemically, we know that our liver is having some of that AAV sink, even though we are hoping that most goes to the brain because of the serotype we're using. So this is, it's also great that it's in the liver because, as you guys know, GAMT is also in the liver. So we are kind of creating this enclosed system for creatine production in the periphery. And then this is a quantification of what we just saw. So again, we see that our affected mice have no AGAT protein signal in the kidney, and the affected mice, the non-affected mice do. And then our affected treated mice have that AGAT protein signal in the liver. And then our GAMT signal is not affected by this increase in AGAT protein. So we know that, So we know that the protein is restored, but let's move on to creatine production. Is that being produced? Short answer is yes, it is. So I have a few different organs here to share with you. So we have the midsection of the brain, caudal section of the brain, the muscle, and the serum. So we can see that our affected mice that were treated with our gene therapy have a significantly elevated levels of creatine in all the affected organs that I'm showing you here compared to the affected non-treated mice. And then we even see restoration to heterozygote levels in some of these organs, such as in the brain and in the muscle, which is amazing news. And then this is just our serum creatine over that study period. So again, 5 weeks is when they entered the study. So this is before any treatment has been administered. We can see in our black here our affected mice that receive our vehicle, and then our purple are our affected treated mice. So we see at 5 weeks, they start with the same amount of creatine in their serum. And then this is 2 weeks post-injection, since their injection is happening at 6 weeks. We can already see that increase in creatine happening, and then it steadily increasing throughout the other 2 time points, which is amazing news. Another important factor to look at when increasing AGAT is the GAA accumulation. So we can see that there's an increase in AGAT protein. We see this creatine being produced, but is there an GAA accumulation in these mice? Short answer, in the brain, there isn't, which is amazing. So in our midsection of the brain, we see our treated mice have lower levels of GAA accumulation as compared to our non-affected mice. So these levels are even lower than what is considered phenotypically normal. And then we look at our muscle, which we do see a little, we see more GAA accumulation here. However, this isn't that much of a concern for us, as we know that GAA is more concerned for neurotoxicity versus in the muscle. There hasn't been any specific studies saying this increase would be neurotoxic, since we're not seeing any toxicity there. Also, this level here is not as high as we've seen in our GAMP deficient mice. So we are not producing GAMP deficiency by increasing our levels of AGAT, which is great news here. And then, so these are just amazing results, and this study will go further with direct CNS route of administration. So we are doing intrathecal injection next. And then I'm working on the dosage study to determine the optimal efficacy of the vector. And then finally, a toxicology study would follow to determine the safety of the proposed human dose. I just want to say a special thank you to the Walia Lab, specifically Dr. Troy Webster and Dr. Robin Binsfield, for all of their support and guidance on this project. And then our collaborators with the Schultz Lab, and specifically Alona Tukova, who does all of our mass spec analysis. And then finally, again, for the ACD organization for the fellowship, and just giving me the opportunity to share my results with you today.
Thank you so much, Tesla. That was a wonderful talk. We will now move on to the question and answer session. I will start with the first question for Alex from Luis. He's asked, in cerebral organoids, depending on the CTD patients, do you look for a deficit of neurogenesis, as was described in his lab?
Right. So we're currently looking at this, and we have immunohistochemistry of looking at neurogenesis, but then we're also we're going to be doing bulk RNA sequencing. And then as soon as we get that back, we can start looking at that. But the organoid models appear to follow the similar trends as the 2D neuronal cultures of having a hyperconnectivity phenotype.
The next question. Yeah, sure.
Sorry, I was going to ask a follow-up question on the observation around hyperexcitability and the synaptic pruning. I was wondering if you could, Alex, extrapolate that to, from a human patient standpoint, like what is the impact of that excitability and pruning in humans? I know it's a phenomenon that is maybe commonly observed in autism spectrum disorders, but it'd be great if you can expand a little bit on the implications of that.
Right. So it's very common in autism spectrum, and it's very common in seizure disorders. So previously, I worked with organoids on autism spectrum disorders and epileptic disorders. And with the lack of synaptic pruning, with epilepsy, there's a huge overlap with seizures within various brain regions following the lack of synaptic pruning. With the autism spectrum disorder, so it's 50-50. Sometimes it does lead to seizure disorders. Other times it leads to either there's enhanced connection between neurons, or there's a lack of connection between neurons far away. So that could lead to regions of the brain that are especially active within certain autistic individuals, and then some regions of the brain are not able to communicate with other regions of the brain. So it's really just depending on patient specific. But what we found is that, well, what we need to do next is we need to plate these, and we need to put two electric, one cathode, one anode, and just testing for the expression of how the action potentials are going within the plate.
There was a question, Alex, for you. You mentioned testing creatine, ornithine, and sodium benzoate. Did you mean glycine instead of sodium benzoate, or was it actually sodium benzoate?
We're still looking at that. It's a little tricky. Some trials say sodium benzoate, some trials say glycine. We're going to be testing them all. Now, we're lucky to be working with a molecular screening core. So hopefully, in addition to that, we're going to be screening thousands of FDA-approved drugs. So it's really just looking at hits. In terms of the clinical trials that I've read, it's mostly been, of course, creatine supplementation. Now, with the sodium benzoate, the efficacy is trying to increase the glycine. So we can do sodium benzoate, we can do glycine supplementation, we can do arginine restriction, we can also do L-ornithine in addition to whatever FDA-approved drugs they have on plates.
Chinny, we had some questions for you, and are you concerned about the BBB opening for a few hours, and do you expect some leakage, and how do you test for it?
Leakage in the brain?
Yeah, with the opening.
So for the BBB opening, usually will cross within 3 days. So the leakage, we will by confirm with the BBB crosser after 3 days or long term, and including the creatine metabolism is quite fast. If the cell did not use the creatine after 2 hours, you have nothing there. So it's probably less potential to be diffused into the other area. So the daily uptaking of the creatine is crucial to the patient, and our BBB opening, how long we can sustain and what kind of parameter we should open is also our, in the future, our need to figure out more to know what will be the best.
Got it. You had a question from Troy. He asked if to permeabilize the CNS cells, do the microbubbles need to be in the CSF?
I don't know how microbubble will diffuse, but it should be including in all the body fluid and in the bloodstream. So because it's a contrast agent, so it will diffuse everywhere, including your organ. It will also diffuse to your organ. So it's a larger bubble, so and will be metabolism by the body by itself very quickly. So it's also some FDA approved daily usage in the medical clinical use every day. So I am not a doctor yet, so I might need to get another medical degree.
The question was, are you able to tell if creatine entered the neurons?
Yeah, that's an interesting part, because in the brain, we did not have too much data. Even they say the brain neuron did not have the creatine transporter, but it's a energy, very important energy product. So if we don't have the creatine, I'm not sure how neuron can develop. Of course, in the neuron and glia development, majorly after the baby birth, the neuron is not have a more proliferation anymore. So it's majorly on the synapse development. So the synapse on the memory formation, speech, and muscle muscular function, it's all after your synapse and the connection need to get more strong. So synapse use a lot of energy and ATP. So I'm not sure when the neuron let go of the creatine, whether it will also create some defect, and including the from earliest introduction, if not have pouring of the dendrite, and also the spine is important for the information communication. This will all be very important to rule out whether only glia need the creatine or the neuron also need the creatine. We don't have the correct answer right now.
Does the FUS affect neurons also along with the blood-brain barrier, or it's just the blood-brain barrier?
I repeat again.
Do you think the FUS will affect neurons, or it's limited only to the blood-brain barrier, as in the opening?
Oh, we do the blood-brain barrier opening, but there is some other people use the focused ultrasound to do the neural modulation to therapy the people with the chronic pain or have other anxiety, sleeping disorder, maybe. So it's already have something they already try to use focused ultrasound, but it's not, it's for the neuromodulation, but not the BBB opening. So their parameter and their device might a little bit different than what we have in the future.
I had a question. How still do the mice have to be? Do they have to be sedated? And in a clinical setting, do you expect it that kids have to be sedated, or it's okay for them to be awake?
For the focused ultrasound, you can
Yeah.
be we will use the awake one version. They have two different versions. One is MRI guided focused ultrasound machine, and in our lab in the future, we will try to use the non-MRI guided machine. So children can watch their video or tablet, whatever they want, and just stay there stably, don't move. Then we can do the treatment. And if we need to treat with the speech area or memory area or emotional control of the limb system, frontal cortex, any place, we can depend on what's their clinical report, how was their brain or behavior change, then have a different approach to the children. It will not be everyone the same.
Okay, thank you,
So
Chin.
maybe we should go to some questions for Tesla as well as Andrea. If Andrea, I see her still on. Andrea, if you're still on, thanks for sharing your story. There's a question for you. How much creatine is your daughter taking now? And what are some of the changes you've seen since you started supplements?
Sure. Hi. She's taking 18 grams of creatine a day, 12 of glycine and 12 of arginine, spread over 4 doses. Changes. She's grown almost an inch. She's put on about 10 kilograms, and it's primarily muscle. Her we redid her speech and language assessment at the 12-month mark recently, and she's shown an increase of 10 points in her expressive language score. Some physical measures with her strength and conditioning team saw increases in strength of around 11% in about 6 months. Apparently, they have said that a person really trying to build muscle strength might work really hard to gain 2 or 3% change. I think the biggest change for me has just been in it's just been in her expressing herself a lot more, and we feel a lot more connected with her at home. And we've had experiences where instead of storming out of the room and slamming the door, she has stayed to argue her point about something, which is like that's a really big change. It's a bit annoying sometimes, but yeah.
Thank you.
We redid MRS at 12 months, but I'm not over that. The specialist is more able to speak about those kinds of things.
Thanks, Andrea. Question for Tesla. How are the AGAT mice doing? I believe they do not reproduce really well. Are they difficult to raise and work with? What's your experience now?
Yeah, so I actually had a different proof of concept study about 2 years ago now, and it took 4 months, I believe, to get 15 knockout mice. So 15 AGAT deficient mice because there were so many issues with breeding. It's because that the knockouts were infertile, they would have small litters and all of these issues with them. They just wouldn't breed. They'd have 1 litter in their lifetime and not breed again. It was actually last year at the ACD conference that Dr. Troy Webster from our lab, he was an ACD fellow last year, and he attended the conference. He got to speak with Matthew Skelton, who developed these mice, and he said that he was working on doing creatine supplementation for them. So I reached out to Dr. Skelton and got in contact about the dosage and stuff like that. And the breeding went so much faster. We filled about 50 knockouts in, I want to say, 3 months. So it was so amazing that way. The knockout males were still infertile. However, the females were able to breed, the knockout females, so we could do our het to knockout breeding and we were getting more litters per lifetime, larger litters. So big shout out to them. They really helped push along my project there.
Thanks, Tesla. I think that's a great tip, particularly for those in the field. I know a lot of these mice are procured from different places. I know that Dr. Skelton has made them available through Jax and a couple other maybe other places that he's sent breeders to. But this type of information does get sometimes maybe lost. So please do reach out to Matthew Skelton if you're planning mouse studies, because he will have a lot of insight into raising these mice. And I do want to call out on that note also both Laura Veroncelli as well as Olivier BRAISSANT, who have also generated animal models in the space of CCDS. Okay, Tesla, one more question on your GAA level across organs. I think you saw more GAA in the muscles, and we know that AGAT primarily functions in the kidney. So what do you make of that finding, the extra GAA in the muscle?
Yeah, so I think a big reason for this is because creatine is primarily in our muscles. We have a lot of the SLC6A8 transporters on our muscles for creatine to enter through. And as we know, GAA also uses this transporter to get into cells. So I think it's just the fact that we have so many transporters there. Our GAA is just finding those transporters and going into the muscle. You're right, AGAT does primarily function in the kidney. We saw that with our non-affected mice. However, when we treat our affected mice, the kidney isn't really a big thing for this viral vector because it's brain-derived. However, the liver is always being affected by AAVs. They suck them up, they love it. So we do get that little enclosed creatine system in our liver that way, since GAMT is primarily functioning in the kidneys, or sorry, in the liver as well.
Great, thank you so much. I think that brings us to time. I do want to thank all of y'all for the session. Apologies for the technical snafu, and thank you for accommodating so quickly to go live instead of recorded. Really appreciate you all being flexible, and this is a great session. Andrea, thank you so much for sticking around and answering questions and sharing about Pony, even though it's so early in the morning now, I guess, in Australia. But thanks for your participation, and shout out to everyone in this panel for a great job, including Kausalya. We'll get started on our next session. So we're getting started with session 8. Our session 8 focuses on CCDS diagnosis, outcomes, gaps, gains, and goals. And the first speaker for this session is Kim Cecil. Some of you may remember that Kim moderated one of our sessions yesterday. Kim joins us from Cincinnati Children's Hospital Medical Center. She's an MR spectroscopist and a professor of radiology, pediatrics, neuroscience, a whole bunch of other things, at Cincinnati Children's College of Medicine. She has noted the absence of creatine in the brain on MRS of a 6-year-old patient. This and subsequent work has been instrumental for the discovery of creatine transporter creatine transporter deficiency. So she's going to talk to us today about brain creatine concentrations measured by proton MRS. Over to you, Kim. I
Hi,
see your
everyone.
slides.
Can you see them?
Yes.
Thank you. Thank you so much for the invitation to talk to you today. So I wanted to talk about some of the gaps in knowledge and the broader questions we have. So recognition of creatine deficiency syndromes. We need to get these diagnosed before the developmental milestones are missed. How much creatine is needed for typical functioning? We have general ideas, but we need to quantitative measurements so we can understand what thresholds need to be met when we go to clinical trials for cognitive, language, and behavioral abilities, seizure activities, especially as we've heard today, some of the older patients with creatine deficiency syndromes, if we can improve their life by minimizing the seizure activity and the gastrointestinal problems, this would be a great benefit. And for clinical trials of these therapeutics, what's the time course for creatine recovery to an appropriate level, and what dose would be needed? So can proton MR spectroscopy be useful is the question I ask. Let me familiarize you a little bit with proton MRS in case you're not. It is an MRI-based method. It is non-invasive, but may require anesthesia. This was a wonderful study by Dr. Curie today saying that 71% were able to complete an MRI without anesthesia, which in my experience, I'm amazed. That is wonderful. And we'll be interested in collaborating with Dr. Curie and some child life specialists to see if we can get some studies performed without anesthesia, which has been a huge barrier. For proton MR spectroscopy, we measure select neurochemicals, and we specifically define brain regions to sample. With MR spectroscopy, can measure N-acetylaspartate, creatine, and phosphocreatine, and cholines. And there's different ways to do MRS, but these three main neurochemicals are observed. We can change our settings to measure glutamate, guanidinoacetate, and myoinositol. And when present, we can measure lactate and many other neurochemicals. MRS is a quantitative method. The concentrations we measure are typically between 1 and 10 millimolar, which means we're not going to measure dopamine or serotonin or other smaller concentration neurotransmitters. The brain regions we sample are typically between 2 and 8 cubic centimeters in volume. So we get a mixture of cell types. So we have a hard time distinguishing is a neurochemical in the neurons, the axons, the glia, other cell types. So that's kind of a limitation. Let me show you an example of single voxel proton MR spectroscopy. So the yellow box is showing the region of interest we're sampling, and we choose this as spectroscopists or the technologist running the scan. We've overlaid the yellow box on some T1 weighted images. So this is a sagittal, this is coronal, this is axial, and this yellow box is an intersection of 3 slices. And when we minimize the water signal in the brain and we choose our regions to stay away from the scalp or somewhere where there's a lot of fat, we can get a spectrum. This is what's called a short echo spectrum. And the output you may have seen on some of the slides, LC model has been the gold standard for quantification. And so this kind of distinctive red fitting, we can see N-acetylaspartate, a chemical in our neurons and axons. As I mentioned, with short echo spectroscopy, we can measure glutamate. We see our creatine and phosphocreatine peak, and it's a combination. At most scanners, we aren't able to distinguish the two until you go to super high field strengths, which are not typically used for clinical studies. We see a mixture of the different cholines, glycerol, phosphocholine, phosphocolines, adjacent here to the creatine, phosphocreatine peak. And again, on short echo, we see myoinositol. And this individual patient spectrum is a mixture of a variety of neurochemicals, which are illustrated in this little diagram. So you put them all together, you can do some linear combinations, and then for quantification, as in LC model linear combinations, we can get information about the concentrations of an individual patient in a specific region. So how is proton MR spectroscopy used clinically? We added on to MRI exams to clarify the brain pathology. So for genetic diseases, inborn errors of metabolisms, we can see dramatic changes in neurochemicals. Oftentimes it's used for cancer to distinguish a cancer from a benign neoplasm. So we're looking at the choline signature. In acquired brain injuries such as trauma, infection, ischemia, stroke, we're looking for elevations of lactate, corresponding to the deprivation of oxygen and blood flow. Sometimes we're looking for changes in N-acetylaspartate. And here's just some examples. In Canavan's disease, we see an elevation of the N-acetylaspartate. And sometimes we get confused on the imaging with other diseases that affect the myelin. So this is an example of Alexander disease, which sometimes we're trying to figure out, is it Alexander's disease or Canavan's disease for a patient? So N-acetylaspartate is lower in this condition. And here's some examples. Again, this is kind of a healthy 5-year-old. And if you just do pattern recognition, which we hope AI will be really helpful for us, you can see these spectra show different things. So qualitatively, we can look and get information. So there's lactic acid. This is an example of a brain tumor. So these patterns are distinct for diseases. So it becomes learning how to recognize them. For evaluating a disease process and treatment response, I wanted to take a peek at the time. Okay, make sure I'm still good. When we have ischemia and mitochondrial diseases, we use MR spectroscopy to look for lactate decreasing. For seizures, when they are being controlled, we see reductions in glutamate. When cancer is being contained, we can see choline declines. And for gene therapy with Canavan's disease, we expect to see a reduction of N-acetylaspartate. When something is added, an exogenous agent, mannitol to control brain pressure following surgery, we can see that, and other chemicals can be measured. So it's easier to see reductions from these atypical signals and monitor progress. So it's going to be really interesting to see increases of creatine. So it's kind of the opposite. So let me just step back a little bit. The first use of spectroscopy in creatine deficiency syndromes was with the diagnosis of GAMT. Dr. Stockler published this in 1994, and they did a nice job of showing differences in gray matter and white matter. And then upon successful treatment, you can see it's very small. The creatine is being restored. At Cincinnati Children's, we made the CT diagnosis after identifying the first patient with the absence of creatine. Everywhere we sampled in the brain had a reduction, almost complete absence of creatine. And this is just a comparison with a healthy age-matched control. And then from our laboratories, our patient had elevated creatine in their serum and urine, but other creatine biomarkers were within the normal ranges we had at the time. Fortunately, Dr. Solomons was able to identify the first variant for the SLC6A8 gene and confirm what was our suspicion at the time from the spectroscopy of the Creatine Transporter Deficiency syndrome. So why isn't MR spectroscopy widely used all the time first thing? Well, we have some challenges. The appearance and quantitation of chemicals in the spectrum are impacted by the different field strengths of the MRI scanner, the different acquisition processes, the software we use to acquire it, the different parameters, short echo, long echo, different brain locations to sample, different age and maturation of the state of the patient. So an infant looks different than a 3-year-old, that looks different than a 5-year-old and a 20-year-old. So we have to always be accounting for how the brain white matter, the myelination state's going on. So there's a lot of different factors, and then how the spectra are analyzed, the software that is used, and then the radiologist and physicist approach. There's a lot of radiologists who only just look at that pattern. They start out and they look, is this a lactate detector? And then do they see the major peaks? There's others we do with the vendors. We'll look at just the ratios of peaks, which can sometimes be misleading. And then ideally, we want to get everyone to using quantification software. And ideally, we want to get everyone doing the same thing. And this has been really hard in my field. Fortunately, there are now some white papers that are showing some standardization. And part of the problem is the different vendors have licenses and patents for different methods. And until we could define and get them all on board, we were all using machines that use different software. But these white papers are showing that the vendors are coming on board to standardize, because we all need this for spectroscopy to really meet its potential. The bad thing is these are mostly done in adults, so we've got to get this into the pediatric field a little more, and there we go. So Dr. Henri and myself and others are working to devise a protocol that could become the standard for all children being scanned for metabolic diseases as well as adults. And what we're proposing is to use a 3 Tesla field strength. There's a sequence called semi-laser, which defines that the voxel, that volume of interest, more precisely than other approaches. We want to use a T1 anatomical sequence, like I showed you, so we can determine how much gray matter, white matter, CSF is in our volume that's sampled, because that mixture has an impact on the concentration. We're defining, and it's going to be really interesting as we think about some of the talks here. We had a priori selected the midline visual cortex because that's a very reproducible region, and especially for adults as well as children. And then also we selected the left hemisphere thalamus because in the evolution and time course of development, that region has kind of stabilized by time of birth, so we won't have some of those developmental changes. However, Dr. Lipschut's report of the frontal lobe being very important may pull us to sample there. And then we need all-in-one packages that can do the processing and get the quantification in a uniform manner. So what's our next step? Our goal is to define and fund a CCDS proton MRS study, evaluate this standard protocol. Can we acquire equivalent data at multiple sites, 2 to 3 sites? We can do tests with phantoms and typical volunteers. And I write here that can we quantify the brain creatine robustly and reliably in a way that's FDA can get approval and that radiologists would adopt. And it would have to be a really easy push button that could be implemented on the scanner itself. So it's just automatically generated. And then we can really tackle what does we can tackle the correlation with the phenotype. So what is the functioning gap we've seen with how much creatine is needed to do what function? In order to see the creatine uptake, can we get the right frequency to see that response, especially in the newly diagnosed patients? And again, because there's no radiation, this is a nice technique to do that. And again, if we can overcome some of the anesthesia challenges, that would be really great. And then as I was just, the evolution I was just mentioning, what brain locations are appropriate and reproducible. So this helps address the diagnosis challenges. Getting everyone to use some standard locations. We can avoid some of the radiologists having trouble interpreting the spectra. If we've defined that standard location, we can publish that, we can show that, and there can eliminate some of the confusion. One of the things with MR spectroscopy, the creatine spectral pattern, as you go sample with long echo times, it drops really rapidly just as a typical function. It's just inherent. And that sometimes confuses radiologists on the interpretation. They may overcall a creatine deficiency. And then when it turns out not to be a creatine deficiency syndrome, they get less likely they don't appreciate the specificity, and they are less likely to use spectroscopy as they go forward. So thank you so much for your attention. Let's keep going. Let's keep making advances, and I'll be happy to answer questions later. So thank you.
Thanks so much, Kim. That was great. We'll move on to our next speakers in the session. We have a duo once again. My pleasure to invite Audrey Thurm and Judy Miller to be our speakers for this next talk. Audrey is a psychologist and the former director of neurodevelopmental and behavioral phenotyping at the Intramural Research Program of NIMH. She has an interest in the study of a wide variety of neurodevelopment disorders and has collaborated extensively with ACD over the past several years. Judy Miller is a psychologist in the Department of Child and Adolescent Psychiatry and Behavioral Sciences at the Center for Autism Research at CHOP. And her areas of expertise include diagnosis and classification in autism spectrum disorders. They've published papers in this area, and I'm really excited to hear them speak today on moving from natural history to clinical trial readiness in the CCDS.
Great. Are these slides looking right?
Yes.
Okay, terrific. All right, what an inspiring couple of days. We're gonna tag team this, but I'm gonna try where I can to sort of integrate what we've learned over the last couple of days, if possible. So we'll see how that goes. But thank you for inviting me today. I'm really excited to be here. Okay, I'm going to start actually by describing a project that I'm not core to, but I'm adjacent to. But Audrey and Heidi in particular have been doing this project, and you've been hearing a little bit about the a lot about the core outcome set, and a little bit of that sprinkled throughout some of the talks. So there's been some great work done by the ACD and colleagues funded by PCORI to find a core outcome set. And as you can see from the figure in the lower right or lower left, they really took a very rigorous approach to this. So they did a lot of review of the current evidence of what could possible measures be in CTD and GAMT in particular. And then they conducted a Delphi survey, which is a very structured and rigorous and evidence-based way to generate consensus about things. And I was happy to be part of that process. And then they had a consensus workshop, which I was also happy to be part of, where they brought people together and just did the final tweaks to what can be a good core outcome set. And the domains they picked are on the right there. There was 8 of them. And you've seen analogous domains. You've heard about some of them in some of the animal work that people have talked about today and yesterday, and the work that Dr. Curie talked about with her trial. And you'll hear about some more when we review Vigilant. Okay. So that was a terrific PCORI project, and it led to part 2, which is how do you now take the core outcome measures that we know are important to families and to researchers and figure out how to operationalize them into actual tools that are going to work. That's a whole different task. And here's a figure kind of describing the process that they're taking with this second PCORI grant, which is really exciting to see. So the goal is to build off the core outcome set. Now that we know what's important, to try to figure out what are meaningful changes that could be recognized in a clinical trial, what are the tools that might be sensitive enough to measure that, and with the overarching goal that we don't want to start a clinical trial that fails for the wrong reason. We don't want to have a trial that fails because the measurement tool was not right. And this has definitely happened to other disease groups, and it's really unfortunate. Sometimes it's also true that the intervention wasn't having an impact, but sometimes the intervention might have been having an impact, but the trial fails because the primary outcome measure doesn't measure that change effectively. Okay, so this is an important process because this is how you create a roadmap for FDA, for the FDA. So you're trying to figure out what is the concept that you're interested in, what's been important. So this is where those 8 core outcome domains come in, and then how do you use it in a clinical trial? All right, so with that in mind, I want to review what we learned from Vigilin, particularly now, if you're new to this conference, then this will be new information. But if you're not new, you've heard this before, but I want to try to help us put it in context with some of the new findings people have described in the last couple of days, or today and yesterday. And then Audrey is going to take us to the next step, which is how do we really think about how to put this into a clinical trial. All right, so to remind people, Vigilin was a natural history study, or observational study. We had 50 males with confirmed CTD, and we touched base with them every 2 months with surveys, and then in person every 6 months, except for the period of COVID where we sometimes didn't see everybody 6 months. And the goal was really to understand the symptoms over time and figure out what measures might be feasible to give. And I think this will be interesting to think about, especially in comparison to what Dr. Curie was describing and what she's been able to find. Okay, so our study participants came for multiple visits, and this kind of tells you the figure on the left shows how many we lost to follow-up, which is not very many. So you can see we have a lot of data on these 50 participants. On the right, you can see how many test points we have and the age at that test point. So what I want you to draw your attention to is that this sample is much younger on average than Dr. Curie's, and that you may see that come up when we talk about the measures. And it'll be exciting to see what of that her battery might be able to go younger into younger kids. She's had some really nice success with a lot of those measures, but her kids are older than ours, so next step might be to try some of those in younger and younger kids. So you'll see most of our kids, if you imagine like the 15-year-old mark, most of our kids are under 15. I think our average age was more like 6 or 7. Okay, and here's a summary of what age our participants were when they hit some of these really important life skills. So everybody starts walking, but they're delayed. So they're walking by about age 4. About 75% begin using single words, and that starts also late. Many have seizures. A few, about a quarter or 30%, develop sentences and can use sentences. So it's not the majority, and you can see that it really happens in later childhood. And then toileting, most people remain dependent on others for some of the toileting. And that includes things like being able to pull up your own pants and wipe yourself. It's the whole thing. So it's not just bladder and bowel control, but it's independent toileting. Okay, so this is a list of the measures that we used. And a side note, this manuscript is done and has been under review for almost, I want to say like 8 months. It's been incredibly slow, and we're hoping to get it a final review, but this is the slowest, longest review I've ever been part of. But these are the measures that we used in this, and we've attached this table into the manuscript as part of our supplement, because we want people who read about the Vigilin study to be able to see where did we have success and also where did we really struggle. So the first group is the performance-based measures. So the Bailey and the EVT and the Mullen are all things that you ask the child to do certain things, and then you see what they can do. The Bailey is like an IQ test for infants. The EVT is a measure that Dr. Curie talked about. She was able to use that in her sample. We were not able to get that on everybody. We started with that late, so we didn't pilot on everybody, but we also were only able to do it on a subset of the people that were in our study, because not everyone had the expressive language ability to do it. And I think she found too, and she can confirm, but I think she found that not everybody was able to do the EVT. The Mullen is like the Bailey. It's an IQ test for infants. The NIH Toolbox was a measure that we used. The PBVT is receptive language measure. Again, like the EVT, we think it shows promise for the future. We didn't start with that. We piloted it toward the end. And so we do think it could be useful. The Stanford-Binet is an IQ test that could be helpful. It starts at age 2. And then the WASI starts at age 6. And you heard from Dr. Curie that it was really not possible to get WASIs on in this population, and that we found that to be true too. So we didn't end up keeping that. We also used parent interviews. So toward the end, we piloted the developmental profile, the 4th edition, and we think that would be helpful. We've used the Vineland-3. That's the measure of adaptive skills, and we really think that is going to be important down the road. And then we also measured the ABC, like Dr. Curie did. We used the ADHD rating scale. We discontinued it. We tried it because it's been recognized in clinical trials, but it just was not relevant. The questions were not relevant enough for this population. They're talking about things like doing your homework independently and cleaning your room independently, and it just didn't seem like it fit our kids. We also used the Parent Stress Index, which we also thought was not really relevant. It's not really designed for things for children who have an intellectual disability, so it's better to use something like a quality of life or caregiver burden measure instead. We looked at autism characteristics also. We used the social communication questionnaire. It's a different measure than what Dr. Curie used, but I think measuring autism is really important in this population. You've heard that come up a couple of times. Not everyone with CTD has autism, but some do. And I think that's really important to know because it may impact other things, like if they're struggling to gain weight, are they also picky eaters because of the autism, or is it solely because of the CTD or the creatine difficulty? If they're having a hard time learning a new task, is that because it's impacted by autism? And then we also had some clinician ratings. So we used the Clinical Global Impression Scale, which is widely used in clinical trials. It's kind of an overall, how does the clinician feel things are going? So they take all the information from what they've heard from the parent, what they've seen from the child, what their overall impression is, how severe is the impairment, and how significant is a potential change from baseline. That's a pretty robust and simple measure for clinical trials. It's a little tricky because you have to just be clear that everybody's calibrating to each other. So we tried to do that in Vigilant, and it's hard to calibrate. You need to calibrate against each other, like the other raters from the other sites. And so there's definitely opportunity there, but you need some time to do that. And then the DDC gas is also another measure that is clinician rated. It gives you an overall sense of the person's level of functioning. For Vigilant, we also tried some wearables. We had some success with an echocardiogram. There's a paper on that. We tried to do seizure monitoring with a wrist wearable, and that our patients did not tolerate that, maybe because they were so young. We also tried cognition, which was like an EEG net on your head that had most EEG helmets, or most EEG nets have a lot of electrodes, and this just had a few, I think 9. So it seemed like it might be easier for our population. It was okay, but in the end, it didn't have the comparison data that we really need in order to make good sense of the results. So I don't think it's a good measure at the moment. Okay, and then just to remind people, if you've seen this or not before, so we gave the Mullen or the WASI, and you heard Dr. Curie talk about the lighter, which is an IQ test, instead of the WASI, and that worked really well for her study. Like her, we found that we can't really rely on the Wechsler test or a WASI. There's a few Wechsler tests, so we can't really rely on Wechslers. But we gave the Mullen. Our kids our average age was younger. We gave the Mullen, but usually the kids were already even too old for the Mullen. The lighter is great and could be really great in a clinical trial, but it starts at around age 3. And a lot of our young kids don't have the developmental level to really participate in that just yet. And so that might be something to think about. How can we get a test that goes even younger than that? The Mullen and the Bailey are for infants, so you can get a little bit younger. And the Bailey could be a good option for that because you can compare your own scores to yourself on the Bailey with the personability score. Okay. Oh, and then in the lower figure, you see the Vineland, the overall score. You can see that in our study, our sample is pretty moderately to severely impaired. Okay, this slide just shows you how many of our kids needed to use out-of-range cognitive testing. So most of our kids needed to use out-of-range. So even though our kids were young, our average age was younger than Dr. Curie's, they were still too chronologically old to really participate in a test that was designed for their age. So just to keep that in mind. Okay, and then just one more reminder of why this matters. When you use a standardized test, you can see over here on the left that see how all these scores are at the bottom? We call that the floor of the test. And what this figure shows you is that in CTD, we have so many kids or patients who are scoring at the floor that it's really hard to interpret those results. We don't know how low those scores would really go. And so we don't know if you make improvement, but you still stay on the floor, we're not going to be able to see that improvement. And that's going to make it impossible to report that as a positive outcome in a clinical trial, because it'll look like there was no change when maybe there was. So on the right, what we want to advocate for more is a growth scale value or a personability score. Many tests, but not all, but many tests have the ability to let you measure yourself now against your former self. If the test didn't do that from the outset, there's no way to transform an existing test into this type of growth scale value friendly test. So it has to have been done from the design, when the test was designed. But when you have it, you can then get a lot more information about how someone changes over time. So for example, you can see these red dotted lines. That is the expected sort of change over time in the general population. And you can see, first of all, that in a lot of these subdomains, our patients with CTD are not on the floor anymore. You can detect their change and you can measure it. There's a few domains where they are, right? Domestic, community, those domains are still too low. This is all Vineland domains. So, but this gives us a window into, we could find some domains on the Vineland that our patients can, we can apply to our patients and we can detect change over time. Okay, and so here is the list of the core outcome set that has been identified for CTD and GAMT by the ACD project. And Audrey's going to take over in just a minute. But I want you to think about many of these things. We know that they're important from the core outcome set. And what was interesting about the last couple of days is how many studies touched on some aspect, right? So the animal work in some of the mice studies talked about grip strength in the mice, or weight gain. Weight gain is not on here, but weight gain could be one of the first signs that something is having an impact. And so we're going to have to, as we move forward, we're going to have to sort of balance what is in the core outcome set that is there because we know it's important. What are the first signs that something in the core outcome set is being impacted? And are there signs that might not be obviously connected to one of these domains, but might also be one of the first signs that we see changing in the model? And then the other thing to consider is sometimes some therapies might impact weight or grip strength, but not cognition. And maybe other therapies look like they're impacting cognition. Things might work differently in different animal models and in different subsets of participants, depending on age, depending on the mutation. So it's been really inspiring to see all the things that are happening, and hopefully they're going to all come back together again as we move forward. Okay, Audrey.
Thanks so much. Can you hear me? Actually, will you just keep it on that side for one more second?
Yeah.
Yeah. So I just want to remind everybody a little bit more about the current Parents to Excite project that we're doing, which is to take these core outcomes, which are what the core outcome means is just that it's a suggestion based on consensus from the community that these areas be measured in any trial or any study really, but not necessarily. We know that more things like weight gain also need to be studied. It's just suggestions for that these are areas that they're agreed upon to be important, not the only ones, and not necessarily even the primary outcome, because that all depends on what the treatment is. And what we think is going to change in the time of the treatment, and who the population is, because not every child is showing all these impairments also to the same degree. And we really have to think about the heterogeneity. So we want to take this grouping and really now make sure that we have agreement on metrics, and that parent feasibility and acceptance of measures that we use are okay, and just have at least suggestions for the types of measures that may be useful. So you just heard Kim talk about the MRS and the issues we have, and I'm going to talk now about the other the neurodevelopmental ones, which are fine motor, expressive communication, adaptive functioning, cognitive functioning, and emotion regulations, which not surprisingly were all things we measured in Vigilant that you just heard about. You heard about some of the findings. And now we're going to look at, now we're going to say, okay, is this just CTD? Because we have no treatment trials to be looking at how these measures worked. So we decided to do is take the rest of the literature on similar conditions that we happen to know from patient-focused drug development meetings and qualitative work with other groups, that actually these are very similar and looked at for treatment studies that have been going on. So we now did a review of that to try to get at what measures are being used and what's been acceptable in those. So next. So that's what we did, is have a scoping review of what's been used in previous trials. And we really want to know how they're used, how effective they are. And we know that the populations are not the same, but we can get some ideas of what's been used. I'm going to go really quickly because we only have a few minutes, but just we wanted to show you and have these slides showing the process that we went through. So next. So we did, we looked with a librarian, we published the outline, you can get it there, which is published in Open Science, of how we did the review. So next, we were able to use peer-reviewed treatment studies in registered trials from the last 5 years since 2019, including children that had at least one of these 5 outcomes included in them as an outcome measure for conditions that involved intellectual disability as well. Next. And so this is all just to show you, one of the things we came up with is that the way people define their measures is not necessarily all the same. So expressive communication, there are many, many words you can use for that. There are things related to it, like apraxia or motor movements of the mouth that are not necessarily what we want. So it's just an issue in looking at literature, especially for emotion dysregulation, which can be many, many, many different things, how you would decide on the terminology for that. So I'm happy to discuss that with anyone later. But those are the terms we use. So next. And then we had eligible studies, and then we tried, these are the important areas that we want to get at, because we want to see how, again, to know if it can work in a study, you need to think about all these different things. What are the phases? What kind of trials? Some are better for randomized trials. Some we know we're going to have pilot studies that are just small, and we need to know that we're going to be able to show change over time with an individual with the measure. So next. All right, so then when we did this, these were the measures that we found that were used in any treatment study of intellectual disability. Again, there were none of CTD, GAMT, or AGAT published, so it's all other conditions, which I'm happy to share what those are later. But you can see these are the top measures that were used. They, they did overlap a lot with what was used in Vigilant, and so that's good. And by the way, these were all over the world, and actually most were not in the US in the end. So next, I'm just going to go through and give you a few examples. So this is for adaptive functioning. So in Vigilant, and again in the French study, we use the Vineland Adaptive Behavior Scale, but we also know that there are other measures of adaptive functioning. So we want to make sure that we look at those. So another measure is called the ABAS. It's different in that it is a questionnaire versus an interview, and there's pros and cons of all that. And you can see just, examples of what some of the questions are, just so that people get familiar with how these questions are asked. Next.
Audrey, you can go a little slower. It's okay. We'll give you a
Okay.
little more time. I understand there's a lot of content. Totally
Okay.
fine.
Okay. Well, I don't. Okay. So then for cognitive functioning, these are the different measures that were used. Judy talked about how we use the Mullen and then the Bailey, which are measures that only go up to age 3 or 5. But some studies of different conditions, use some other measures that are much for older children, and we're able to use the Wechsler scale. So this helps us know that. And the brief is a parent questionnaire that can get at some aspects of cognition, including executive functioning. Next. And then again, just so that we continue to familiarize ourselves with it. It says no for CTD, but we just started to use it in Vigilant. So these are just examples of measures, and it's just important to know that different IQ tests and different measures of nonverbal and verbal cognition actually get at different subdomains. So you have to be thinking about what subdomains within them you might want to capture, and what might be the most changeable. Next. Then for expressive communication, you're seeing some of these measures come up again in different areas, like the Vineland and the Bailey, because they are broad measures that cover many different areas. So the Vineland is a measure of adaptive functioning, the way the title says, but it has subdomains in it that are specifically focused on expressive communication. So it can be an overall adaptive functioning measure with a composite score, but can also be a measure of expressive communication. And same with the Bailey and the Mullen, which is nice actually, because it's one measure that then can capture multiple areas within the core outcome set. Next. So again, the Mullen, which we've mentioned here, when it's used for expressive language, it has, about 30 or so items that range from the very earliest that you would see in an infant of cooing or chuckling or laughing, all the way up to, using 3 or 4 word sentences. And so that's what we have to think about is there enough granularity in those items of that measure or any measure that we're going to see change. And I'll talk about it in the end, change especially that parents find to be clinically meaningful. That's the piece that we're really concerned about in this phase of the Parents Excite project. Next. So again, the Vineland with just different examples. So it does, like the other one, go very maybe too quickly from early items of crying using sounds and then single words, all the way up to very complex, being able to give someone directions. Next. And then fine motor, same thing, where some of the measures are the same. The Bailey and the Vineland have those areas. And again, and you can see here, so the Bailey fine motor was actually used as a primary outcome in some trials. So this is really helpful information. And then we need to look at the timing and how it was done. And I mean, whether it was successful or not, I think, is one question, because we can't, necessarily make our decisions based on that, but we can see the acceptability, the feasibility, and how it's being used. Next. Oh, no, yeah, thanks. Yeah, so the Bailey, again, for fine motor, you can see here what the items actually look like. So those of you who've done it, there's a pegboard, there's puzzles, there's, using spoons and different things, where you can, all the way up to scribbling and how you hold a pencil are the examples. Next. And then emotion dysregulation, which we know is a very important area, and it actually has been used as a primary outcome more than I expected with the aberrant behavior checklist, which has been used extensively in autism. But as we said, we're really not sure about whether there's enough of a signal here to be using it for CTD. So we probably need a little bit more data, but it looked like maybe some subdomains, but again, not everybody has these issues, so we really need to think about that. Other measures like the child behavior checklist and some of the autism measures are not validated at all in intellectual disability, even though they're being used. And so we need to really worry about that if we're going to go forward with those. Next. And so these are just, again, some of the examples of the measures, again, that we have tried in the vigilance study, and like I said, that are used maybe with concern about their validity and intellectual disability. Next, I think, yep, we're there. So now just the next steps is that we just want to keep on introducing you to these measures and really starting to understand how they're used, why they're used, and get at, like I said, looking now at details of these studies and how they worked in those studies. And we're going to publish this whole project so that we can have that available for this community and compare it then to what we found with our natural history study data that we've collected so far in CTD. And then the next step for our big project is to put out a community-wide survey for caregivers in the broader community. All of you here, look out to really gauge preferences and feasibility and really start to think about what it would mean to have clinically meaningful change, especially maybe given, being within undergoing a treatment trial for these areas. And then to figure out what would be the next very detailed qualitative research we would do to try to really get at quantification of clinical meaningfulness within these areas. I think that's it.
Thank you, Audrey. Thank you, Judy. It was a bit of a sped up version, I think, than we originally intended, but do want to thank you for all the conceptual sort of explanations and how these could be used in different scenarios. We'll come back to questions later. I'm going to introduce our next set of speakers as a trio. So this next set of questions or speakers really talk about work on utilization of different measures in clinical trials, specifically plasma GA. So our next set of speakers are Karine, Tony, and Marzia. They are participating in our Parents 2.0 project, and they'll be speaking together on different aspects of plasma GAA as one of our core outcomes and how it could be utilized in clinical trials, even how it may be best be measured in the clinic in the care of CAMT patients. So while I give Karine's introduction, maybe Melinda, you can pull up her video. So Karine is a mother of 4-year-old Mias, who was diagnosed with CAMT deficiency when he was about 1 year and 9 months. She has a background, she has a scientific background, has a PhD in wine sciences and a lot of training in techniques such as gas chromatography. And so she utilized or leveraged this technical training to support Mias's health and development using her analytical skills to manage his condition. So we'll start with Karine's talk, and then I'll introduce Tony and Marzia.
All right, Sangeeta, are you seeing the slide? Just let me know. Okay.
Yes, I am. You can unmute and play them. That'd be great.
Hi, everyone, and thank you so much for the opportunity to share our story at the symposium. So we have a 4-year-old little boy. His name is Mias. He was diagnosed at 19 months old. And in South Africa, it was a real challenge to find a laboratory that was willing and capable to analyze guanidinoacetic acid in the plasma. So we had to take it upon ourselves. Luckily, I come from a scientific background, having worked in many labs, and I was able to connect with old colleagues of mine from the university who allowed me to come into the lab and actually analyze Mias's plasma myself. So just to give you an idea of where we started, initially we had Mias on a protein-restricted diet, and he was obviously getting creatine, ornithine, and sodium benzoate. We tried to spread this out throughout the day with 5 doses, with the 3 main doses shown in yellow, and then which has all 3 of the supplements, and then the 2 blue doses I kind of see as top-up doses with just ornithine and sodium benzoate. So when I collect a plasma sample for analysis, I try to always be consistent with the time of day that I do it, so that I can always compare apples with apples. And so we've chosen 12 p.m. for practical reasons to collect the sample at 12 p.m. right before his first, or rather his second, big dose of creatine, ornithine, and sodium benzoate of the day. So here you can see some of the results. We started around 5th of September, and you can see we're pretty happy with Mia's results. It's all below 5, 4.4, 4.7, 3.7, 4.3, and 3.6. So, super happy in general with where we are at. And then the creatine, it started slightly lower. Whether it's too low, I'm not sure, but we did end up increasing the creatine dose, and we could see that in the bloods done afterwards. Another thing was we lifted the protein restriction from when we started, and definitely we did not see any change in the guanidinoacetic acid levels, at least that time of day, even when we lifted and increased the protein intake. So at this point, Mias has no restriction on protein intake. So earlier this year, he had a lymph infection. It was quite severe. We had to have blood tests taken initially to figure out what was going on, and I asked the nurse to please take an additional sample while we have to stick a needle in his arm, get another sample so we can analyze it in the lab for the guanidinoacetic acid. So this allowed me to kind of get a little bit more of a scope of where we are. And the initial sample was at 4 p.m. right before the top-up dose, and we were really happy with the results with the guanidinoacetic acid at 2.8. So about 10 days later, he had to go under anesthesia so that they could remove the lymph, and he had to be fasting. So we had to skip this dose completely, and at around 3 p.m. he was under anesthesia, and again I asked him to take a sample while he was in theater, and this sample's guanidinoacetic acid was 6 micromoles per liter. So you can see when you miss this dose, it does tend to creep. And then a month later, I did my normal checkup analysis at the normal time, and just to make sure, and I saw that it was 3.6, and super happy with that result. So this just shows you how the guanidinoacetic acid can increase if your doses aren't spread ideally, or if you were to miss a dose that you usually have. And I think this is kind of a field that we're not quite sure yet, what the implications are, what time of the day should the doses be, could it be even better than what we are seeing now, things like that. So yes, we have a lot of questions, but obviously sample handling, like I said now, the sample timing, and also the frequency, how often are we supposed to measure the guanidinoacetic acid? And then just practical questions like how long after taking the supplements did sufficient absorption take place? I'm thinking now of when Mias has a stomach bug and he vomits after the supplements. I tend to be quite anxious whether it is being absorbed or not, and if it was enough time. And then factors affecting absorption, are there certain foods that can affect absorption? Sleeping late or staying up late, should I wake him up at 6 so that he can take his dose, or can he sleep a bit late? Does it have an effect? Larger doses with a lower frequency throughout the day versus smaller doses and a higher frequency. I think everybody's got that question. And then increases in guanidinoacetic acid, if it does increase, how long does it take to lower it back down again? So for us to be able to address these questions, first of all, it would be ideal to have an easier sampling method, especially with the little ones, and then also an accurate and precise measurement with low cost and simple method. So that would be the ideal. Thank you.
That was Karin's talk. I will keep us going and introduce Tony Tedesco. Tony is a medical doctor working in general practice in Australia. He has a 12-year-old daughter diagnosed with GAMT, and he also played a significant role in establishing the ACD Australia chapter in 2023. Tony will be also talking on the same topic of around the variation of plasma levels of GAA during the day. And as we bring up his talk, Tony, feel free to bring up your slides. Shout out to both of these parents who are doing all of these different types of things to monitor their children's conditions better. It really illustrates the idea that when we're confronted with the unknown, parents step up and face those challenges with all the tools in their toolbox. Tony, over to you.
Thanks for having me here today. Thank you everyone for organising this. So, as you said, I work as a GP here in Australia, in Sydney. I have a daughter diagnosed at 5. She's now nearly 12, and interestingly, she has a very mild phenotype. At diagnosis, you could see her readings are classic GAMT, and the MRS showed she had very little creatine in her brain, so that's why she has a mild phenotype, I'm not really sure. So we're talking about monitoring GAA. When is the best time to measure it? We need to know how it fluctuates during the day. Do you measure it just before we give some ornithine, or do we wait for a lower trough level to occur in the ornithine levels? If GAA can't be metabolized, which obviously can't in GAMT, that's the problem, it has to be excreted. So what things affect this? Like urine acidity, does protein or amino acids affect it, fluid? Because they're also important, I think, in not just looking at ornithine. And what is the half-life of GAA? So if we developed a miracle drug that inhibits AGAT, how quickly would you see that effect in a blood test? So what I did, I collated a lot of my daughter's blood test results. Bloods were collected, over the years, and I've collated all those down to get this data. And obviously it's quite limited because she's, it's done over many years, and not controlling for any other factors. It's just done quite randomly. So here from this image here, you can see because we collected blood at various times, the profile of creatine, it matches the normal pharmacokinetics you'd see with a drug. So when you have a dose here at the beginning, you get a steep rise and a gradual decline, and it's way above the normal range. So creatine lasts for a long time, it builds up in the body, and we can safely be above that normal range most of the time. For ornithine, it's quite different. It still follows the same curve, the pharmacokinetic response, but you can see it drops down quite quickly. So after about 3 hours, the level of ornithine in the blood is probably not high enough to really be inhibiting AGAT. So, you can see people who are dosing maybe just 3 times a day, every 8 hours, you've got several hours where you're really not doing anything to inhibit AGAT. I also looked at data at how does the GAA change after the timing of the dose, and obviously soon after the dose it's lower, and as more time goes by the GAA levels rise. I'm wondering if this is dose response for that, whether, you know, GA drops down quickly and then climbs back up. This is just me fitting a curve to it. We obviously need better data than what I've got. I looked at the effects of creatine on GAA as well as ornithine on GAA, and you can see they both, as the dose of, as plasma creatine is high or as ornithine is high, you'll get lower levels of plasma GAA. Now, the problem here is, of course, you give creatine and ornithine at the same time, so they're correlated purely because they're both given at the same time. So if you look at plasma creatine, plasma ornithine, that effect we see with creatine at lowering GAA could purely be coincidence because we're giving it at the same time as the ornithine. We don't know. How does GAA vary during the day? This is just a collection of the data over a day. You'll see you've got some very high levels in the morning. It drops down during the day. If I show you when my daughter has her doses every 4 hours, you see that morning dose at 7 a.m. Still we get that high reading there. Then I realised this data at the top here is from the night before where she had missed her morning dose. If we exclude that, now that looks like more what we'd expect, and maybe the curve of the GAA varies like this during the day. Again, we probably need better data, but it matches with the previous speaker how we can get quite big fluctuations especially when doses are skipped. I also looked at the effect of amino acids on guanidinoacetate, and the top two graphs you can see, one looking at GA versus arginine, the second is GA versus glycine, and you can see that even over a very narrow range of arginine, we've got a huge change in plasma GAA, same with glycine. But interestingly, some of the amino acids, especially I found the essential amino acids, there was a fairly strong correlation at how the higher you have these other amino acids, the lower you get the plasma GAA. So thank you very much for listening to my talk.
Thank you so much, Tony. And please stay on for the Q&A. I know you're joining from Australia, right? Early in the morning there. Okay, hopefully you can stay on a little longer for the Q&A. Okay, so the third speaker in this mini session is Dr. Pasquale. Dr. Pasquale is a professor of pathology at the University of Utah School of Medicine and medical director of biochemical genetics and newborn screening at ARUP Laboratories. She's board certified in clinical biochemical genetics and has an interest in newborn screening, particularly in the disorders of carnitine and creatine metabolism, as well as lysosomal storage disorders. So Dr. Pasquale, it's my pleasure to welcome you back in this session. Please bring up your slides when you're ready, and if you could turn on your camera, we are ready for your talk.
Okay, thank you. I am trying to share my screen, and I ran into some privacy issue.
I can drive your slides if you would like.
Do you have my slides?
I do, yes.
Yeah, then why don't you do that?
Pull those up right now.
Thank you. Thank you very much. And I'm just following up on the previous two presentations regarding the measurement of guanidinoacetate in plasma, especially when we're thinking about clinical trials, but also for clinical care. Next slide. I want to go back to the core outcome set, both for CTD and GAMT. And GAMT deficiency. I am going to specifically talk about the study that we are going to start, and this is only for guanidinoacetate, and so this is going to be specifically for patients with GAMT deficiency. But I think that the concept of identifying the best practice, if you want, to collect the data. And we have seen it even in the presentation when about MRS, having a consistency and knowing among different laboratory, different sites is very important. So next slide. So guanidinoacetate has been identified as a core outcome for guanidinoacetate methyltransferase deficiency. High level of GAA have significant neurotoxic effect on the brain. Treatment is based on the GAA blood level, but do we know how GAA levels vary throughout the day? I think we have seen now we've seen evidences that there is a variation. There can be a variability in the measurement in the level of GAA throughout the day, and also are the levels of GAA affected by the time of meals and or the time of the medication and supplement. So based on this, we want to start this circadian study, this project, and the principal investigator of this project is Dr. Schulze. I'm talking here on his behalf. He couldn't be here with us today. So the objectives are to evaluate of this study are to evaluate GAA levels throughout the day, before and after taking medication and supplement, and before and after meals. Next slide. We want to enroll 10 patients with GAM deficiency receiving creatine and ornithine supplement. Because we want to do several collections during the day to make things hopefully easier for the family, we thought to use a blood spot to measure guanidinoacetate in blood spot. So the blood will be collected at home by a finger prick and spotted on filter paper. The paper is going to be dried before it is sent to Dr. Schulze's laboratory that is going to do the measurement. Next. The duration of the study is 2 weeks. Frequency of collection, we are asking to collect 2 weekdays and 2 weekend days over 2 weeks. Family can choose the day, the weekday or the weekend day that they prefer. There are going to be 6 collections per day. The first collection is going to be in the morning. Can you see? Yeah, okay, thank you. It's going to be in the morning before breakfast and before any medication or supplement are given. Then patients are eating breakfast, taking their supplement, and then about 30 to 60 minutes after breakfast and medication, this is the time of the second collection. Third collection will be before lunch. Fourth collection will be 3 to 4 hours after lunch. Then the fifth collection is before dinner. And then the sixth collection that we are asking to do, but it might be optional. It could be optional depending. It will be late at night before bedtime. So this way we can evaluate if we have all this measurement for 2 weekdays and 2 weekend days with 10 patients, I think that we can have a pretty good idea and we can be reasonably confident that we are going to be able to determine whether there is a variability and whether the medication, the supplements, and the meals are affecting the level of GAA. The laboratory test that will be performed on the dry blood spot will be measurement of guanidinoacetate and creatine and measurement of arginine and ornithine. And the families will be asked to sign a consent form and then a document on a specific form that will be given to the family, the date and time of the blood collection, the date, time, and dose of the medication supplement, the date and time and approximate serving size and composition of meals and or formula. So this is the study that we are planning to start. The internal review board of the institution where Dr. Schultz practices is evaluating the protocol, and as soon as we have their approval, we're going to start enrollment. And there will be an announcement through the ACD about the enrollment. That's it. Any question? I'll be happy to try to answer them.
Thank you so much, Marzia. So I'll invite Alex to moderate this session with me. So I do see there's a couple questions in the Q&A box. I think in the interest of time, we might do a couple questions, per person, one or two questions, and then leave you guys on here to answer them in writing. Okay, Alex, would you like to take the first one?
Yes. So I combined a few of the questions into one question. So starting off with Kim, let's see, lots of the questions were relating to the safety and then relating to different experimenters in different locations and how you're aiming to standardize that.
Yes. So most people practicing MR spectroscopy have what the MRI vendors provide them as FDA approved sequences and processing. And so everyone will have the same tools. It's just choosing. So it's like having software. Do you use Word or Excel, let's see, Excel, numbers, different software to do the same thing. So it's just trying to herd all the cats to do the same thing. So having FDA clearance on this semi-laser, we are achieving consensus that way. The biggest variability, I think, is getting a uniform quantification. That was probably the biggest hurdle we have left. And then just asking everyone to use the same echo time. So short echo time gives you a lot of peaks. Long echo time is simpler, but creatine naturally declines. So sometimes that gets confusing. And for radiologists managing hundreds of diseases, we have some special things for creatine. So we've got to try to blend, help them have a benefit from doing something in a standardized way that would benefit all their diseases and conditions. So I hope I addressed that. So
right, yes. MRS is a very powerful technique, and I know it's one of the gold standards that we try to use for looking at creatine levels. Another question that I had is, if an AAV-mediated gene therapy for SLC6A8 deficiency, starting there, restores some transporter expression, what specific MRS biomarkers, other than the creatine peak itself, would you monitor to evaluate downstream metabolic and functional recovery?
Well, the challenge right now has been creatine is our standard peak that we base a lot of semi-quantitative, the N-acetyldecreatine, choline decreatine. So we think NAA is okay within kind of normal limits. We think all the other metabolites are fairly close. Again, if a child's having seizures, the glutamate's going to be disrupted. So certain things, but the main marker we want to look at is the creatine. I think the other metabolites, we would anticipate they would really be off in their concentrations, but from what we can tell so far, monitoring creatine is the main one. Creatine and phosphocreatine.
Right. Yeah, thank you so much. I really look forward to reading some of those results. For Judy and Audrey, I combined a couple of the questions together. So in a trial, can different tests be used for kids at different levels? So even if some are outside of the normal age range, for example, and what terms of like documentation of school or doctor test results, how would that help? And then a few other were relating to separating out floor level data. And then how would this progress with the way that the FDA would accept this as a clinical trial?
Do you want to take, do you want me, is it?
You go first.
Okay. All right. So these are great questions. The beauty of it's always about trying to find the best of not perfect options, right? So the beauty of a test that has this growth scale value is that it's one test, but you are compared against yourself instead of compared against the population. So it gives you the flexibility to say like, well, I started here and I made this much progress, and somebody else on the same test can start somewhere else and they made this much progress. So there's some beauty in that because you've got one thing. What we do and that would cover this idea of if, binning all the data out the floor would capture improvements. When you've got the growth scale value, you're not stuck at the floor anymore. So you can see some movement. Without that, you are left with something like getting a raw score. Like here's the 10 items, like Audrey was putting up a lot of the items from the test. You're stuck with, answering those individual items, but that's not very rigorous. They're not equally, there's not an equal interval between them. It's just really not very rigorous. And the FDA probably would not be super excited about that. So the best is we come up with either one measure that can fit everybody using a growth scale value, or a measure that is so simple and specific that everybody can use it and everybody can demonstrate something, like maybe a pegboard or a grip strength or something like that. And then the question about should you save your tests? You should save your tests because you might need them later for eligibility and other stuff. They might be interesting in a long-term study, but they would be really interesting, particularly if your child is showing a lot of variability. But generally, save them for your kid. Maybe they'd be useful in a study, but probably not.
Yes, thank you.
What do you want to add, Audrey?
Well, just that actually, the regulators are incredibly specific about what they want. They actually have not really accepted these growth scale values yet. They actually have accepted raw scores, even though they shouldn't. They don't know many times. Yeah. So they don't actually understand the psychometrics necessarily of these measures. And a lot of the measures, this is a newer phenomenon to use growth scale values. Mm-hmm. What they do is take into account item difficulty, right? Is that we all know that not all items, the difficulty is the same as kids are getting older. So we have a lot of work to do to show the regulators that these measures work in these populations and that they show change over time, which is again why the natural history study was so incredibly useful, and we probably need more data. And then also, so we do have a problem of heterogeneity, a major problem, even within this population. So it is probably true that we're going to need multiple tests. And if we do that, we just need to pre-specify that we're going to have subgroups that we're going to be looking at based on which test we're going to use. We can't do it willy-nilly. We have to know this all beforehand. But they require enormous amounts of data about the measure in the population you're using it over time in order to accept it, certainly as a primary outcome. But we would all want that even as a secondary outcome, is really establishing that these measures show the change, and we can actually show change. And then we can also show that change is clinically meaningful, just as important.
Right. Thank you. Thank you so much. The FDA is very strict about these guidelines. Before I move on to the final question, just one suggestion came up for Tony. In terms of looking at just specifically ornithine, creatine can also inhibit AGAT activity. And then something else that I do just want to note is that GABA with other guanidino compounds can also be used as a substrate for AGAT, as it is a promiscuous enzyme. So the last question is going to be related to Marizia, which is, when will the study take place and when will you publish the findings? And is this related to females or males or both?
Okay, so the study will start as soon as we have approval from the IRB of the Dr. Schulze's Institute. So hopefully in the next few weeks. That's when the enrollment is going to start. Regarding, it's open to male and females and with gamma deficiency. And definitely this is, I would say that we consider this an exploratory study right now that might might lead to a more formal and a bigger study. And definitely, depending on the data and what we need to do next, depending on the results, but we will definitely plan on publishing the results of the final study.
Thank you so much, Marzia. Thank you, Alex. I think we'll bring this Q&A session to a close. Before I conclude, I do want to maybe give a quick shout out to the fact that many of the speakers in this session are actually part of our Parents 2.0 project, and there are many ways for community members to be involved. I just want to mention, particularly with Kim's work, thank you for being a part of our Parents Project. And I believe we have our expert panel meeting coming up specifically on MRS on October 10th. And if there's members of the audience who'd like to attend and participate, please do email us so we can include you. Also specific to the talk that Marzia, Tony, and Karine mentioned, we have a poll coming up that we would really like to get input on. So with that, I will conclude this session. And while I make introductions to our next set of speakers, please do take the poll and let us know your thoughts. Thank you very much to session 7 speakers, and we're ready for session 8, I believe.
And regarding the poll, we do invite everyone to participate. So you'll notice that the first question says caregivers, but we did provide a not applicable answer so that all of the researchers and clinicians can jump in here and answer that second question. We'd love to get your thoughts.
Thanks, Heidi. All right, it's my pleasure, meanwhile, to do introductions for our next panel. So this next panel is meant to be interactive, titled Road to Rare, Innovating and Delivering Therapies panel discussion. And we have panelists for this session. And I'll just introduce them. We have Andrew Steinsapper. He joins us from Deerfield Management, as well as Apertura Gene Therapy. Andrew is the Acting Chief Technology Officer of Apertura Gene Therapy, and the Director of Gene Therapy Program at Deerfield's Management. Next, we have Clayton Beard. Clayton is the Chief Scientific Officer and Site Head of BridgeBio Gene Therapy. Clayton has significant experience in the development of RNA and DNA-based viral vectors for vaccines and gene therapy. We're excited to hear from him. And then last but not the least, we have Dave Jacoby. Dave is the Senior Vice President and Head of Global Clinical Development at Ultragenyx Corporation. He's responsible for the clinical advancement of portfolio from research all the way through approval. So with that, maybe a quick note that how many seconds do we have left on the poll, Heidi?
We've just we demoted some of our panelists so they have the opportunity to respond. So maybe we'll give it 30 seconds.
Great. And I want to thank everyone for hanging out with us. I know it's a little later in the day and sitting to a virtual conference that has talk after talk after talk, where you can't really, maybe there's not enough time to stretch legs is probably challenging. But I do want to acknowledge and thank you all for your patience and enthusiasm in hanging out with us so far. So for the purposes of this panel, it'll be Heidi and me moderating, but we do invite people to post questions in the Q&A. Let's get started in about maybe 20 seconds. So everyone take deep breaths. Okay. So I see that the poll has disappeared. So we are now officially in our panel discussion. So thanks very much to our panelists for joining us today. Before we get started, maybe I'll invite you all to say maybe a word or two about your experience thus far, through this conference, as well as, general commentary on rare diseases before we get started. How does that sound? Great. So maybe we can start with Andrew.
Sure. Thank you so much for the invitation. It's been great dialing in and listening. I have to applaud just overall running a virtual conference is really hard, but I appreciate that this is much more accessible for folks, especially for caregivers and whatnot, to be able to be here and be present. The thing I come away with, and this is true, I think, every single time I go to a conference like this, we need to make sure that we stay centered on the patients and on the caregivers and understand where the needs are. Because at the end of the day, the drugs we're developing are for those folks. It's not necessarily for the doctors. It's not necessarily for the payers or the folks at the companies. It's actually for the folks that are going to be using them. So hearing and centering those stories before we walk through science every single time, it's great. So really appreciate that and enjoying and seeing all of the research and development just across all the modalities here. I'll hand it over to my other panelists.
Thanks, Andrew. Clayton?
Great. Well, thank you. I'm thrilled to be here as well. I think these meetings are amazing. Over the years, my early part of my career, I made vaccines where I knew who the patients were, who the people we were serving were. But over the past decade, and really over the past few years at BridgeBio, getting to know the people we're actually trying to make a therapy for, and understanding what their experience is, and understanding, not from a scientific point, what needs to be corrected or helped or changed, but hearing from the people themselves. It's really changed the way I think about science. It's changed the way I develop drugs, and it's made it a much more rewarding experience. So I'm thrilled to be getting to know this community. We've just started to get to know you all over the past few months, and it's been a wonderful journey so far. So thank you to ACD and to everybody who was sharing their lives yesterday and today. It's wonderful.
Thanks, Clayton. Dave?
Yeah, no, I couldn't agree more with my co-panelists. It's, for me, listening in for the last couple of days, the density of really important information in terms of things that we can use to think about how to develop medicines in this field was just, I think, really outstanding. I've learned more about really salient information for drug development in the last day and a half than I have in months and years of sort of being peripheral. So the meeting's really outstanding. I think the other point that I'd like to make is that the degree of sophistication that this group is using to sort of deal with the challenges of development in rare diseases is really laudable. Phenotypic heterogeneity, endpoint development, the use of biomarkers in clinical development, these are all really critical issues, and they're all difficult, right? And they require a degree of really strong scientific input in order for it to have the validity to actually work as a drug development tool. And the sort of, as I said, the sophistication on show in the last couple of days really does hearten me that we will have leverage when we have regulatory conversations, both in the US and beyond. So kudos to this group. It's been a great meeting, lots of information for somebody like me.
Thanks, Dave. So I'll pose a question to all of you. I'd like for all of you who have perspectives to just feel free to chime in. I think one of the things that's obviously different about rare diseases is affecting a really small population. And I was wondering if you might be able to comment on the clinical development and regulatory timelines between like small and large molecules for rare diseases. What can we do to accelerate it? I'll give you some backdrop for this question, right? I think we got a lot of questions from our community during COVID that, you know, there were all of these, you know, there's emergency authorization, there's other ways to get this ahead. So why are there not different mechanisms to accelerate therapies for a group that is in need, even if it's small? Can you comment on that?
Who wants to go first? Otherwise, I can kick it off and maybe hand it over to the other two folks. I mean, there are accelerated pathways. When it comes down to it, there are processes and procedures and these different designations that one can get on their therapy that can allow you to market your drug earlier. Under accelerated approval, you still have to do confirmatory studies and trials after the fact, but this is built out so that medication that's showing significant benefit in a population that doesn't have anything else at that given time can have access more quickly. Now, with that, the only way you get to there is having something that is working really well. And I think the only way that we know that something's working really well is to make sure we also understand what it looks like normally. Because in some cases, working really well means slowing decline or halting decline. Sometimes working really well means showing improvements. But it's very hard to do that if you don't have a strong baseline. I know we heard from folks talking about natural history studies, the importance of that, and of registries as well. But maybe Clayton, Dave, so I don't like hog the spotlight here. How do you see that across small molecules, large molecules, cell and gene therapy?
I'll disrupt the order that we'd have previously established. I think there are tangible benefits to working in rare diseases in terms of development timelines. I mean, I think of this as a two-step process rather than a traditional three-step process. And we're really trying to assign dose, and we can do that usually with real detailed kind of mechanistic information about target engagement and biomarker development and, pharmacodynamics and pharmacokinetics, whether it's a small molecule, biologic, or gene therapy. And then, we can trim down that sort of like endpoint development and do a pivotal study in a patient population. Now, there's still issues here, right? Endpoint development is difficult in these diseases. Sometimes the real critical in neurological diseases, things that we want benefit in, take a long time to mature. And so can we take advantage of some of the regulatory tools to get early evidence of efficacy to get approval, and then allow these studies to mature as a function of time? And there are pathways, but they are difficult conversations with regulatory authorities for obvious reasons. They don't want to be letting drugs onto the market that don't have evidence of efficacy. So I think a critical point here also is that there are still certain modalities, gene therapy comes to mind, in which the FDA, as not just the FDA, regulatory authorities in general, make you go fairly slow in first-in-patient studies. And so I can understand a degree of patient community dissatisfaction with the pace of even first-in-patient types of studies, because safety surveillance is a really important thing. And we're usually using relatively high doses to get therapeutic benefits, especially in CNS disorders. So there is still some difficulties in study design parameters in rare diseases that mean that we can't go quite as quickly as we want to. So there are still some impediments in terms of the speed to do development for all of us.
Yep. I fully agree. And I think it is encouraging over the past 6 months or so, we have heard a couple of very public conversations with the FDA where they've been talking about wanting to really accelerate getting therapies for rare diseases moved ahead more quickly. I think we're all cautiously optimistic there. Sometimes there's a little bit of a disconnect between what's said publicly and what we experience down in the trenches interacting with the regulators. But it's always a good thing, I think, that at least they're talking about moving things ahead more quickly. And I think we are also rethinking how we move products into the development stage, into the clinic, maybe with a little less emphasis on getting it commercialized, but maybe doing some things that will allow us to get people dosed and get that therapeutic effect happening while we move a little more slowly towards commercialization. And that's something that's more easily done with really rare conditions, because the lure of the commercial side is not as big as a large number of indication with a large number of people. So
maybe sorry, a quick plug too for the three of us is that, you know, we work in industry, we're considered biased when we go to regulatory agencies. There's a huge amount of leverage ACD has in terms of both the education and validation of both endpoints and biomarkers. So you're our most important ally, most important partner in terms of development, because you have sort of a lot more validity and credibility in these discussions with regulatory agencies than we do by definition. So meetings like this are really not just critical for us, but they're also educational for regulatory authorities as well.
That's excellent feedback. Thank you. I'm going to grab a question out of the Q&A. With the FDA's Rare Pediatric Disease Priority Review Voucher program undergoing the sunsetting schedule, how can we adapt to these changes as rare disease researchers?
Maybe I could touch on this. So I work at an investment firm. We do venture capital efforts. The rare disease, the priority review voucher has been important for some folks, but it is rarely the reason why someone develops a drug to begin with. As it stands right now, the only way to get one is actually have your drug fully approved, which means that even with where it's been sunsetted right now, there's an opportunity, should it be put back into place, where that's something we can go after again. Now, to that end, when thinking about what does that mean as a rare disease researcher, I've always been taught and have been trained to think with the end in mind. And in my mind, that end in mind is not that priority review voucher, which you receive at approval, but rather thinking about the 100th or the 1,000th commercial patient. So a patient that's receiving drug after the full approval. So as a rare disease researcher or someone from that perspective, I would say that you shouldn't necessarily change anything or your behavior for these things, as frankly, most of what you're doing right now is still focused on that same goal. And it's a question as to whether or not we're going to see folks catching up on the government side and reinstating that opportunity. But I would never be counting it chicken before it hatches, which what that priority review voucher always has been, no matter when it was instated or not. I don't know, Clayton and Dave, if you have a similar perspective here.
Yeah, I fully agree. I mean, it's always been sort of a nice to have thing that, you know, we could always put that when you're trying to get, you know, board approval and move things ahead, you can always put that PRV as sort of a carrot hanging that, hey, you know, we get this and we get it fully licensed, we would have this potential to have that additional income. But I agree with you, Andrew, that you shouldn't, as a researcher, you shouldn't change anything, because that's never been the driver, at least where in my experience, it has not been a driver. So.
Yeah, I mean, I hope they reauthorize it. I think it's an important incentive, to be completely honest. It just seems to me that drug development is a really expensive thing. A lot of rare disease drug development occurs in small cap companies. I think you're just potentially taking away one of the legs of drug development in rare diseases if it's not reauthorized. So while I totally agree, I don't think people start off thinking, hey, the PRV is going to be the great way. I think that you burn cash, and if you get to the finish line, being able to actually have an immediate capitalization, selling it's expensive too, and for a small cap company trying to put together a commercial organization is a very difficult thing to do. So it's a great question. I just really hope it's reauthorized because I think it's really an important incentive.
For folks that might not be familiar, the priority review voucher is something that's transferable. So your program itself gets accelerated review because of the designation you have. But on top of that, you effectively get a golden ticket. And that golden ticket, this might sound silly, but it effectively is a fast track to getting all your documents reviewed when you're submitting for licensure of a drug. So you can imagine folks want to apply that if they're coming in with, say, a generic or a Me Too, because that moves them forward by about Clayton, Dave, like 9 months-ish, kind of a speed up there, which can be huge for those folks. So as a result, there's an opportunity to sell that voucher, and you can imagine the value there is going to be dependent on speeding those things up. I will note that there are folks that are still effectively, when they're working on deals or looking at licensing terms, even right now, still including the priority review voucher or talking about what they would be doing with it should it come back in. So I think if we just keep saying that it will and keep pushing, and I think this is also a place where the voices from rare disease patient communities are really important in lobbying, maybe we get back into that world, because frankly, it does not cost taxpayers anything on this. It's actually a pure incentive to focus efforts. I'll note there's also a priority review voucher. There used to be for what we called rare and neglected diseases, which included tropical diseases as well, and things that usually don't get investment, as well as anti-infectives. So a lot of things that we want to see people actually working on.
Thanks, Andrew. So I see one more maybe question in the Q&A box. What about Section 525 as a pathway? Are there thoughts on the Montana legislation for market access on phase 1 data as long as it's manufactured in Montana? And it'd be great if you could start off maybe with a brief description of Section 525 as well.
I could try to start further back than that, which is like, what is the FDA and why does it have powers, right? So the FDA has all of its powers because they've been given to it by Congress, because Congress has the ability to legislate and control interstate commerce. So that's where FDA's authorities come from, because you cannot market, which is to say sell a drug, if the FDA has not said okay, which means really cross state lines. So Section 525, or what Montana's put forward, actually isn't new, because technically, if you had done something always internal to your state, you could have gotten out of this because you're not doing interstate commerce. Now, with that, in reality, there are other ways that can get pushed down or locked down, and saying to what degree is there interstate commerce that resulted in this drug occurring. I would say overall, the most important thing is to recognize the FDA is really the gold standard of how drug development and regulatory science have occurred to date. Many, if not all, of the regulatory bodies across the world model themselves after what the FDA has done, move forward. I think it's important to think about how to work inside of that. I'll note that there are different forms of expanded access programs and others that can be used for drugs that are in clinical trial with existing guidance that is outlined. So I think that the Montana legislation that was put forward is doing what it's trying to do, which is signal this should be part of a conversation. I don't think it materially changes what folks in drug development would be doing. I think there's a lot of risk and uncertainty about how one would get clamped down on there. That being said, it is important that we recognize why this was passed, because people want to see opportunities for folks that are sick, that don't have access to medication, which that's the part we should be listening to out loud. I don't know, Clayton and Dave, if you have, if you've been in meetings on 525 or other things like this as well.
No, I'm glad you're here. It doesn't really, it doesn't really drive decision making for what we do. I mean, honestly, in rare diseases, it is really it's a globe. I mean, it's a global problem, right? And so you're frequently leveraging regulatory access, but it's really a find the patient kind of a problem. So these sorts of things for us are much more about trying to find and leverage worldwide expertise.
Mm-hmm.
Yeah, I agree with Dave. I'm glad Andrew was here. And it sounds like you've thought about it much more deeply than
I've
we
got
have.
I've gotten the question. I've gotten the question a number of times.
Yeah. Well, we'll move on to our next question. This is around regulatory expectations for evidence, right? Particularly when you're considering like first-in-class therapies for these rare diseases. Are there shifting expectations for what this evidence should be? Like we're hearing about biomarker validation, natural history study data, incremental sort of changes. What are your thoughts on how the landscape is shifting around us, or maybe what it should be?
Go ahead. I was going to say, I think this FDA is a little bit inscrutable, and so there's just a little bit. We just have to wait and see. I think that's an easy excuse for us drug developers. There's a degree of just being really careful and being very scientific and methodical and presenting good, strong, well-validated data sets to regulatory authorities. It really does drive the discussion to much better places if you're prepared and you've done good work. And I also think patient advocacy groups have a really important role in this discussion as well with regulatory agencies. I think that in rare, we do have the ability to leverage these sorts of early evidence of efficacy, biomarkers, clinical surrogates. But the agency's careful in terms of the way they use these tools, and so that's why we have to be thoughtful in terms of the way we present them to the agency. And in our experience in the past, in terms of drug development, my experience in the past, we were able to use phenylalanine levels to approve drugs in PKU. We were able to use factor VIII levels to approve gene therapy for hemophilia A. And that's largely because the body of work that substantiated the relationships of these variables to clinical outcomes was extremely strong. And so I think, with the creatine disorders, that's the challenge in front of us, right? And that's, we spent a couple of days thinking about how we can relate these kind of like really clear, measurable observational differences in patients to something that feels, functions, and survives that the regulatory agencies are going to hold on to. So I think I think that you need to be loaded for bear when you have these discussions. You need to have strong data sets. You need to believe your data. You need to get as many allies in the room as humanly possible. It needs to really have a great scientific story. You need to, I think, really land with 2 feet in terms of why that target engagement and mechanistic information is really valuable to the patient. So I think it is sort of a brand new world, but it's one that we should view as an opportunity, and one that we should try and leverage with the best quality data as we can going forward.
Yeah, I totally agree with Dave. I do the idea of being able to support approval with biomarkers is certainly more of a reality now than it's been in years past. And I think groups like this are what are making that possible, this understanding how to pull together. And when I say groups, I mean patient support groups like ACD, but also all of these people, everyone who's spoken today, all the researchers who are looking at what correlates with improved outcomes. Jerry talking about imaging, a lot of people talking about imaging, and blood markers, and really all of that coming together to help support that. If we see, this, whatever this is, this biomarker change, we can support that down the road that is going to lead to better cognition or better state, because that is one of the difficult things in rare disease, that some of these changes we want to see are going to take a while, years to develop, and it's also hard to sort those out away from the therapy and from the current standard of care. I think we heard somebody mention, or multiple people mention, yes, we changed this medication or we changed this and we saw this improvement, but we also don't know if that improvement was because of the speech and occupational therapy that our child was getting. So the more like Dave said, the more science we bring, the better off we are to be able to show that the change we're seeing biochemically or through imaging is supported by science, that this is going to lead to an improvement down the road.
I'm curious.
We spoke a little, sorry, go ahead, Heidi.
I was just going to say, we are an association of creatine deficiencies. We know very well and represent 3 different disorders. And I've always thought, we have such a great example with these patients with GAMT who receive creatine for the first time. They have a measurable increase of creatine in an MRS of their brain, in their plasma, and they have very good improvements. And I wonder if there is another example of seeing a biomarker move in a disorder, justifying that this is a valuable biomarker, and then applying that in another disorder, because it's a really tricky thing, I think, for a rare disease to say, you need to first prove that changing this biomarker is important. Well, that's the whole point of trying to find a treatment. We don't have a CTD patient per se, maybe 1 or 2 here or there that have a partially functioning, mutation. They've done the treatment, and we think that it helped. But we're saying treat them and then prove that the treatment works. But we're in a tough spot. And what do you think about this evidence with GAMT and applying it to CTD? What's your impression there?
I think you hit something that is rarely appreciated, or at least stated out loud, which is, I think that developing biomarkers and being able to prove that they're working is in many cases harder than developing the initial chemical matter that becomes the drug. And that's because being able to show an improvement off of metrics that have never existed before in a system that's never existed before in a patient with their own their own polypharmacy, in some cases, is complicated. I think, to Dave's point and Clayton's point, starting from the same way that we position and put together evidence for bringing a drug into clinic, which starts always with, what is the biological rationale? Why does this make sense? It's how you don't get into that argument that this glass of water will be the drug, but rather, what is the biological rationale behind it? With pathways like this that are really well known, I think you're in a better position to start to do that and try to push. Now, whether or not that's listened to, or how to make sure that people are listening to that, I think that requires collaboration and making sure that those voices are heard. I was going to link it, but I don't know how to do that in the chat. There's a really wonderful post from the FDA about patient-focused drug development. And there's a whole guidance set on this of how to collect those data, how to have those conversations, how to move things forward. And a lot of times the thing I've seen across other diseases, maybe Clayton, Dave can speak to this as well, is it comes out of conferences like these ones, where there is a working group or a working paper coming out of this saying, we believe that these things make sense. We believe that these things can connect. And it becomes both a call to research as well as a solidification of that thesis that then people can beat up on. And it becomes part of the scientific process. I think that helps push folks forward. It has make sure that dissenting voices have a view and an opportunity there, because it'd be very dangerous if we didn't listen to folks that said, oh, I don't know, or here are cases where it doesn't, because we could be walking down a path where it isn't necessarily predictive. I don't know, and maybe Dave Clayton, I don't know if there is a disease I could point to right now that says they ported over this biomarker or the other, but in many cases it is pointing to basic physiology we do understand and then building up from there, which I think that this community's done a lot of work in.
I don't know of an example. I know that when we were developing medicine for PKU, there's a huge amount of literature on the sort of the toxic relationship of hyperphenylalaninemia to all of the developmental changes that occur in kids. But we don't really have pediatric PKU anymore. And we had to make the argument that the neurocognitive problems that adult PKU patients had were also due to the toxic effects of hyperphenylalaninemia. And it was not the easiest discussion in the entire world. However, I think GAMT actually is highly leverageable in terms of thinking about validating biomarkers in the disease or treatment effect in the disease as well. I think that we always have a harder time when we're trying to use a transgenic four-legged creature to justify our sort of mechanism of action. And I think the fact that if we have two-legged creatures that have natural history and a disease course that is relatable to the interaction that we're going to plan in terms of our medicine, I think it's much, much better. It's an easier conversation to have. But, you know, the FDA, they are plausible skeptics. I mean, that's their job, and that's what they're supposed to be doing. So it really is incumbent on us to put forward the best supported argument for going forward with expedited review.
That's a really good point. There's an expectation that you are the expert. And as a result, there's also like, they listen. So when you say, this is why we think this makes sense, they listen and they're going to push back on it. But if you continue to have that evidence, they'll say, okay.
Did you have a comment before we wrap up?
I was just going to say that, I don't know of any crossovers that have actually happened, but I do know, one of our gene therapies is in the field of leukodystrophies, and in leukodystrophies, as you lose brain myelin, there are some biomarkers that become apparent, and those biomarkers change if you are able to stop that demyelination. So it is a very hot topic among researchers and drug developers in the leukodystrophy field of trying to find a biomarker that is sort of just a marker in general that could be used across multiple rare leukodystrophies. So I do think it's something that we should continue to work towards.
Okay, well, I think we are at time, and thank you all for participating. Thank you everybody in the audience for your comments and questions and attendance today. And Sangeeta has a very brief wrap up with the responses from the poll questions that I've peeked at them. They're very interesting. So hang on with us for about 2 more minutes, and I'll turn it over to Sangeeta. And thank you again, everyone.
Great, thank you.
Okay, I'll get us moving. Thanks for sticking with us for the close. Okay, review of conference poll results. Our first question was around whether patient samples have been utilized in your research. An overwhelming 68% have used this, and I hope that this proves to be more of a boost for those of our community members who have not yet donated a patient sample. Please do so. They're incredibly valuable in research. And it's just another way to make your representation count. Get in touch with us if you'd like ideas on how to donate a patient sample. Poll number 2, or no, part of poll number 1, question 2. Would natural history data be helpful to you in understanding CCDS? And a large number of researchers said yes. And I think this really underscores, the fact that, of course, we have Vigilon that was done for CTD, and the publication is expected soon. But this really also maybe is a signal for us to conduct a GAMT natural history study, as well as an AGAT one. I know that AGAT patients will be hard to find, but we have to put the call out for both of these diseases and really encourage a natural history study to get going. How important would a tool antibody against SLC6A8 be to you? We have a number of gene therapies associated with SLC6A8 in development, and I think, again, 73% researchers indicated that it would be incredibly important. Thank you for this. This means that we will be investing some efforts into making this antibody available. Poll number 2 from today: how helpful would patient-derived iPSCs be to your work? This is interesting. I thought there'd be more appetite. It feels like we're split down the middle. 20% say it would be very useful, but the remaining are maybe it is just representative of where they are in their research. But something that we will keep an eye out on. I'll confess I was surprised by this. Next up, would mass spec core facilities for detection of creatine and creatine analogs be useful to your work? 86.2% indicated that this would, and thank you for this. Again, this is something that we as ACD will look into. I know there were, we heard about assay development of different kinds, colorimetric, spectrophotometric assay development, but it seems like a mass spec-based method is highly sensitive, and there might be some use to identifying a core facility that could standardize methods that could be used across maybe samples and the like for detection of creatine and creatine analogs. If any of your universities specializes in this, and you know of their willingness to maybe work with outside entities, please get in touch with us so we can evaluate possibilities there. Poll number 3, which of the following is not a part of the core outcome set? We published this paper not too long ago, and one of the elements that was not included is the caregiver burden. We also heard today from Judy and Audrey that they will be rolling out a survey associated with this, and I think this is all tracking very nicely together in terms of identifying areas of need. Poll number 3. How many prescription drugs is your child with GAMT or CTD or AGAT taking? And 37.5% indicate that they're taking more than 4 drugs. And again, interestingly, an equal percentage said they take no drugs. And 25% indicate they are on 2 to 3 drugs. Interesting finding. We'll be diving into this a little bit more through our registry surveys. Poll number 4, would you collect and donate dried blood spots for your research study? 47% said yes. And I imagine a number of researchers said no. 40.4 must be the researchers, and 12.4% said that they're unsure or on the fence. And it's great to hear that there's so many folks who are willing to donate a dried blood spot for research study. We know of efforts that are going to be initiated imminently between Dr. Pasquale and Dr. Schulze, and this is very heartening to see. We do need better detection methods, well, maybe not detection, better diagnosis, particularly of CTD. Having a method to diagnose CTD and a method to more accurately measure GAA on an ongoing basis are all areas of need. And we're hoping that we can address these through our studies, but your willingness to participate is key. So thank you for that. Do you feel that reducing GAA below the currently achieved levels would create better outcomes for GAMT patients? 69% of responders felt that it would. Highly interesting and something for us to focus on. Okay, that brings us to upcoming events. This is now bringing us into 2026. Please save the date. We are announcing our Walk for Strength on July 25th, 2026. We'll send out a lot of reminders before then, but I hope for those of you who participated in this year's walk, please do so again next year. For those of you who are looking to get involved, this is a great way. Find out if a walk is happening in your city. Okay, working backwards, we also have our in-person conference next year, and this will be held in Utah on June 25th to 28th. Please do save the date, and it'll be a different kind of experience because we'll be interacting in person. Be sure to attend that. We'd love to see you in person and not just on a two-dimensional screen. Okay, gives me great pleasure to announce this initiative by ACD in collaboration with our board member, Jeffrey Allen. We are announcing this documentary, Life of Lucas. Documentary will be played in 6 different cities starting this September. These are the cities that are the planned screening is on docket for. We'd love for you to stay and watch this trailer that I'm going to play next. And if you have interest in hosting this documentary in your city, please do get in touch with us. It is a great way to raise awareness for CTD and cerebral creatine deficiency syndromes in general. So with that, I will play this trailer for us.
We are not hearing the sound, Sangeeta. Sangeeta, we're not hearing the sound. I think there is a setting.
I knew I wanted to be a dad. I didn't know when, I didn't know what it would look like, but I knew I wanted to be a dad. We have 2 amazing boys, Jack and Lucas.
When I can get myself out of the way enough, he will light up my world, and I will forget how hard it can be.
And kind of everybody had the common theme that he is developmentally delayed.
The disease doesn't show itself at birth. It's as the child is developing that parents will notice deficits in the development of the child.
Creatine deficiencies are caused by a genetic mutation. Most parents, like myself, first time they hear about these disorders is when they're getting a diagnosis.
As the months went on, we got more and more concerned. We called in, I feel like, every favor we could to get into the right specialist. Who was diagnosed with Creatine Transporter Deficiency, this ultra-rare brain disorder.
Having missed 7 calls from my pediatrician, and she goes, well, I've never heard of this.
There's no cure. There's no therapy. What do we do? I got scared.
It is highly likely, until we find a treatment, that Lukey will be with us for his entire life. There's less than 500 globally diagnosed, though it's probably severely underdiagnosed.
We'll generally have developmental deficiencies, difficulties in communication, in learning, in speech.
He's a smart boy. I feel it, I see it. He has a hard time communicating. You know, he's not yet potty trained. I forget that he's 7. It happens every day, where he is upset, he's angry, he'll throw tantrums. He can get physical.
Whenever he's in a good mood and I play with him, I usually have fun.
One really exciting thing about creatine deficiencies, a single gene mutation is responsible. When you know what gene is causing a disease, it gives you a really good chance of solving the disease.
But I know that Jen and I and ACD can't do it by ourselves. We need your help. By simply sharing this film, you can help raise awareness.
We make people aware of the disease so that we can get the funding we need to progress the programs to the point where drugs are ready for clinical development.
We have 6 active gene therapy research projects going on right now with really promising results.
I am confident we will fund and find a cure.
With all the resources and technology that we have, there's no chance that there's a future where we don't figure this stuff out.
There is a lot of hope in coming up with a treatment for this chronic disease.
And you can make a difference by supporting this cause. We will find a cure for creatine deficiencies.
Well, I have watched that trailer many times, but it still gets me each time. So I hope that that video left you with a feeling of urgency, with a feeling of inspiration, with a feeling of hope, and hopefully that on the heels of the past 2 days gives you that urgency, that momentum, the need to get back in the lab to go and do more studies, the need to find a faster and better way to get these get the drugs that are in discovery into later stages of development and into people, to find something that's safe and effective, and to find something that can truly change the lives of those impacted by CCDS. So with that, thank you for staying with us throughout these past 2 days. It means the world to us to have your support, both researchers as well as the researchers, clinicians, industry stakeholders, as well as our community of wonderful parents logging in from so many different parts of the world. Thank you very much for making this virtual conference a resounding success. We loved your participation, and we hope to see you in person soon before next year's conference. If there's an opportunity for us to see you in person through a screening in your city for this documentary, we'd love that. Please spread the word, get in touch with us if you'd like to learn more. And with that, my cat needs my attention, so I'm gonna log off.
Thank you, everyone.
Thank you.